Chapter 10. The Action of Various Salts. Protoplasm contains salts, and these salts play an important role in the life of the cell. So much is common knowledge. There can be no doubt but that certain salts or ions are essential for life. These salts or ions must be present within definite limits of concentration, and there must moreover be a balance between the concentration of one ion and another. Because of the obvious importance of the protoplasmic salts, and because of the relative simplicity of these constituents, physiologists have very frequently studied the effect of the more common salts on various aspects of the life of the cell. In studying the action of specific salts or ions, it is of course essential to exclude other factors from participation in the experiment. In the first place, a salt must not be used in such concentration that it exerts an osmotic effect. The solution used for the experiment must neither be hypertonic nor hypotonic. It must neither extract water from the protoplasm or give up regarding the particular action of some salt from experiments in which cells or tissues were immersed in solutions, which exerted a decided osmotic action. It is not always a simple matter to decide just which strength of solution to use. Most workers assume that solutions which have the same freezing point are equal in their osmotic effects. But this could only be true for an ideal semi-permeable membrane, a membrane which is permeable for the solvent. In this discussion, water, and impermeable for all dissolved substances, no such membrane is known. Certainly, the osmotic membranes of cells show differences in permeability toward various types of salts and other dissolved substances. Hence, a theoretically isosmotic solution may in actual practice extract water from the cell, or it may have the opposite effect. Indeed, it seems certain that other factors are involved in addition to a differential permeability, so that it is quite out of the question to know from available physical data just what strength of solution will be in osmotic equilibrium with a given type of cell. How then can one know what strength of solution to use? For some cells, it is possible to test the osmotic effect of various concentrations of any given substance merely by determining the volume of the CCU in different concentrations. That concentration which causes neither loss nor gain of water is osmotic. When cells have irregular shapes, it is not always easy to determine their volume. Such cells are not to be recommended for experiments with salts, for it is impossible to exclude osmotic factors. Most plant cells are surrounded by a stiff wall, which prevents any marked increase in volume. The only action of a H3 protonic solution on these cells is to cause an increase in the pressure exerted on the protoplasm by the cell wall. With the ordinary type of plant tissue, the osmotic factor is probably unimportant as long as hypotonic solutions are used. Perhaps also hypotonic solutions may safely be used in experiments with fresh water protozoa. A second factor which must not be disregarded is the hydrogen ion concentration. The older experiments on the action of salts took no cognizance of this factor. Colloid chemists have repeatedly emphasized the importance of the hydrogen ion concentration in determining the behavior of various colloidal solutions. Nor have biologists been slow to realize that the concentration of hydrogen ions is a variable of great importance to life phenomena. Hence, if we are to study the action of salts, we must be certain that our solutions either have the same pH as the normal medium, or that they have a pH which can be shown to have no marked effect on the protoplasm. In studying an inanimate colloid, it is only necessary to know the pH of the solution, that is to say, of the dispersion medium. But when we immerse living cells in various solutions, we are interested not so much in the pH of the medium surrounding the cells as in the pH of the protoplasm itself. In an inanimate colloid, the pH is what we measure it to be. But when cells are placed in different solutions of the same pH, the hydrogen ion concentration of the cell interior may be very different. Consider, for example, 2,2-O solutions, in which the pH is determined primarily by carbonic acid. Let us suppose that both solutions have the same pH, but that one of the solutions contains carbonates, whereas the other does not, as a result of the law of mass action. It can readily be shown that the solution containing carbonates has a much higher concentration of carbonic acid than the solution without carbonates, in spite of the fact that it has the same pH. Because of such a higher concentration of carbonic acid, far more acid diffuses into the cell. The two solutions are therefore not comparable, even though at first sight they might appear to be. In experiments with cells, it is not always sufficient to keep the pH constant. Some attention should also be paid to the source of the pH. In studying the action of salts, it is necessary to use osmotic solutions and solutions whose pH is not likely to have an effect on the CE protoplasm. But even when we use such solutions, there is another difficulty which it is almost impossible to circumvent. In most instances, we do not know how rapidly a salt enters a cell, and there is even some question as to whether salts enter at all. The weight of the permeability evidence certainly favors the idea that salts do diffuse into cells, but there is every reason to suppose that some salts enter more rapidly than others. If we compare the action of two salts, any differences that we observe be due to a difference in the permeability. In general, this does not seem to be possible in those cases in which the two salts produce opposite effects within the cell. But in those cases in which the difference in the action of the two salts is a difference of degree, we cannot very well exclude the permeability factor. From what has gone before, it is obvious that the greatest caution must be exercised in interpreting the results of experiments with salts. The conditions of experimentation should be such as to preclude osmotic effects or effects due to a difference in hydrogen ion concentration. And we must always recognize that the speed with which various salts enter the cells is a factor to be considered. Many types of protoplasm are very sensitive to salts of heavy metals. This fact was first emphasized by Nageli, who found that Asphaerogyra cells were killed by copper and mercury salts in extreme dilution. Since Nageli's time, it has been customary to speak of this action of substances in extremely small concentration as an oligodynamic action. In recent years, there seems to have been a revival of interest in the phenomenon of action. References both to new and older literature may be found in the papers of Sissybold 27 and Ludwig 27. From data collected by Ludwig, it appears that paramecium is killed by copper sulfate and mercuric greater than 10 mol. Asphaerogyra is even more sensitive and is in view of the fact that salts of heavy metals coagulate, same effect in vivo. By actual viscosity tests, it can readily be shown that low concentrations of HECl produce a coagulation, or at least a great increase in viscosity, in various types of protoplasm. Arbacea eggs treated with M10-HGClAg in seawater very soon become altered so that the granules of the cells can no longer be moved by centrifugal force. The same experiment can also be performed on protozoan cells. When the flagellae euglena is centrifuged, it loses its spindle-shaped contour and becomes spherical, the granular inclusions massing at one end. But when a small amount of mercuric chloride solution is added to the culture medium in which the euglena lives, no shifting of granules occurs when the protozoa are centrifuged. In both euglena and arbacea cells, a great increase in viscosity follows treatment with mercuric chloride. The protoplasm ceases to behave as a fluid and is a gel or coagulum. The coagulative action of mercuric chloride, at any rate in higher concentrations, apparently involves the hydroprecipitation of proteins from the hyaline ground substance, that is to say, the intergranular material of the protoplasm. Not only is this a logical assumption, but there is also actual experimental evidence in its favor. When Arbacea eggs are first centrifuged and are then treated with dilute solutions of mercuric chloride in seawater, new granules can be seen to appear in the hyaline region previously free from granules. In very dilute solutions of mercuric chloride in seawater, there can, of course, be no question of an osmotic effect. Moreover, these solutions have a pH very close to that of seawater. Other solutions of metallic salts are frequently acid. Thus, copper sulfate and copper chloride hydrolyze in aqueous solution so that there is always some sulfuric or hydrochloric acid present. But it is easy to show that the copper ion has a specific effect quite apart from any acidity that may be present in solutions of its salts. Dilutions of copper chloride, which have little or no acidity, may have a very pronounced action on the protoplasm. Thus, in one of a series of unpublished experiments, the following 5 solutions of copper chloride in seawater were prepared. M10, solution W as dilute enough, so that there was practically no effect of the hydrogen ion concentration. The pH of this solution was 7.8. All of the solutions produced a coagulative effect on the protoplasm of sea urchin eggs, as was clearly shown by centrifuge tests. This coagulation did not occur very rapidly. Thus, a test of the eggs in the M50 solution showed no coagulation and no increase in viscosity after a 20-minute exposure, whereas a test after 54 minutes did show coagulation. For some protoplasm, the copper ion apparently does not exert so pronounced a coagulative effect. Claim 95 placed leaves of various aquatic plants in a 10% copper sulfate solution. The streaming of the protoplasm was not interrupted, even though enough of the copper salt diffused into the coals to distinctly color the cell sap. Compare also Pinning-Shanghai 24. The action of the ions of heavy metals on protoplasm, as far as it is known, seems easy to understand. Apparently metallic ions, literally those of mercury and copper, cause a precipitation of the proteins of the cell, although perhaps this is not so certain in cases in which the metals are only present in very great dilution. The behavior of protoplasm towards salts of the alkaline and alkaline earth metals presents a more difficult problem. These ions have no very great effect on proteins, but they have a profound influence on practically all types of living substance. Over and over again, workers with both plant and animal material have found that solutions of sodium chloride alone or potassium of calcium or magnesium chloride is sufficient to offset the toxicity of the sodium or potassium ion. Often, too, though not always, the addition of sodium or potassium ions is necessary to save the life of the tissue or organism. It is not our intention here to consider the vast literature on the subject of these ion antagonisms. Some of this literature has been summarized in a recent book of Zondek 27, and many details will be found in the last edition of Hobeb 26. The review of Rub Kiestein 28 may also be consulted. Not only have physiologists studied the action of vabious salts of sodium, calcium, and other ions on the life and death of various tissues, they have also made numerous experiments to show that in a wide variety of biological processes, the physiological behavior is strongly influenced by the presence of one or the other of the ions in question. To discuss adequately the large and often contradictory literature on the antagonistic action of the salts of the alkaline and alkaline earth metal, we shall confine our attention almost exclusively to the studies that deal with the effects of the ions of these metals on the colloidal state of the protoplasm. Here, too, our task will not be simple, not only because of the intrinsic difficulties of the subject, Itziak, but because also the subject has now and again been attacked by biologists with no training in the elements of physical chemistry. Thus, not infrequently, one finds comparisons between the effects of equimolar solutions of calcium and dissociates into 3 ions instead of 2, and therefore has a theoretical osmotic pressure roughly 1.5 times as great as the sodium chloride solution, when both are present in equimolar solution. Because of the unsatisfactory state of the literature, we shall not attempt any chronological survey, but instead we shall present a thesis and see how it fits the facts. In view of the pronounced action of the alkaline and anomalous earth metals on all types of living substance, it seems logical to assume that the ions of these metals have definite effects on the colloidal properties of the protoplasmic coat. That this should be true is readily understandable if we remember that protoplasm is a suspension, CP 21. Colloidal solutions of proteins are on the W whole very insensitive to the ions of sodium, calcium, etc., whereas on the other hand, lyophobe colloids and suspensions are very sensitive to these ions. The stability of a suspension depends on its electric charge, and this charge is primarily due to the adsorption of ions. If a negatively charged succinylchoid or succinylcholine is placed in contact with a suis of bivalent cations, these are soon adsorbed. The charge on the colloidal particles is neutralized and precipitation or coagulation follows. So too, a positively charged suspension is readily precipitated by bivalent or trivalent anions. These facts are of course well known, and further details will be found in almost any hook on colloid chemistry. Consider now the protoplasmic suspension. We have every reason to believe that it remains stable because of an electric charge. The fact that in living cells, generally bivalent cations are more abundant, i.e. bivalent anions, is an indication that the charge on the protoplasmic colloid is positive. Normally, the protoplasmic colloid is in equilibrium, both with monovalent and with bivalent cations. If now the percentage of bivalent cations were to be increased, obviously the positive charge, if there is a positive charge, which was put. On the other hand, if the percentage of bivalent cations were decreased and their place taken by monovalent cations, we should expect a decrease in the charge of the protoplasmic colloid with perhaps a complete neutralization of charge. Should such a neutralization of charge occur, the protoplasmic suspension would lose its stability and precipitation or coagulation would be the result. The point of view that we have been considering is in the main that proposed by Heilbrunn in Lem 22. In the present chapter, no attempt will be made to discuss all the evidence which points toward a positive charge of the protoplasmic succedention. This will be done in the following chapter, which deals with the electrical properties of protoplasm. If, however, we assume a positively charged protoplasmic colloid, we are in a position to understand the actual facts in regard to the effect of sodium and calcium ions on the physical state of the protoplasm. Heilbrunn 23 studied the effect of calcium, magnesium, the eggs of the sea urchin Arbacia. The eggs were GDAC in solutions of the chlorides of these metals. In these solutions, the hydrogen ion concentration was carefully controlled, and the concentration of the salt was so adjusted that the solution was isosmotic with the eggs. That is to say, the solutions used caused neither a shrinkage or an increase in volume of the coals. This does not mean that the solutions had the same freezing point. A solution of calcium chloride, which produces the same freezing point, lowering as a solution of sodium chloride, is demonstrably stronger osmotically with reference to the semipermeable membrane of the Arbacia egg. Whether this is due to a greater permeability of the sodium ion or not does not concern the action of various salts present discussion. The point we wish to emphasize is that in the experiments of Heilbrunn, solutions were used which had no osmotic action on the cell. In the presence of an increased proportion of the calcium ion, or in osmotio calcium chloride solutions, the protoplasm of the Arbacia egg becomes more fluid. This was clearly shown by centrifuge tests. The magnesium ion produces a similar effect, but its action is not so strong, and the change in viscosity is relatively slight. Potassium, sodium, and ammonium ions cause an increase in the viscosity of the protoplasm. This effect becomes much more pronounced when the eggs are washed relatively free from the seawater in which they occur normally. Even traces of seawater markedly retard the coagulative action of EA3, potassium, greater adsorbability of the bivalent magnesium and calcium ions. If one arranges the cations in the order of their effect in decreasing the viscosity of the protoplasmic suspension in the interior of Arbacia eggs, one obtains the following series. This order is exactly the order of decreasing adsorbability, see books on colloid chemistry. It is thus apparent that the greater the amount of cation adsorbed, the more fluid is the protoplasm of the Arbacia egg. Some of the results of Heilbrunn 23 have been redated in subsequent unpublished experiments. Compare Heilbrunn 25c. Particular attention has been paid to the action of calcium. There can be no question but that the calcium ion causes a decrease in the viscosity of the protoplasm, but when Arbacia eggs are kept in solutions of isotonic calcium chloride for a long time, a secondary change often occurs. This involves a loss of pigment from the pigment granules, and eventually the egg undergoes a change, which by embryologists is usually called cytolysis. As a result of these transformations, the egg becomes coagulated. In later chapters 13-14, it will be shown that the loss of pigment from the pigment granules, or the breakdown of these granules in the Arbacia egg, always initiates a complicated series of reactions which result in the production of numerous characteristic vacuoles in the protoplasm and a resultant complete coagulation. Apparently, the first stage of this series of reactions depends on a reaction between pigment granules and free calcium. When eggs are placed in calcium solutions, the calcium as it enters the egg is first bound or adsorbed. However, when enough calcium has entered a reaction with pigment granules occurs, and an entire series of changes follows. Curiously enough, it was found that the behavior of the eggs was very different depending on whether or not they were washed il sievate following removal from the ovary. If eggs were not washed and were placed directly from the ovary into calciumfaction of the protoplasm, if, however, they were thoroughly washed in seawater and adhered and placed in an isotonic calcium, so Colloid cytolytic changes to occur. The reason for this difference in behavior is not known. Perhaps a difference in permeability is responsible. Perhaps, as Heilbrunn 25C suggested, a difference in the electric charge of the egg membrane may play a role. The fact remains that the primary action of calcium on Arbacea egg protoplasm is to produce a decrease in viscosity. Experiments with the girotoplasm of the protozoan stentor gave similar results. In working with stentor, it is not such a simple matter to determine the concentrations of salt solution, which are exactly isomotic with the protoplasm. That is to say, cause neither decrease nor increase in the volume of the cell. This is due to the fact that the stentor cell is not spherical, and it is therefore impractical to determine its volume. In studying the action of various chlorides on stentor protoplasm, Heilbrunn 23 used concentrations of magnesium and calcium chloride of slightly lower freezing point than the solutions of sodium and potassium, that the calcium and magnesium solutions were, if anything, stronger osmotically than the sodium and potassium solutions. The solutions were so chosen poetically. In general, the more dilute a solution, the more it tends to decrease the viscosity of the protoplasm. In the experiments of Heilbrunn, it can hardly be assumed that the observed liquefying action of calcium chloride was due to the fact that it was present in lower osmotic concentration. In the work on stentor, sodium, potassium, lithium, and ammonium chloride were used MM32 concentration, and magnesium and calcium chloride were used in M40 concentration. The salts were dissolved in water redistilled from an apparatus made of fused silica and quartz. The pH of the ammonium, the other solutions about 8 dot. It was found as a result of centrifuge tests that sodium, potassium, ammonium, and lithium chlorides all cause coagulation of the protoplasm. In solutions of these salts, there was no movement of granules through the stentor cell, whereas in their normal culture fluid or in solutions of magnesium or calcium chloride, the granules moved into one half of the cell when the protozoa were centrifuged. A more rapid coagulative action of the monovalent cations was observed when the cells were first washed free of the culture fluid. This was obviously due to the fact that the culture fluid contained mixtures of ions. The relative effect of these ions on the viscosity of the protoplasm was measured. It was found that in the order of their liquefying or viscosity decreasing effect, the order of the ions was as follows. This order is practically the same as the one found for Arbacea protoplasm. The only difference is that the sodium and potassium ions have changed places. In view of the fact that the colloid chemists usually find no decided difference in the adsorption of sodium and potassium ions, this shift is of no importance. So far, we have considered the action of calcium, sodium, and other cations only on the interior protoplasm. The action of these ions on the surface membrane of the cell is quite different. The primary effect of calcium ions on the cell membrane of the Arbacea egg is to make it brittle. It probably decreases in thickness, but this cannot be observed under the microscope, for observations at the surfaces of cells are at best uncertain. That the membrane is actually more brittle following exposure to calcium can be shown by experiments in which the egg is subjected to gradual pressure from above. The eggs are placed in a small drojo of seawater on a slide, and then a large cover slip is placed over them. The distance between slide and cover slip decreases, and the eggs are gradually compressed under these conditions. Eggs in calcium chloride solution break through the membrane at lower pressures than those in sodium chloride. This is indicated by the fact that the sodium chloride eggs do not break until they have suffered considerably more distortion. The extent of the distortion is determined by measuring the diameter of compressed egg discs. Sodium and ammonium ions in the absence of calcium cause a visible change in the membrane of the Arbacea egg. the membrane absorbs water and swells. At the same time, it becomes sticky. The presence of calcium prevents this swelling of the membrane produced by sodium and ammonium ion. It should be noted that after long exposure to calcium, the membrane, which was at first brittle, also swells and becomes sticky. These effects of various cations on the membrane of the sea urchin egg were interpreted by Heilbrunn 2325 skip as indicating a negative charge of the colloidal particles of the membrane. It was thought that as the percentage of the calcium ion was increased, the charge on the colloidal particles diminished. This would then tend to cause closer age approximation of the particles and loss of water. Then, on longer exposure to the calcium ion, the charge on the colloidal particles might be reversed, and then the membrane might swell in calcium chloride. Although possibly this explanation may be correct, it is not felt that it rests on as sure ground as the explanation of the effects of the various cations on the interior protoplasm of the cell. And there is indeed some evidence that the colloidal particles of cell membranes are charged positively, see PISO. In the previous discussion, it has been assumed that the action of calcium, sodium, and other cations was due primarily to an effect on the electric charge of the protoplasmic colloid. If this were the case, it is evident that trivalent cations SLILed have a far more pronounced action than either calcium or magnesium ions. In experiments with inanimate colloids, it has often been shown that trivalent ions are perhaps thousands of times as effective as bivalent cations in altering the charge of suspensions and suspensoids. It became important, therefore, to discover whether or not trivalent cations had a marked effect on protoplasmic viscosity, and whether this effect was in accord with expectation. The first experiments with aluminum salts met with no success. The addition of aluminum salts to a solution always produces a marked acidity, and this acidity is sufficient to cause coagulation of the protoplasm. As a matter of fact, practical why all of the aluminum experiments in the literature of plant and animal physiology are open to the objection that the results obtained were due more to the hydrogen ion than to the aluminum ion. Attempts were made to neutralize the acidity due to the hydrolysis of aluminum sulfide. Such neutralizations, when they were made in the presence of seawater, always led to an abundant precipitation. The neutral or nearly neutral solutions had no effect on the protoplasm, almost certainly because it was discovered that if small amounts of aluminum chloride were added to solutions of pure sodium chloride rather than to seawater, the solution could be brought to a pH of 6 or more without precipitation. A pH of 6 is without any observable effect on the protoplasmic viscosity, so that it is possible to work under these conditions. Why there is no precipitation of the occurs in the presence of seawater is not certain. Sodium aluminate is soluble and calcium aluminate is very insoluble, but one would hardly expect any formation of aluminates at a pH. And perhaps this double salt formation is the important factor. It is hardly necessary to go into the chemical reasons one way or the other. The fact remains that in the presence of only sodium, this fact makes it possible to work with aluminum salts with the knowledge that one is studying only the effect of the aluminum ion and not the combined effect of aluminum and hydrogen ions. Once the method of work was discovered, results were easy to obtain. It was soon found that very dilute solutions of aluminum chloride produced a very marked effect on the viscosity of the protoplasm of the Arbacia egg. Even a M25 solution in isotonic sodium chloride solution was sufficient to cause a very pronounced decrease in the viscosity of the protoplasm. Solutions of cerium chloride behaved in the same way as those of aluminum chloride, except that somewhat higher concentrations had to be used to produce the same result. The primary effect of aluminum and cerium is thus to cause a decrease in the viscosity of the protoplasmic suspension. However, after long exposure, especially if the eggs have been washed in seawater before being placed in contact with the solutions containing aluminum or cerium, a secondary effect occurs. This is first indicated by a change in the appearance of the outer membrane of the egg. This membrane is at first brittle, but later it may absorb water, swell, and become sticky. As soon as this happens, there is usually a coagulation within the egg interior. The account here given follows in general the preliminary note of Heilbrunn 25C perhaps. At some time in the future, a more complete record of the experiments will be published. From what has been said, however, it must be clear that the action of trivalent cations is in accord with the proposed theory. The first effect is certainly a decrease in viscosity. As the trivalent ions enter the protoplasm slowly, they are probably absorbed at the surfaces of the dispersed phase of the suspension, and this results in an increase in electric charge and a decrease in viscosity. But when the outer membrane of the egg is changed, the trivalent ions may enter too rapidly, and they may then have an effect on the proteins of the disperse on medium. The experiments of Heilbrunn show clearly that in Arbacea protoplasm and in C7er, various cations act in a manner exactly comparable to the way they act an inanimate suspension. There are complications, it is true, but the main facts seem plain enough. In general, it is our claim that, aside from secondary reactions, calcium and magnesium tend to cause a decrease in protoplasmic viscosity as compared with potassium, sodium, and ammonium. We shall now proceed to see whether or not this point of view finds support in the literature. Perhaps the most striking evidence that sodium and potassium ions cause a coagulation of the protoplasmic suspension is found in the observations of Esnikov and Chambers, 25 in Chambers, and Rezhikove 26. Those authors immersed amoebae in solutions of sodium chloride and potassium chloride, and they found that within relatively few minutes, the granules of the amoeba protoplasm clumped together and sank through the cell. The picture that is described and figured by these authors apparently is a typical coagulation picture, and yet they interpret their results as showing a decreased viscosity or a liquefaction of the protoplasm. Surely, when particles of a suspension aggregate and then fall, this is not due to a change in the viscosity of the dispersion medium. It is due to the fact that, in accordance with Stokes' law, the speed of fall of a spherical body through a fluid varies as the square of the radius. As the particles of an inanimate suspension, or as the granules of amoeba, come together, they form a larger aggregate, and this, because of its greater size, falls rapidly. From Stokes' law, it can be shown that, on any assumption as to the magnitude of the absolute viscosity of the protoplasm of the amoeba, on any reasonable assumption as to the magnitude of the specific gravity of the granules, these granules of the amoeba cell could not possibly fall at even approximately the rate observed by Chambers and Rezhikove, if they fell singly. Indeed, these authors describe a clumping of granules, and the clump remains at the bottom of the CC for an indefinite period of time. In all the literature on the colloid chemistry of protoplasm, there is perhaps no clearer case of coagulation. That sodium and potassium produce coagulation of amoeba protoplasm is also indicated by the fact that these ions are far more toxic than the ions of calcium and magnesium. Thus, according to Chambers and Rezhnikov, 25. Amoebae will live for 24 hours in MS6.5 magnesium chloride and for about 5 hours in M6.5 calcium chloride, whereas they die within an hour in M6.5 sodium chloride. And they die within an hour in solutions of CoA as dilute as M104, whenever death occurs in solutions of sodium or potassium salts. This death is accompanied by a clumping and sinking of granules. Amoebae will live for 3 days or more in solutions of M13 MRGC or M26 K-col, so that it seems probable that in solutions weak enough to be without osmotic effect, amoeba will live indefinitely in pure calcium, or Rezhnikov and Chambers. And Rezhnikov also describe experiments in which they injected solutions of in these experiments too. The sodium and potassium ions caused a sinking and clumping of granules. Evidently, under these conditions also, the sodium and potassium ions produce coagulation. Chambers and Rezhnikov are of the opinion that the injection of calcium into the amoeba protoplasm causes a coagulation. This may very possibly be true. Ordinarily, when calcium enters a cell, it enters slowly and it is adsorbed as soon as it enters. The injection of calcium salt into a cell would expose the protoplasm to free calcium ions. In a later chapter, chapter 14, it will be shown that free calcium initiates a complicated series of reactions in protoplasm, the end result of which is a coagulation. The observations of colony 23 are similar to those of Chambers and Eilers-Neuer, and they show very clearly that further, this coagulative action is prevented by the presence of tensis, an aquatic plant. He placed pieces of roots in glass dishes and found that they could live for a long time, over 24 hours, in water doubly distilled from glass. They also live as long as this in solutions of calcium chloride or other calcium salts. And this is not due to a failure of the calcium to enter the cells, see below. But when the roots are placed in solutions of sodium or potassium salts, coagulative changes can be seen to occur in the protoplasm, and death follows. Colony's description of these changes is very clear, and it is here given in his own words. Wenn wir aber die Wurzelhaare in Entsalzung oder in die Losung eines anderen K-Salzes bringen, so kommen wir nach einer gewissen Zeit, die von der Salzkonzentration abhängt, aber stets schneller als in einer Stunde, folgende interessante Erscheinung beobachten. Die Plasmabewegung wird immer langsamer. Also, Pachimas 28 reports experiments on the effect of salts on amoeba. His results do not agree 154 fully with those of Oyambex and Reznikov. Brindley 28b has studied the effect of salts on the viscosity of amoeba protoplasm. He used the amplitude of Brownian movement as an index of viscosity, and observations were made almost immediately, within 2 minutes after entrance of the amoeba into the solutions, personal communication. In sodium chloride, the amoeba appeared to increase in volume, owing to a dissolving effect on the plasmalemma, personal communication. In calcium and magnesium chloride, there was probably a volume decrease, for the solutions used were apparently hypertonic. When the amoeba volume increases, there is more room for Brownian movement, and when it decreases, there is less. This is probably an explanation of Brindley's results. It would be hard to assume that the salts could diffuse far into an amoeba within 2 minutes. Note added to proof. Dies durch Viskositatia-Gierthermik verursacht wäre und zugleich ist an der Haarspitze selten an anderen Stellen eine Anhäufung des Protoplasmas zu beobachten, welches hier bald ein festes Gerinnsel bildet. Dieses Plasmagerinnsel hat gewöhnlich so grobe Dimensionen, da es sogar mit Hilfe einer Lupe zu sehen ist, wobei es als triebweibes Klubenchen, welches dem peripheren Teil der Zelle ansfut, erscheint. Diese Erscheinung ist als allmalige Plasmagerinnung zu betrachten. Bald darauf folgt der Tod. In N20 of CL Lasungen starben alle Haare, gewöhnlich nach 30 to 40 Minuten AB. No one reading Colloid's description could doubt that the potassium ions cause a coagulation of the protoplasm of triannucleates. If a little calcium, or if a root hair in which the potassium ion has already been made to produce its effect, is placed high a solution containing calcium, either in a pure solution of a calcium salt, or in a solution of a liquefaction of the coagulum, and a renewal of protoplasmic streaming. The coagulum does not always disappear, at times a small part of it remains. From Colloid's account, it is certain that the potassium ion produces a coagulation within the protoplasm of the root hairs of Trinat. Colloid states that sodium, lithium, and ammonium behave in similar fashion. Lation has been initiated by the potassium ion. This may be reversed by immersion of the roots in a solution containing calcium. Obviously, the calcium must enter the cell to produce this effect. At any rate, this is certainly highly probable. Another clear case in which the sodium ion causes coagulation and the calcium ion liquefaction is provided by the experiments of Weber 24D. Weber treated filaments of Spirogyra orthospira with a spira with solutions of calcium and with a solution of sodium viscosity, both by the centrifuge method and by the plasmolysis form method. Both methods gave consistent results, and it is evident that in this type of protoplasm, the sodium ion causes coagulation, the calcium ion liquefaction. Weber's experiments action of salts of calcium and sodium on the protoplasmic viscosity of Spirogyra Weber 24D readiness, with which chloropius move on centrifugal treatment. Chloropius move readily, chloropius move readily, chloropius move readily, chloroplasts move readily, ci chloroplasts move very, chlorinolis move with great difficulty, chloropius move with in the viscosity of the protoplasm of the treated cells as compared with that of the controls in tap water, and that a sodium chloride solution has the opposite effect. It should be noted that the plasmic viscosity higher, not lower, as was actually found to be the case for effective hypotonicity CP212. In his paper, Webb cites Hanstein-Kranner 10I as having shown that plasmolysis by a potassium chloride solution is uneven or angular, whereas when cells are plasmolyzed by a calcium, say the plasmolysis is convex. Hanstein. Kranner worked with the epidermis cells of the scales of the onion. His figures are very beautiful and show clearly two different types of plasmolysis. Colloid 24 also found in onion cells an irregular or echid plasmolysis following plasmolysis with sodium chloride solutions. But Weiss 25 and Pratt 26 working with the same material, but apparently unaware of the older results, both found that when the onion caesules were plasmolyzed in ammonium, potassium, or sodium chloride solutions, the surface of the cell was smoother, i.e., more convex than when plasmolysis occurred in calcium chloride solutions. This divergence of results is probably not hard to explain. Hanstein-Kranner compared normal solutions of potassium and calcium. In List's experiment, the potassium chloride solution was the stronger osmotically. Wise, on the other hand, used solutions of equal molarity, and in his experiments, the calcium solution was stronger than the solutions of sodium, potassium, and ammonium chloride. Pete gives the concentrations of his solutions in percents, but apparently he too used stronger solutions of calcium chloridine. As a matter of fact, all of these experiments in which the cells are placed directly in the plasmolyzing solutions are probably of little value as a source of information regarding the viscosity of the protoplasm in the interior of the cell. The cell plasmolyzes before the calcium and sodium ions have time to penetrate into the interior and produce a result. It is to be noted that in Webb's experiments, he treated the SG and Rogira cells a long time with the salt solutions before he subjected them to plasmolysis. The experiments of Chambers and Reznikov, those of Colloid, and those of Weber, all support Heilbrunn's claim that calcium ions cause decreased viscosity of protoplasm, and that sodium, potassium, and ammonium ions have the opposite effect. There is thus direct evidence, not only for Sorcine eggs and for stentoprotoplasm, but also for the protoplasm of amebon, for the root hairs of Triania bogotensis, and for the protoplasm of B. Phrygia, that this view is correct. There is also some indirect evidence. Source 24b describes some visible changes in Spirogyra cells following treatment with various bivalent and trivalent cations. These he regards as indicative of an increase in fluidity. Such an interpretation would accord very well with the point of view expressed in the preceding pages, but unfortunately Scarth's reasoning is not very convincing. In the first of his two Scarth mentions, the fact that Ba was found to produce rapid streaming of protoplasm in the case of the onion, and he also cites Searize 22, who found that strontium-stimulated protoplasmic streaming in Madia cells. The fact that these bivalent ions favor a rapid rate of protoplasmic streaming is consistent with the idea that they cause a decrease in protoplasmic viscosity, although, of course, the effect of the ions may be on the motive force of the streaming. Unfortunately, there have not been many experiments on the effects of various cations on protoplasmic streaming. The experiments of Seifriz PB are not entirely satisfactory, for he compares the action of 0.128 yum K-Colgea with that of 0.128 M-Nakel, and these solutions are not as osmotic as he believes them to be. Cajo 21b 24a has performed many interesting experiments on the effects of various salts on plant cells. In these experiments, the salts are used in relatively high concentration, and their toxic action is compared by noting how soon the cells are killed. In order to determine the death point, Cajo treats the cells with a F-plasmolytic agent. If they plasmolyze, they are considered to be still alive. If they fail to plasmolyze, they are dead. It seems probable that the failure of a cell to plasmolyze is the result of coagulation of its protoplasm. We can therefore interpret Caho's results in this fashion. Caho 21b found that when Tradescantia cells were exposed to 0.2 N solutions of potassium, ammonium, sodium, strontium, magnesium, barium, and calcium of salts with monovalent cations than in those with bivalent cations. Thus, in the potassium chloride solution, almost half of the cells were dead after 4 hours, whereas even after 20 hours, all of the cells exposed to the calcium chloride solution were still alive. It should be pointed out, however, that the solutions of salts with bivalent cations are weaker osmotically, and Tixat this may be an important factor. Caho believes his results are due to differences of permeability, and this may very well be true. An alternative interpretation is that the bivalent cations do not cause coagulation, whereas the salts of the monovalent cations do. In this connection, it might be worth mentioning that since 1804, it has been known that the roots of bean plants can live indefinitely high solutions of precalcium chloride. Although they are killed or injured in weaker solutions of various other salts, in more recent years, this knowledge has been extended for many other types of roots by Hanstein-Kranner, 1014-22. This author finds that calcium chloride is non-toxic even when it is present in what he considers a relatively strong concentration of N50. Obviously, in these experiments, the calciactuous of various salts, Cm enters the cells of the roots, for the plants live for indefinite periods in the solutions of calcium salt. In view of the work of Hackstein-Quanker and of the older work of Wall and others, it is rather surprising to find that Adams claims that the calcium ion has a toxic effect on the root hairs of the wheat plant after only 10 minutes exposure. Hanstein, C. Banner, Wohl 64, Bohm 75, all found that the roots of various plants could live for indefinite periods when exposed to calcium. Adams, after 10 minutes, finds a toxic effect, which is indicated by a coagulation of the protoplasm of the root hair. The explanation is not far to seek. Adams used N10 solution, and this WA is evidently too strong osmotically. This explains why the solutions exerted an action within 10 minutes. One could hardly expect calcium ions to diffuse deeply into the protoplasm within 10 minutes. Adams also finds coagulative changes following exposure to N10 solutions. Of all of these cases, the effect may be due to the osmotic action of the solution. However, in the case of the zinc and aluminum solutions, the high concentration of hydrogen ions probably played a part. The work that we have considered so far, with the exception of that of Adams, has favored the view that calcium ions tend to produce liquefaction or decreased viscosity of the protoplasm, and that sodium and potassium ions have the opposite effect. We shall now consider two sets of experiments which do not fit in with this conciliation. Edish and Josephus 27 studied the appearance of nerve fibers of the frog in the dark field using the cardioid condenser. The U of cows seems to have been very carefully done, and the photographs are excellent. Unfortunately, the nerve fiber is surrounded by two sheaths, and the outer of these sheaths gives a very bright image under Darl-Dield illumination. Under such conditions, observation of the interior of the fiber can scarcely be very accurate. One need only be reminded of the warnings of Deichkoff 10 and Peebice 14 against the use of such material for dark field study. CP 38. Ishiwago 26 found the nucleus of leukocytes optically empty in Darmild, whereas structures were visible in it in the bright field. This he rightly attributes to the brightness of the nuclear surface, which renders observation of the interior impossible. It is readily understood why Edish and Joachim's find the interior normal nerve fiber optically empty, in spite of the fact that there are doubtless visible colloidal macellai there, just as there are in all other types of protoplasm. The appearance of brightness within the fiber may be due either to the appearance of a new phase or to a change in the optical relations which permit more light to enter the fiber. Etish and Jokuvis report that in the presence of calcium salts, the interior of the fiber becomes brighter, and they regard this as evidence of coagulation. But they state, and their pictures show, that the calcium ion has a marked effect on at least one of clashes of the nerve fiber, and the increased brightness might be due to this factor alone. Finally, we shall consider the peculiar results of Timbal 27. This author studied rather a wide variety of plants, but in particular Galtha paestris and Monarda cladiuma. He studied stems and sections of stems and determined viscosity by the centrifuge method. When the stems are centrifuged, ordinarily there is no movement of chloroplasts, but when the culls are exposed for a short time, 5 minutes, to a half-molar solution of potassium plant tissues are centrifuged. Potassium nitrate seems to produce the strongest effect. Potassium chloride and sulfate have a less pronounced action. Ions of sodium, lithium, magnesium, barium, and calcium have no noticeable influence. It must be admitted that these results are very unexpected, and one is at a loss for an explanation. One would expect that if potassium had a liquefying action, sodium would likewise have such an action. Let us examine Timmel's experimental evidence a little more closely. In the first place, it is noteworthy that he used solutions which plasmolized the cell. He states, however, that solutions which did not cause plasmolysis, 0.4 N or weaker, also have the same effect. How much weaker the solutions could be and still prove effective, Timmel does not state. But he does state that 0.1 N potassium nitrate solutions are ineffective. If the action of the salt is due solely to an effect of the potassium ion, one is tempted to inquire why these weaker solutions do not alter the viscosity. After sufficiently long exposure, Timmel exposed the tissues to a 0.1 N concentration of the salt for 24 hours. After 24 hours, exposure to 0.1 N KCl, probably more potassium ion would penetrate the cells than after a minute's exposure to 0.5 N KL. Another interesting fact regarding Timmel's experiments is that in some months of the year, FJ potassium is without an effect unless it is combined with another plasmolytic agent, such as cane sugar. From these considerations, it seems probable that the peculiar results of TVDL are due as much to the abnormally high concentration of the salt as to the specific effect of the potassium ion. But it is nevertheless very remarkable that hypertonic solutions of potassium salts can cause a decrease in viscosity, for in protoplasm, generally, decrease in water content of the cell is accompanied by increase in viscosity. It seems certain that TGI Thell's results are peculiar to certain types of protoplasm and cannot hold generally. He found that relatively concentrated solutions of potassium salts, but not plant cells, to be more readily moved by centrifugal force when the experiments were performed at some seasons of the year rather than others. Perhaps the potassium salts react in some peculiar water with constituents of the plant protoplasm, but there is also another type of explanation which is possible. The ores of the cells he studied are close to the outer boundary of the cell. Perhaps in this position they may, under certain conditions, be anchored to this membranous boundary. If large granules or chromatophores are anchored to the outer membrane of a cell, one could hardly expect them to be moved by centrifugal force. Thus, Heilbrunn 20 have found that in the sea urchin egg, at times when the protoplasm in general was of low viscosity, the pigment granules might assume a position close to the outer membrane, and there resist movement following centrifugal treatment. In plant cells also, a slight movement of chromatophores away from the outer boundary of the cell might make them much more readily movable by centrifugal force. In view of the fact that the size, the shape, and the position of the chromatophores within plant cells are profoundly modified by all sorts of external conditions, it is plus unsafe to use the movement of chromatophores or their failure to move when cells are centrifuged as any index of the viscosity of the protoplasm, unless at least one can be certain that the conditions of the experiment have no effect on the position of the chromatophores within the cell. SEM 8 has gathered together a great mass of evidence concerning the movement of chromatophores under different conditions. It is interesting to note that Keister, in whose laboratory T-Bimmel's work was done, IMSEC reports movement of chromatophores away from the outer wall of plant cells under the influence of solutions more concentrated than the normal environment. but not concentrated enough to cause plasmolysis. See Custom 5. In the discussion up to this point, we have considered only those authors who have attempted a physical study of the action of salts on protoplasm. In the literature, and particularly in the older literature, many writers expressed opinions as to the effect one salt or another might have on the colloidal state of the protoplasm. Some of these opinions were based on guesses, some on analogy, and others were arrived at after the authors had looked at cells immersed in various salt solutions. Thus, Gibeli IV, from morphological appearances, attempted to distinguish coagulation and liquefaction in paramecium, after this Prof. Gazone had been exposed to different solutions of salts. The figures Greeley draws are not very convincing, nor do his conclusions appear very probable. Thus, for instance, he is of the opinion that M40Kl coagulates the protoplasm, whereas to M40Nacii he ascribes a liquefying effect. Speak 20 experimented on the action of various salts on paramecium. He found that Liber, Leckel, and Cassien caused the paramecia to increase in volume, and he attributed this effect to a direct swelling action on the colloids of the protoyondasm. By actual viscosity measurement, it can be shown that lithium salts cause a coagulation, that is to say, a great increase in viscosity in the protoplasm of the protozoan stentor, see above, and also in the piatoplasm of paramecium, unpublished experiment of Mr. L.G. Barth. Very frequently, the increase in volume of cells is accompanied by coagulation. In 1921, Speck studied the effect of various salts on the protoplasm of Actinospherium, and he described certain types of morphological change, which he regards as significant of colloidal change. In this, as in all of his papers, Speck makes the error of assuming that equimolar solutions of sodium and calcium chloride are isosmotic. In observations on the protozoan parasite Opalina, Speakpa finds that the addition of rather large amounts of 0.3 m calcium chloride causes shrinkage of the Opalina cells, whereas the addition of 0.3 m sodium or potassium chloride apparently has no such effect. Quite apart from any specific action of ions, it should be clearly understood that a 0.3 m calcium, particularly as a sodium chloride solution of the same molar concentration. If one were to assume a relative impermeability of the B even stronger osmotically as compared with a sodium chloride solution, it is not probable that all of the effects described by Speake are due to the osmotic differences of the solutions he used, but it hardly seems worthwhile to attempt to interpret his morphological data, complicated as they are by his misunderstanding of osmotic relationships threes. Giesbig's studies of the action of salts on the protoplasm of amoeba are in the same category as those of Speake. He too compares solutions of equimolar strength. In one of the two tables in which he compares the action of sodium and calcium chlorides, he finds that M20 calcium chloride leaves the protoplasm perfectly normal for 12 days, whereas M20 sodium chloride causes the production of granulations and various other pathological changes in the protoplasm. In the other of the two tables, M20 and death within 2 days. Gibbsby's comment is that one could hardly expect to get the same result each time. According to Hesnikope and Shambeb's 25, amoebae can live for at least 3 days in M26 calcium chloride. In resume, it may he stated that the work of Heilbrunn, Shambeb's and Hesnikope, Kolodny, and Webb all shows clearly that for various types of protoplasm, sodium, potassium, and ammonium ions cause coagulative changes, and that these changes are prevented by the ions of magnesium and calcium, which tend to have the opposite effect. The work of these authors rests on a safe, sure basis and can hardly be challenged. In addition, there is some indirect evidence that is in harmony with the above thesis. On the other hand, there is apparently no reliable experimental evidence that is in conflict with it. In the future, it may perhaps be shown that some tyides of protoplasm behave very differently from the protoplasm of sea urchin eggs, STC Trionea, amoeba, and Spirogyra. This is quite possible, For it may be true that some types of living material have colloidal particles charged differently from those of the cells we have considered. But at the present time, there is no direct evidenza that this is so. It should be remembered that our statement regarding the effect of sodium, calcium, and other similar ions on the protoplasm is only true for the protoplasm of the interior of the CC cell. Moreover, it should also be noted that the primary effect of feces ions may be complicated by secondary effects, such as what we have described for calcium in the case of the sea urchin EVP. There has been very little study of the action of anions on the colloidal properties of living protoplasm. In tin sea urchin egg, such study would not be easy. Varphorous anions, iodides for example, cause changes in the osmotic inine brain of tli, and these changes result in a large increase in teleo volume of the cell. When sea urchin eggs are placed in solutions of sodium iodide, which at first appear to be osmotic, after a time the cell toxicates up a considerable quantity of water, and the colloidal clei of the protoplasm are due as much to this water intake as lie any specific effect of the anion. EO 21b exposed sections of Tratus cameolose and TF red cabbage to various salts of sodium, potassium, and ammonium. Not only the cations, but the anions as well deforcanal interwire effect on the cells. Cajo studied the time required for 2,2 cuttles to be queued in the different solutions, using as an index of ELITH the film E of the cells to plasmolyze when treated with hypertone solutions. Such a failure to plasmolyze is probably, as 11-0 supposes, evidence of coagulation. Cajo found that. The order in which the various anions favored coagulation was TLN CNSFI 30H. In his experiments, Cajo compared equinormal solutions. So teleate the solutions of tartrate, citrate, and sulfate were much weaker osmotically than the other solutions. This may be a factor to be considered, for apparently Cajo generally used hypertonic solutions. It is understandable, therefore, why tartrate, citrate, and sulfate are less toxic than the other salts. The salts of monovalent anions were used in approximately osmotic concentrations, so that the results with these salts are more interesting. From Cajo's results, it seems possible to conclude that the sulfocyanide ion and the iodide ion have a much stronger coagulative action than the chloride ion. This is readily understandable in the theory that we have produced. The sulfocyanide ion and the iodide ion are known to be more readily absorbed than the chloride ion, and hence they would have a greater effect in neutralizing the positive charge of the protoplasmic suspension. Cajo himself believes his results are explainable on the basis of differential permeability, the ions which penetrate most rapidly causing death or coagulation soonest. This seems a perfectly possible alternative interpretation. Chapter 11. The electric charges of protoplasm. In any colloid, the electric charge on the colloid particles or micellae is of great importance. More than anything else, it is this charge that keeps the particles from coalescing. When the charge is neutralized, the particles form larger aggregates, and these settle out of solution. Of the two main types of colloidal solutions, the lithocolloids are even more dependent for their stability on the electric charge of the dispersed particles than are the thiophile colloids. On the basis of its colloidal behavior, protoplasm really has more in common with lithocolloids than with colloids. This is readily understandable when we remember that protoplasm is actually a suspension. Obviously, one of the most important problems in the study of the colloid chemistry of Jane protoplasm is the determination of the sign of the electric charge on the colloidal particles of the protoplasm. A colloidal solution may have particles with a positive charge or with a negative charge. For the colloid chemist who deals with inanimate colloids, it is usually a simple matter to decide whether the colloid he is studying is of the one type or the other. The colloidal solution is placed between suitable electrodes, and from the migration of the particles either to the anode or to the cathode, the sign of the charge can be determined. For the biologist, the problem is not so simple. If he attempts to study the cataphoresis of living protoplasm, he must follow the movements of the colloidal particles not in any container of his choice, but within the cell. Every student of cataphoresis knows of the complicating effect of the wall of the cataphoresis chamber. If colloidal particles travel in one direction through the center of a cataphoresis chamber, they may travel in the opposite direction along the walls, for there they are carried by the dispersion medium, which can move in a direction opposite to that of the colloidal particles. If the wall of a living cell is charged electrically, as it probably is, there would be a tendency for dispersion medium to flow along it. But this is not the only difficulty, or even the chief difficulty. When one passes an electric current through an inanimate colloid, there is no coagulation or other disturbance except in the immediate vicinity of the electrode. But when even relatively weak currents are sent through living cells, there is a practically instantaneous coagulation of the entire protoplasm. Such a coagulation would, of course, prevent any cataphoretic movement of the colloidal particles of the protoplasm, or it might very well bring about a reversal of the normal charge. From what has been said, it is obvious that any evidence regarding the cataphoretic migration of protoplasmic granules or colloidal particles must be viewed critically and carefully. Fortunately, the colloid chemist also has a second method which makes it possible for him to determine the electric charge of a colloid without the use of an electric current. This second method is particularly applicable to suspensoids and suspensions. It can therefore be used for the protoplasmic suspension. The principle of the method is simple enough. In recent colloid chemical theory, it is generally assumed that the charge on the colloidal exoorticals is primarily due to adsorbed ions. It is well known that bivalent ions are adsorbed more readily than monovalent ions, and that trivalent ions are adsorbed far more readily than bivalent ions. If then bivalent or trivalent cations are added to a colloid, they will tend to neutralize the charge on the colloidal particles and produce precipitation if this charge was originally negative, whereas if the charge was originally positive, they will have no such effect. On the other hand, bivalent and trivalent anions tend to exprecipitate positively charged colloids. For details concerning the use of this method, various books on colloid chemistry may be consulted. Analyses of living cells typically show an excess of bivalent cations over bivalent anions, CP25. Thus, protoplasm usually contains more calcium and magnesium ion than it does sulfate ion. This is in itself an indication that the arthroplasmic succession bears a positive charge. Much stronger evidence, however, is obtained when the protoplasm is exposed to the action of various ions. Normally in the cell, there is a mixture of monovalent and high-valent cations. If we expose cells to pure solutions of salts with monovalent cations, i.e., sodium or potassium or ammonium, the amount of bivalent cation adsorbed on the surfaces of the colloidal particles or granules of the protoplasm is decreased. If the charge on the protoplasm were negative, this would have no effect or would favor liquefaction. As a matter of fact, the sodium, potassium, and ammonium ions produce a coagulation of the protoplasmic suspension in all cases in which accurate data have been obtained. See previous chapter. Hence, from this fact, and from the fact that the addition of calcium or magnesium ions produces liquefaction as a primary effect, we may conclude that the charge on the granules of the protoplasmic suspension is positive. Just as in the case of inanimate colloids, the action of trivalent ions is far more potent than that of bivalent ions. So too in protoplasm, the trivalent ions aluminum and cerium are extremely effective in producing a liquefaction of the protoplasm, CP 1-51. We are thus led to the conclusion that the colloidal particles of the protoplasmic suspension, or the granules, bear a positive charge. This is an extremely important conclusion, and we can hardly be content with one line of evidence. The protoplasmic colloid is far more complicated than any inanimate colloid, and the chances of errors of interpretation are proportionately so much greater. In the pages that follow, therefore, we shall attempt to see whether or not the assumption of a positive electric charge on the colloidal elements of the protoplasm is in harmony with various known facts of cell physiology. In the first place, we shall try to assemble all the known experiments on the cataphoresis of the protoplasm in the interior of living cells. The literature on this subject is more extensive than is generally thought. At first sight, it appears inconsistent, for whereas some authors have described a migration of protoplasmic particles toward the cathode, others have described a migration toward the anode. Fortunately, a critical survey of the literature seems to indicate that there is no contradiction between the results of the different investigators. The study of the cataphoresis of protoplasm is as old as the study of cataphoresis itself. In what appears to have been the first clear-cut description of cataphoresis, Jugensen 60 studied the migration of carmine particles and of lycopodium powder in an electric field. In the same paper, Jugensen also reported some experiments on the effect of an electric current on the protoplasm of the leaves of Vallisneria dot. In his description of his results, J.T. Juergensek states that the current first killed the cells, and that then the protoplasm migrated to the anode. This sort of experiment has been frequently performed by subsequent investigators, and it seems certain that after death, the protoplasmic colloids have a negative charge. But of course, we are more interested in the charge of the protoplasmic colloid during life. The first description of cataphoresis of living protoplasm is that of Kuhne 64. On page 79 of his classic monograph, Kuhne describes an experiment in which a constant electric current from 6 small growth cells was passed through some myxomycete protoplasm. The electrodes were broad platinum plates, and they were 4 min apart. As soon as the current entered the protoplasm, it caused the movement of granules toward the cathode. Moment, wo ich die Kette schlopp, fand eine ruckweise eintretende Beschleunigung der Kornchenstromung statt, welche vom positiven zum negativen Pol gerichtet war, während die entgegengesetzt Fliehende für einen Augenblick stillstand oder auch etwas zinieckig. Umkehrungen der Ersternung von irgendwelcher langeren Dauer konnte ich von dem konstanten Strom nicht erreichen. Ich überzeugte mich aber durch rasches Umwerfen einer in den Kreis geschalteten polschen Wippe davon, gab die Erscheinung auf das Chittier-Bensensche Phänomen zurückzuführen sei. Jede Bewegung der Kornchen, die sich überhaupt abhängig zeigte von der Pichtung des elektrischen Stromes, verlief vom negativen zum positiven Pol. Ohne Zweifel haben wir es hier wirklich mit dem Jugensenschen Phänomen zu tun, denn eine entsprechende Kontraktion auf dem einen, bald auf dem anderen Ende der Myxomyserte in Abhängigkeit von den Ein- und Austrittstabellen des E-Stroms konnte nicht beobachtet werden. In the above quotation, the reference is doubtless to Juergensen 60. In spite of the difference in spelling, so that Juergensen phenomenon is equivalent to cataphoresis, in one respect the quotation from Kuhne is ambiguous. After stating clearly enough that the electric current caused an acceleration in the movement of granules toward the cathode and a retardation of the granules moving naturally in the opposite direction. Kuhne states that in order to make sure that the effect was due to the electric current, he reversed the current and then found a movement of granules toward the positive pole. There is only one way to interpret this so as to make sense. It seems certain that after Kieser reversed the current, the movement of granules was toward the pole which was originally positive. In the further discussion of his experiment, Kuhne is careful to note that there was no contraction of the protoplasm at the points where the current passed in and out. There was, it is true, a slight contraction at the make and break of the current, but this was equally evident at both positive and negative poles, and was not confined to the parts immediately in contact with the electrodes. Kuhne notes that the effect of the constant electric current on the movement of granules does not last. His electrodes are polarizable, and gradually the diffusion of substances formed at the electrodes interferes with the phenomenon. Kuhne seems to have had a clear understanding of the conditions of his experiment, and it is probable that he actually did see a cataphoretic movement of the protoplasmic granules toward the negative electrode, that is to say, the cathode. In 1876, Velton 76C wrote a paper which is primarily concerned with the action of electric currents on protoplasmic streaming in various tiges of plant cells. In Tlia, a summary of this paper, appears the following statement. Bei starken elektrischen Stromen sehnt sich das Protoplasma vorzugsweise gern an der positiven und negativen Polen zugekehrten Zellwand in Form von flachen oder ellipsoidischen Körpern an, die stromstarken an welche eine Veränderung des Zellinhaltes nach dem positiven Pol hervorrufen, übersteigen die Strome, die hier als sehr stark bezeichnet werden. Welten then goes on to say that the terms strong, weak, etc. are only in relation to the effects of the electric current on the protoplasm. Welten found that the protoplasm might wander either to the anode or the cathode, but that it was the very strong currents that caused the movement to the anode. From this one can infer that the weaker currents caused the migration toward the cathode. The observations of Welten are very interesting, for if the interpretation we have taken is the correct one, the results of Welten are exactly like those reported many years later by Hardy. In a short paper of only 4 pages, Hardy 13 brings forward what is apparently the most clear-cut evidence regarding the cataphoretic migration and the electric charge of the internal protoplasm of living cells. Hardy placed onion root tips horizontally between non-polarizable electrodes, the final lead to the tissue being through some of the fluid in which the root had been growing. A field having a mean value of 5 to 20 volts per centimeter was established for from 2 to 10 minutes, after which time the root was instantly fixed in a solution of acetic acid and absolute alcohol. Hardy did not attempt to determine the strength of the field to which the living tissue was really exposed, for there is EG6, cataphoresis of protoplasm in cells of the onion root tip after Hardy 13. No way of knowing how much of the current actually passed through the cells themselves. Hardy states that the results obtained were decisive and consistent. The protoplasm collected typically at the negative end of the cell, although sometimes it was condensed into an equatorial plate. This is shown clearly in A and B of Figure 6, which is copied from Hardy's paper. However, after prolonged exposure to a strong field, the protoplasm was condensed at the Hardy further. The appearances in the cell substances are such as would be due to the simple migration of colloid particles in a salt, that is to say, of electrically charged particles distributed through a fluid. The sign of the particles being at first positive, they move towards the negative end of the cell. Here reversal of the charge occurs, and the particles move in the opposite direction. Considering Hattie's observations on the migration of particles within the nucleus, we shall speak later. Hardy's description indicates that the colloidal particles of protoplasm may reverse their charge after the passage of an electric current. It is not necessary to suppose, as he does, that the particles first move through the cell before the charge is reversed. In an earlier chapter, CP 130, it was shown that in many types of protoplasm, electric currents cause coagulative changes. These coagulative changes may very well be associated with a loss of charge. If the original charge of the protoplasmic granules were positive, and this charge were lost following the passage of an electric current, it would seem likely that a negative charge would be assumed. In solutions near neutrality, most inanimate suspensions are charged negatively. The observations of Cooney, VBLTPN, and Hardy all indicate that in weak electric fields, there is a cataphoretic migration of protoplasmic particles toward the cathode, and their results speak for the positive charge of the colloidal elements of the protoplasm. Similar results were apparently also obtained by Zeidler 25 Thiels. Hardy's results are especially instructive in that they serve as a guide to the understanding of what might otherwise seem conflicting results. With two strong currents that produce coagulation of the protoplasm, it is easy to understand how it is possible for migration to occur in the opposite direction. Consider, for example, the results of Meyer 21. This worker sent electric currents through the roots of onion, lupine, T3A, and bean plants. The peat roots were studied most. Instead of placing the root in a watery solution, as Hardy did, Meyer attached two ends of the root to non-polarizable electrodes. The seedlings were held in position by glass forks in a moist chamber, so that the current passed through the length of the root. An electric light current was used, the current actually passing through the root being measured by a milliammeter. In Hatch-LRD's experiments, only a small fraction of the current G passed through the root tissue. In E, it might be argued that the movement of the granules is due to the fact that they are carried by a current of water produced by electroendosmosis. But this can scarcely be true, for according to Hay, the nucleus does not shift its position at A. Meyer's experienced all the current went through the root. It is obvious that Meyer used very much more powerful currents than those used by Hart. He found indeed that after about 30 seconds exposure to a current of 0.3 milliamperes, the root began to lose its normal color and became watery in appearance. After about 2 minutes, numerous fine droplets appeared over the surface about 3 to 6 millimeters from the tip of the root. The root became very appreciably shorter. Meyer himself states that the effects he observed were produced by currents just strong enough to cause death. In Meyer's experiments, the protoplasm was certainly coagulated. His results are therefore comparable to those obtained by Hardy with strong currents for relatively long exposure. For currents which produce death, Meyer found a movement of protoplasmic particles toward the anode. There can be no question but that the currents used by Meyer were too strong. Various other observers from Huygens BN in 1860 until the present have also used too strong currents, so that their results give no information regarding the normal electric charge of the colloidal particles of living protoplasm. Verwer 90, in studying the effect of electric currents on the plasmodia of the Myxomyceae lalium septicum, found that the particles of the protoplasm might migrate either to the anode or the cathode if they migrated at all. He states that he could find no regularity in their behavior. From his description of his experiments, he seemed to have used stronger currents than quinine, for the electric current always caused a cessation of movement when it was first passed through the protoplasm. Taylor 25, in what is apparently a preliminary note, also reports observations on the cataphoresis of myxomycete protoplasm. He used microelectrodes in a small hanging drop. Taylor states that the visible granules moved toward the anode, but it is possible that he did not make a clear distinction between protoplasmic granules and ingested material, for he says that following Lister and Boldetupi, 1. The granules may be filtered out with cotton. Lister himself found that only some ingested spores were so filtered, but that the true granules passed through the cotton. Taylor also observed ultramicroscopic particles and noted that they moved either toward anode or cathode. Unfortunately, the conditions of the experiment were not very satisfactory. Microelectrodes have a great resistance, and the heat produced might injure the protoplasm. Moreover, in tiny droplets of fluid, electroosmotic currents might be important. Finally, Taylor does not state how the passage of the electric current affected the normal streaming of the plasmodium, so that it is not possible to compare his observations with the previous observations of Kuhne and Viwoid. Mo Clendon 10 figures a section of a hyacinth root through which a current of 0.5 milliamperes had been sent for 30 minutes. The protoplasm has moved toward the anode. McKee-Plegoide 14 states that both in animal and in plant cells, there is a migration of too many substances of or in the protoplasm toward the anode. But he does not state which cells he used in his experiments or what strength currents. Kokitz J23 seems to have used too strong currents. He sent 50 volts through the hair cells of Tritus cantia in air and found a migration of cell contents toward the anode. In Koketsu's experiments, the current caused an immediate cessation of streaming and evidently produced coagulation. So far, we have discussed only the cataphoresis of protoplasmic granules of plant cells. At the very end of the 19th century and at the beginning of the 20th, many observers studied the effects of electric symptoms on protozoa. The physiological journals from 1896 to 1906 are full of papers which deal with this subject. Most of these papers are concerned primarily with the reactions of the animals and the behavior of the cilia, and there are unfortunately very few observations on the interior protoplasm. What there are all favor the view that the colloidal particles of the protoplasm in the interior of protozoan cells are charged positively. Heilbrunn 89, 90, 96a describes the effects of electric currents on amoprotoplasm. During the passage of an electric current, the granules in the interior of the amoeba all migrate toward the cathode. The figures in Heilbrunn's 96 paper are especially clear. As a matter of fact, the observation has been confirmed by various other workers. Whereas it is certain that the movement of the granules of the amoeba protoplasm toward the cathode is consistent with the idea that they bear a positive charge, it must not be overlooked that this migration may very well depend on other factors. According to the leading theory of amoeboid movement, the advancing GIS pseudopodium of amoeba has at its surface a region of lowered surface tension. It might be thought that the electric current would cause a drop of surface tension at the cathodic side, and that the movement of the amoeba would then correspond on to the movement of a drop of mercury in an electric field, as studied by Christian Secking. However, in order to assume such an effect of the electric current, we should have to suppose that the surface charge of the amoeba cell was positive. And this is contrary to the evidence, which we shall cite later, which indicates that the surface of living cells bears a negative. The studies of paramecium seem to give clearer information. In the amoeba, the movement of the granules under the influence of the electric current was not distinguishable from the movement of the granules as it occurs during the normal movement of the animal. One cannot be certain, therefore, whether the migration of the granules toward the cathode depends primarily on electric forces or whether it is not an end result due to the movement of the entire animal. In the case of paramecium, there is no such difficulty. Before we examine the experimental evidence, let us inquire into the conditions of cataphoresis in the paramecium cell. Apparently, this cell is full of granules. After long centrifugal treatment, there is no observable movement of granules, see P81. This is probably an indication that there is no room for the granules to move, that they are too closely packed. What then can happen when we subject such a cell almost solidly full of granules to an electric field? The granules are doubtless charged, and they would tend to move in one direction or the other. They would press against the wall on one side the cell, and if there were enough space between the granules, they would leave a small region at the opposite end of the cell totally free from granules. This is exactly what does happen in paramecium. LXAJ Loft 95 sent electric currents through paramecia suspended in a gelatin gel to keep them motionless. When currents of about 1 milliampere were used, the granular endoplasm of the cell pushed toward the cathode. On the passage of an electric current, the cell bulges toward the cathode, and when the protoplasmic granules have left the side of the cell toward the anode, the wall in this region tends to collapse, thus resulting in the formation of a sharp point at the anode end of the cell. This is clearly shown in a series of diagrams taken from for Warn 96A. These are reproduced in Figure 7. One of Lenlo's figures is also shown in Figure 8. If the paramecium cell is full of granules, and if these are positively charged, there is hardly any other way in which they could respond to the passage of an electric current than that shown in the figure. The evidence, therefore, is good that the granules of the paramecium protoplasm are actually charged positively from our survey of the literature on cataphoresis in living cells. It seems not unfair to conclude that the weight of the evidence and electric current on paramecium protielectric charges of protoplasm favors the assumption of a positive charge on the colloidal particles of the protoplasm. The observations of Kitijajeste, Veltest, Hedy on various types of plant tissue, together with the observations of Lubloff, Warn, and others on paramecium, all indicate a movement of granules toward the cathode. Probably cataphoretic studies are not possible for all types of protoplasm. It is likely that in many cases, weak currents cause coagulation, and once this has occurred, the normal charge of the protoplasmic particles is no longer in evidence. As we shall see later, P182, the charge on the exterior of a cell is normally negative. When cells are suspended in a solution of good conductance, normally most of the current does not enter the cells at all, and the migration of the cells in the electric field is determined entirely by the charge at the surface. But there is always some fraction of the current that passes through the cells, and if this is the case, the charges of the colloidal idarticles in the cell interior would be a factor in determining the speed and possibly also the direction of migration of the cells as a whole. We will assume for the present that cells generally bear a negative charge at the surface, at least in ordinary media. If this is true, and if it is likewise true that the colloidal particles in the interior of the protoplasm are positively charged, then as the size of the cell increases, the charges in the cell interior should become increasingly important. Or, in the case of a sphere at least, the volume increases as the cube of the radius, the surface only as the square of the radius. This line of argument was used by Heilbrunn 23, and he cites an experiment of PVS lilies to show that the size of a cell may have an influence on its cataphoretic migration. Lily Zaru found that when frog blood cells were immersed in a sugar solution, the red blood cells and the smaller leukocytes, i.e. those about 10 in diameter, almost always move toward the anode. Leukocytes of medium size, 15 to 20, are usually slightly negative, although acapas if the protoplasmic granules are positively charged and migrate toward the cathode. The suspension medium in the cell should migrate toward the anode. There would thus be an equal migration in both directions, and the resultant force on the cell might be zero, if it were not for the fact that the cell membrane is permeable to water and not to granules. Protoplasma monographleinophorine. Heilbrunn and essentially indifferent or slightly positive. On the other hand, the more voluminous leukocytes, 25 to 30, were almost always decidedly positive, moving definitely toward the cathode. Lilly describes the appearance of cells in a given field moving in opposite directions. It is thus not probable that the movement of the large cells toward the cathode was due to their being carried in the stream of liquid which travels along the wall of the cataphoresis chamber. In commenting on Lilly's experiment, it is to be noted first that the cells which migrated in opposite directions were of the same type and from the same animal. It would hardly be profitable to compare the speed of migration of cells of different animals or of totally different types of cells from the same animal. Secondly, in Lilly's experiments, the cells were immersed in a sugar solution. This would provide an outer medium of low conductivity, and as a result, a relatively greater fraction of the current would jias through the interior of the cells. This is the very condition which would favor an influence of the interior charges of the protoplasmi on the direction and speed of migration of the cells as a whole. Bernadette 26 diluted the blood of the mollusk Phalhesia with sugar solution, 1-2 blood to 5-part sugar solution. Under these conditions, he found that the smaller cells migrated to the anode, whereas the larger yellow cells migrated to the cathode. In his description of his methods, Bernard states that he was careful to avoid deception from the movement of the film of fluid next to the glass. When the cataphoretic migration of cells is studied in blood serum or in salt solutions, one would not expect to find very great differences between the rate of migration of the small and the large cells. That such differences in rate of migration actually do appear is at least indicated by the exact quantitative determinations of Freundlich and Abramson 27 on the cataphoretic rate of blood cells of the horse suspended in serum. From the data of these authors, the average speed of migration of various types of white blood cells has been calculated, and a comparison may be made between the speed of migration of small lymphocytes, large lymphocytes, and the presumably larger leukocytes of the cells in microns per second for an electric field of 1 volt per centimeter. If Heilbrunn and Abramson separated their observations into two groups, those made 1 to 6 hours after the blood was obtained, and those made after 30 hours. In the cataphoretic migration rate of white blood cells of the horse in microns per second for 1 volt per centimeter, small lymphocytes, large lymphocytes. In view of the small number of observations reported by Heilbrunn and Abramson, these figures may not be statistically significant. It is interesting to note that the red blood cells, which have a volume still less than that of the small lymphocytes, were found by F.B. Echinaleo and Abramson to have a greater speed, 1.01 microns per centimeter. For a field of 1 volt per centimeter, but the comparison between different types of cells is probably not justifiable. It might be thought that the slower rate of migration of larger cells in an electric field was due to the greater resistance offered by the medium to the passage of the cell. As a matter of fact, both from the standpoint of theory and from the results of actual experiment, compare Freundlich and Abramson, the speed of migration of a particle in an electric field is independent of its size. The experiments of Elinese 3, Bernardi 26, and Freundlich and Abramson 27 all indicate that with increasing size of a cell, it tends to move less rapidly toward the anode and may even move in the opposite direction. So far, the evidence on this joint is rather meager, but what there is favors a view that the interior of the cell contains particles charged positively. Ideally, there is a hit of indirect evidence, which, although it is not as important as the evidence we have already considered, may be worth mentioning. Kolpe 97 advanced the view that the red blood cells were permeable to anions, but not to cations. Following Kolpe's paper, there has been an abundance of 1 degrees TP-pi to cations UAC that retard the passage of cations as SL to CX and phions, indicates that the all-new NEN of these cells is composed of particles charged tint inside of the negatively charged. Surface ap or ifix composed of positively charged particles does not prove that the colloids in the interior of Talu. Summing up, TV find the following evidence in Favoichi, positive charge on the colloids in the interior of living pro-IP enzyme 1, the excess of bivalent cations in living cells. 2. The fact that bivalent and trivalent cations ledive a lipefaction of the chirotoplasmio suspension hun us aa. DCUEs in the proportion of bivalent cations normally PNS myrazal as in 3. The cataliresis experimerity of Javiandino Violkian, UISPAI those of Hardy. All of those hyperenians to ELK administration of the protoplasmic granules of plant eels toivard ihecat. 4. The movement of the granules to third, ply in secretor ol inner paramecium cell toward the cathode. The fact that when the catioresis of diulsninials of the same TAE is compared, the larger coles slilo a grueller tendency to move toward the cathode. It is, of course, quite possible that different TIFSS of protoplasm differ in the sign of the charge on there. Colloidal parallel. So far, there is no evidence to indicate that this is the EST. All the TQQIs of protoplasm that have biosynodal C2TF behave in the same manner. In our discussion up to this point, would you have it? UNC. This does not agree with the opinion of Harrietian 2511 regarding the charge on the colloidal particles of the Aultane Tutnurain of the CN Rhine egg. This opinion was not based on Viney CTNUUT. There is good evidence that the colloidal particles of the nucleus are charged with an opposite sign from those of the cytoplasm. That is to say, that they are charged negatively. The experiments of Hadi H3 previously referred to very clearly demonstrate a migration of the materials within the nucleus toward the anode following the passage of an electric current. This is shown in all 3 of the sketches in Figure 6. Apparently, the colloids of the nucleus do not reverse their charge as do those of the cytoplasm, but remain negatively charged at all times. Haxity reached his conclusions regarding the negative charge of the colloidal particles of the nucleus acts apparently quite independently of the earlier work of PBN Amali and Penamali Tainin past currents of from 0.0186 milliamperes to 0.049 2 milliamperes through hyacinth root tips for 20 to 45 minutes. He used non-polarizable electrodes, and in different series of experiments, the current was sent in various directions through the root. Penamali was able to demonstrate a movement of the chromosomes of dividing cells toward the anode. However, the chromatin of resting nuclei showed little or no movement. Mo Clendon 10 repeated Pentamali's experiments using approximately the same strength of current for 30 milliamutes, both in the onion root and in the root of the hyacinth. Miki Manan was able to demonstrate a movement of chromatin toward the anode, but Mo Klekbon was able to show not only a movement of the chromatin in dividing cells, but also a movement of the chromatin in resting nuclei. His figures leave no suggestion of doubt. Mo Olaizer states also that the chromatin of the cells of the newt demicti moves toward the anode, and that nucleoli of frogs' eggs travel in the same direction. He gives no details of these experiments with animal cells. In a second paper, Peta Timoli 12 repeated his earlier experiments on the hyacinth root using stronger currents. In this paper, his figures show very clearly a migration toward the anode of chromatin, both in resting and in dividing cells. Heidelberg 25 has also described a migration of chromatin toward the anode. Pentemolex, Meekland, Habby, and Zeibel agree in ascribing a negative charge to the colloidal particles of the nucleus, and it seems certain that in the cells studied by them, such a charge is actually present. The experimental procedure used by the above-mentioned authors is simple, and it is rather surprising that more experiments of the same type have not been performed. Surely the electric charge on the chromatin is an important subject for investigation. By sending an electric current through cells for varying lengths of time, some idea of the magnitude of the charge might be obtained. Although in order to determine the charge exactly, a knowledge of the relations of the current within the nucleus would be necessary. Other types of cells could also be studied, or the cells could be treated in one way or another to see what effect such treatment might have on the electric charge. As far as the evidence goes, the colloidal particles of the interior cytoplasm bear a positive charge. Those of the nucleus, a negative charge. We shall now consider the charge on the surface of the cell. The charge on the colloidal particles of the cell interior is determined largely by the ions present inside the cell. Concerning these ions, we have little definite information, except that apparently there is an excess of inorganic cations. The charge on the outer surface of the cell would be determined largely by the ions present in the outer medium. Concerning these ions, we have complete information, or at any rate we may have. The outside membrane of the cell is probably protein. Most of cations of high valence are charged negatively. It is not surprising, therefore, to find that in cataphoresis experiments, generally living cells have been observed to migrate toward the anode. This is particularly true for small cells, such as red blood cells and bacteria, for in such cells the charges of the interior play less of a role. The first description of cataphoresis of living cells is that of Chitubjiksen 60. He found that blood cells migrated toward the anode. Since Huygens Bien, there have been very raw papers published on the cataphoresis of living cells, and it would scarcely be possible to refer to all of them. The more recent workers, especially following the publication of Convenient Directions by Michaelis 21st, have usually been careful to guard against the error due to the movement of the fluid in the cataphoresis chamber. The following papers may be consulted. Some of them contain references to older literature: V. St. Georgi, 20, Putty, 21, Nedab 25, Tamita 26, Binabdi 26, Winslow and Upton 26, Soho Deb 26. Just as the charge of an inanimate particle is influenced by the ions of the surrounding medium, so too the charge on the surface of living cells is altered when various ions are added to the fluid surrounding the cells. In the presence of a sufficient amount of bivalent cations, the charge on living cells may be changed from negative to positive. See, for example, Bieber and Abdi 26. Trivalent ions are especially effective in producing a change in direction of electric migration of living cells. See, for example, Kazawa 14. Our discussion has shown that the granules in the cytoplasm of hiving cells bear a positive charge, that the materials of the nucleus have a negative charge, and that the surface of the cell is usually charged negatively. There is also some indirect evidence that the colloidal particles of the osmotic membranes of cells, blood cells at least, have a positive charge. It should be clearly understood that the charges on colloidal particles, living or non-living, are not free charges. They cannot be made to travel along a wire. For each positive charge on a colloidal G particle or macella, there is a negative charge in the dispersion medium immediately adjacent. The potentials that we have been considering are moreover all electrokinetic potentials or C potentials. It is this type of potential that is measured in cataphoresis experiments. In recent years, various authors have attempted to determine the difference in thermodynamic potential or E potential between the inside of hiving cells and the outside. C. Edishampati 25AB, Peter Fee, C. Keller 25, Taylor and Whitaker 26, Ostabout, Dariax, and Jacques 27, Jickort and Umrath 28. Differences in the thermodynamic electric potential between the inside of a cell and the outside may be due to diffusion potentials, to potentials resultant G from the differential permeability of the cell membrane to anions and cations, and possibly to other factors. In some of the experiments cited above, there is also the possibility that an injury potential may have played a role. The results that have been obtained, although very interesting, have no very direct bearing on the colloid chemistry of protoplasm. For a discussion of the difference between electrokinetic potential and thermodynamic potential, see recent books on colloid chemistry, especially Edisch 1927, Die physikalische Chemie der colloiden Systeme in Bethy's Handbuch der Normalen und Pathologischen Physiologie Vol. 1. Chapter 12, The Action of Acids and Alkalis. The study of the action of acid on protoplasm is beset with unusual difficulties. Indeed, it must be confessed at the outset that for the most part, these difficulties have not been overcome, and that our present knowledge regarding the action of acid on protoplasm is rather unsatisfactory. The older physiologists who experimented with acids compared solutions of similar normalities. In recent years, it has been clearly recognized that two solutions of the same normal concentration may differ widely in acidity. That is to say, in hydrogen ion concentration. One may be thousands of times as acid as the other. With the development of simple and easy methods of measuring hydrogen ion concentration, and with the recognition of the importance of slight differences in such concentration on the life of plant and animal cells, biologists have now generally learned to express acidity in terms of hydrogen ion concentration. Every beginner in physiology is now taught to measure the pH of pH solutions he uses in his experiments. Unfortunately, the assumption is often made that two solutions of the same hydrogen ion concentration are strictly comparable insofar as the effect of the acid ion on protoetherealism is concerned. As a matter of fact, in two solutions of the same pH, the amount of acid which enters the cell in a given time may be vastly different. Consider, for example, a solution of a strong mineral acid and a doubly weak organic acid. At the same hydrogen ion concentration, the organic acid has a concentration thousands of times as great as that of the mineral acid. Because of this difference in concentration, its diffusion rate is enormously greater, and very much more acid enters the cell. If the cell were a sac containing pure water, this difference in diffusion rate would not be important. For because of the weakness of the organic acid, proportionally more acid W would have to enter to produce the same degree of acid at 3, but the cell contains various substances which combine with acids. These probably combine with both weak acid and strong acid in the same stoichiometric proportion. The normality of the entering strong acid is much lower than that of the weak acid. The strong acid is thus more rapidly used up than the weak acid. It is thus easy to see that a weak acid may produce more acidity within the cell than a strong acid of the same hydrogen ion concentrate. As a matter of fact, it has often been found that organic acids are much more toxic for living cells than inorganic acids of the same hydrogen ion concentration. See, for example, Brenner-Height and literature cited by it. This greater toxicity of weak acids has generally been interpreted as being due to a difference in the permeability of the plasma membrane to different types of acids. Such a difference in permeability doubtless plays a part, but certainly the difference in diffusion rate is also an important factor. When cells are placed in buffered solutions of different hydrogen ion concentrations, it is obvious that the interior of the cell is not the same as the pH of the solution surrounding the cell. In order that the two be the same, we would have to assume that buffer and acid penetrated the cell at the same rate of speed, and that moreover the cell contains zero buffers of its own. It is easily possible to conceive of cases in which increase in the hydrogen ion concentration of the outer medium might be accompanied by a decrease in the hydrogen ion concentration of the cell interior, or vice versa. Such a case has been noted experimentally by Jacobs 20. In this instance, carbonic acid was present in high concentration in the outer medium, but the reaction of this outer medium remained alkaline because of the presence of carbonates. Being a weak acid, the carbonic acid had little effect on the external pH, but because of its high concentration, it diffused readily into the cell and there produced acidity. In interpreting the results of experiments with acids, we must therefore remember that the hydrogen ion concentration of the outer medium is not an accurate measure of the changes in acidity that occur in the cell interior. However, by a suitable choice of solutions, we can at least be reasonably certain that an increase in the acidity of the outer medium is reflected by a parallel, even though not by a proportionate increase in the acidity of the interior. Not absolutely certain, perhaps, for protoplasm is constantly producing acid and perhaps also alkali. And it is quite possible that with increase in alkalinity of the external medium, there is an increase in the cellular production of acid. Indeed, it is sometimes stated that alkalis do actually produce an increase in the rate of metabolism, that is to say, an increase in the amount of carbonic acid manufactured by the cell. In view of all these uncertainties, we must be very careful not to draw too far-reaching conclusions from the experimental evidence that we now have at our disposal. The action of acid and alkali on protoplasm has been studied in various ways. One or two authors have investigated viscosity changes. There have been a few observations on the effect of acid on protoplasmic streaming. And finally, several authors have described the morphological changes which follow acid treatment, both in bright field and dark field. Jacobs 22 studied the effect of carbon dioxide on the protoplasmic viscosity of paramecium and copidium. In these protozoa, the granules do not move very readily under the influence of centrifugal force. Jacobs overcame this difficulty by feeding the organisms India ink. Vacuoles containing India ink appeared within the protoplasm, and these moved very readily when the animals were centrifuged. Jacobs bubbled carbon dioxide through the fluid in which the organisms were swimming. He found that short exposure to this carbon dioxide produced a marked liquefaction in the protoplasm of both paramecium and copidium. Longer exposure caused an increase in viscosity. In paramecium, it was either-teasable to show that in this stage of increased viscosity, the viscosity was greater than at the beginning of the experiment. In copidium, the viscosity was at least as great, but it could not be shown to be greater. Jacobs also studied the action of carbon dioxide on the protoplasmic viscosity of sea urchin eggs. When Arbacia eggs were exposed for 5 minutes to seawater saturated with carbon dioxide, centrifuge test showed coagulation. The pH of the seawater saturated with carbon dioxide was 5.5. When the seawater was made acid by adding strong acid, and the carbon, the action of acids, and allyl dioxide was removed by shaking at a pH of 5.5, no coagulation of the protoplasm WA is produced. Apparently, therefore, at the same pH, carbon dioxide is more effective in producing coagulation than is strong acid. From what has been said previously, this is readily understandable. At a pH of 550, the concentration of carbon dioxide present is vastly greater than the concentration of mineral acid, which would be present at this pH. The carbon dioxide thus diffuses much more rapidly into the cell. According to Jacobs, the coagulative effect of carbon dioxide on Arbacia protoplasm is reversible. After a 5-minute exposure, the protoplasm returns to its ordinal fluid state. Jacobs also performed some experiments with Sparagira, centrifuging the filaments after exposure to carbon dioxide-saturated water. Here, too, short exposure caused liquefaction, whereas longer exposure caused coagulation. With one set of material, this coagulation, WAI, is apparently reversible, but in other material, it was associated with the death of the cells. Summing up these results, it is seen that carbon dioxide produced both a liquefaction and a coagulation in the protoplasm of Protozoa and Spiragira. In the Arbacia egg, Jacobs records only coagulation. The coagulation is generally reversible, although in one set of experiments with Spiragira, this was not found to be the case. That carbon dioxide may cause a reversible increase in protoplasmic viscosity is also indicated by the older experiments of Kuhne, 64, and Lopriob, 95. The latter treated stamen hairs of Tratus scantia with mixtures of carbon dioxide gas and air. He found that 80% carbon dioxide caused cessation of protoplasmic streaming after 3 to 4 minutes. When the stamen hairs were returned to air, the protoplasm began to stream again. It is, of course, an open question whether or not the effects produced by carbon dioxide can be attributed to the hydrogen ion or to the undissociated gas present in solution. Jacobs himself inclines to the view that the action of carbonyl dioxide is specific, and he cites experiments in which mineral acids at the same pH do not produce as powerful an effect as carbon dioxide. This is, however, readily understandable on the basis of our previous discussion, for at the same pH, carbon dioxide is vastly more concentrated than the mineral acid. On the other hand, there is good reason to believe that carbon dioxide in solution may produce effects quite apart from its acidity. A solution of carbon dioxide in water has a lower surface tension than the pure solvent, and might very well act as a fat solvent. In this connection, it is interesting to note that carbon dioxide is often used as an anesthetic in physiological experiments, and most anesthetics are known to dissolve fats. Each solvents typically produce a decrease in protoplasmic viscosity, see Chap 12, and perhaps the liquefaction caused by carbon dioxide is of this nature. Before going further, it should be pointed out that the addition of any acid may result in the liberation of carbon dioxide within the cell. Very probably protoplasm contains carbonates, and these would be broken down in the presence of most acids. Thus, when WUA treats cells with acid, we can never be sure that the effect is not due to carbon dioxide. The most extensive experiments on the effect of acid on protoplasmic viscosity are those of Sakamija and Lu 25. These authors centrifuged Spirogyra filaments after exposure to solutions of varying pH. The exposure time in all of their experiments was 5 minutes. In order to obtain solutions of different XUAA, Sakamaiwa and Lu used solutions of the two following types. In all, Sakamuba and Lu performed 11 experiments or series of XX experiments. The first 6 experiments were performed during the last week in February, and on the 4-E and 8 GU experiments, a solution of type A was used. The other 5 experiments were performed from the 7th to the of July, and the filaments were immersed in a solution of type B. The pH varied from 3.5 to 8.2. In measuring the viscosity, Sakamokra and Lu noted the percent of cells which showed movement of chromatophores after treatment with centrifugal force. They distinguished 4 different degrees of chromatophore displacement, from no displacement at all to a very conolete displacement of chromatophores, and they counted the percents of cells which showed each of these 4 degrees of displacement. From their data, Sakamvia and Lu conclude that, as the pH is changed from 3.5 to 8.2, the protoplasmic viscosity goes through 3 successive stages of increase and decrease. That is to say, if one were to plot a curve, the abscissae of which were pH values and the ordinates viscosity values, one should obtain a 3-peak curve, i.e. one with 3 maxima. Sakamcha values either for pH or for viscosity, and the curve as it stands merely represents the general conception of the authors. The points of high viscosity in the curve, Sakamchka and Lu regard as isoelectric points, but in a postscript they state that if protoplasm is a sol instead of a gel, the points of low viscosity represent the isoelectric points. Although these points of maximum and minimum viscosity are not located on the curve that Sakamura, which is copied from the original paper. It will be noted that the position of the points varies with the seasons, and Sakajviyuka and Lu state also that it varies with other external conditions. Protoplasm, according to Sakamura, and Lu for plants in the winter and spring. For plants in the summer, if it is true that protoplasm goes through successive stages of high and low viscosity, as the pH is varied, this is extremely interesting. In view of the fact that Sakamura and Lu's work has been generally accepted and frequently quoted, it may be worthwhile to inquire into the justification for their statements. The appended tables give all of Sakamura and Lu's data for least and greatest degree of displacement of chromatophores, of cells which show very strong displacement. It is hardly necessary to include data concerning intermediate degrees of displacement, for these appear to be less certain. Each row of figures in the tables is a single experiment. The first column gives the number of emutes the cells were centrifuged, and the other columns the percentage of chromatophore displacement or non-displacement at different hydrogen ion concentration. In all but the Fourier experiment, the centrifuge turned 2,000 times per minute. In experiment 4 it turned 1,500 times. It is to be remembered that the first 6 experiments were performed in the winter and spring, the other 5 experiments in the summer. Sakontra and Liu's observations, when collected together in the form in which they are presented in tables 2 and 3, scarcely carry conviction. The data are not consistent. Let us compare, for example, the 3rd experiment in which the filaments were centrifuged for 2 minutes with the 5th experiment in which the filaments were centrifuged at the same speed for 3 minutes. The shorter centrifugal treatment perhaps accounts for the fact that the percentage of cells which showed no disrelacement of chromatophores at pH 6 is 4 times as great for the 3rd experiment as it is for the 5th. But how explain that at pH 4 and at pH 4.3, the percentage of displacement is 2 or 3 times as great for the 5th experiment as for the 3rd? Examples of this sort can be multiplied many times. The reader may easily find them for himself. Let us consider now the evidence for Sakamuba and Liu's points of high and low viscosity. To take one example, for winter plants there is thought to be a point of maximum viscosity at pH 7.5. In all, there are only 2 measurements at this pH. In the first experiment, 40% of the cells showed no displacement at a low of 7.5. This percent is a little over the average for the series, although not much. On the other hand, in the only other experiment, the second 20% of the cells showed no displacement at pH 7.5. And this value, instead of being high, is next to the lowest one of the whole series of 10 determinations. Again, for summer centrifuged following exposure to solutions of varying pH, the acids and alkalis, CNJI, CQ, to the DO on plants pH point is said to be a point at which the viscosity edgy. What is the evidence? There are no nitroamides at pH 0.3. If one averages the 10 determinations at pH 5.2, A and D 4 determinations gives a value of 52%, and at UMM 5 a average of 4 determinations, 51%. How then a maximum in of viscosity finally for the summer plants, Saccharomyces and FAN, that below pH 4.7 the protoplasm is life fluid. But in the data that they offer, they show no record of tests at any pHa of 4.7. It is highly doubtful if a single one of Saccharomyces and Liu's eats of high and low viscosity has any Jex extification on basis of the experimental data that they have published. All the tables show is that the material they used in TLNDVXP fumates was highly variable. Robus 26 exposed eulodia leave us to the non sodium J ichiosphate. Solutions in VRI, the pH varied from 4 to 7.2. To Alawile, a 4-hour exposure to the solution, S, the logiv das fit mutu. The chloroplasts were thrown to OE vitivi, he udmeldus, he centrifuged at 2,000 revolutions per titino. RS, INT diffused at 650 revolutions per minute, or almost and those at 1,000 revolutions were not affected. Muelli no difference between the leaves from the acid and alkali-phos nigel of M. J. germes could be detected. Robins rather expected to fin. L such have one or perhaps differences in EDW been noted if tests had been made at SJOL's idern in TRN ETRL's idern and DRNL web bubble viscosity determinations. RIA Iochia acidified TLI umala eho agents for the onion cells. He states that, in general, acid solutions are more apt to produce a perfect convex plasmolysis. In neutral or alkaline solutions, the plasmolysis is imperfect or concave. Only in relatively high concentrations of acids does irregular or concave plasmolysis occur. Hydrochloric and sulfuric acids produce a concave plasmolysis at a pH of 2. Acetic acid produces the same result at more dilute hydrogen ion concentrations, pH 3 or 4 or 5. This is readily understandable on the basis of the discussion at the beginning of the chapter. Babat's results indicate that with an increase in hydrogen ion concentration, there is a progressive decrease in viscosity or liquefaction of the protoplasm, until finally, when the concentration of hydrogen ions becomes too great, coagulation sets in In addition to these observations on the plasmolysis form, Pebat also made some morphological observations, which will be referred to later. In this connection, it is well worthwhile to consider the effect of acid on the rate of protoplasmic streaming in plant cells. In spite of the fact that these results are always capable of a double interpretation, CP51 study. Clem 95 studied the effect of rather high concentrations of various acids on protoplasmic streaming in several different sorts of plant material. He found that the streaming first became irregular and finally stopped. Nuthman and Zukeb 12 also found that various acids caused a stoid page of protoplasmic streaming. LaQ 14 observed 0.5% sulfuric acid initiated streaming in 1 to 2 minutes. In this short time, it is hardly probable that the dilute sulfuric acid penetrated the cells, and it seems likely that the action of the acid was primarily on the cell surface, where it might very well have produced some change which would affect the driving force responsible for the streaming. Hobbies 26 studied the effect of solutions of different hydrogen ion concentrations on the rate of streaming in Alodia leaves. 13 sodium phosphate buffer solutions were used, and in these the pH varied from 4.8 to 7.2. The leaves were kept in the solutions for 5 to 6 days, following which rates of streaming were determined by noting the time required for a plastid to pass a measured distance. These times are given in the following tier. Different hydrogen ion concentration, Robbins, I6. From these observations, Robbins concludes that there is a slower rate of streaming in solutions of pH 6.4 to 7.2. It will be remembered that Bobbins found no differences in viscosity between the cells in the alkali and those in the acid solutions. It should be noted that in his viscosity tests, Robbins exposed the leaves for 4 hours instead of for 5 to 6 days. The evidence is not very conclusive, but as far as it goes, it indicates that the differences in rate of streaming do not depend on differences in protoplasmic viscosity. Strigeb 26 studied the effect of solutions of differing hydrogen ion concentration on the rate of hydrotoplasmic streaming in the root hairs of Hordeum vulgare, barley. He found the rate of streaming to increase from pH 6.8 to JDH 6.4, then to decrease markedly to a minimum value at about pH 6.5. At this point, the protoplasm stops streaming. Then, as the pH is lowered from 6.15 to 5.85, the hydrotoplasm flows ever more rigidly, reaching a maximum speed at the latter point. With further increase in hydrogen ion concentration, the speed of streaming rapidly falls off until it stops completely at pH 5.65. This is the lethal concentration. Such a succession of maxima and minima are very interesting, suggesting, as they do, possible isoelectric points in the protoplasm. We shall therefore examine the experimental evidence closely. The solutions used by Estebugor contain sodium acetate and acetic acid in differing proportions. Stedbx does not clearly state the concentrations of the solutions he used, but apparently at the higher concentrations of hydrogen ion, the acetic acid reached a concentration of 0.0032. Solutions of this concentration might very conceivably have an effect on the surface tension of the cell, for acetic acid in aqueous solution does have a markedly lower surface tension than the pure solvent. Stabud usually began his measurements very soon after the immersion of the root hairs in the solutions. In 3 of the 15 experiments he cites, he began measuring after 2 minutes. In another experiment, after only I minutes. 10 determinations were made in each experiment, and these required a time of 3 to 15 minutes. It is interesting to note that the measurements made at the end of this period do not differ appreciably from those made at the beginning. When the root hairs are immersed in the solutions, apparently an equilibrium is reached within 2 or 3 minutes, or possibly only 1 minute. This could hardly be the case if the acid produced its effect only after diffusing into the protoplasm. There is evidence, therefore, that the results of CBT-GGR are, to some extent at least, due to a surface action of the solutions he used. This is borne out by the fact that SCPT Jaeger clearly states that the volume of the protoplasm increases in certain of his solutions. Such an increase in volume might easily be the result of a lowering of surface tension of the protoplasm. The volume increase indicated in the diagrams and photographs of CFDB is a very large one, and would certainly result in a decided decrease in the protoplasmic viscosity, quite apart from any effect of the H ion. Rather a clear indication that the results of CF debugger with acetic acid are due in some measure to a surface tension effect is the fact that he finds solutions of chloroform give the same morphological picture as the acid solutions. Both in the presence of chloroform and in the presence of acetic acid, the protoplasm shows increase of volume with occasional bursting of the cell wall plasmatips, formation of new vacuoles, appearance of new granules, and also what Strugeby calls regeneration phenomena. It seems clear that in Strugger's experiments, surface tension lowering can perhaps also a fat solvent action of the acetic acid play a large role. From the morphological side, Strugger describes both an increase in the volume of the protoplasm and the formation of new granules within the protoxolasm. The increase in volume would, of course, tend to produce a lowering of protoplasmic viscosity for actual data, see P212, whereas the formation of new granules would certainly increase the viscosity. When the root hairs are subjected to various concentrations of acetic acid, there are thus two factors operative, one of which tends to decrease the viscosity and the other of which tends to increase it. As one or the other of these TAVO factors gains the ascendancy, it is easy to see that the viscosity would go up or down, and we might have any number of maxima or minima if we plotted viscosity or speed of streaming against hydrogen ion concentration. This is almost certainly a partial, if not a complete, explanation of the peculiar results obtained by Statku. It should be noted that this explanation does not depend on the assumption that surface tension lowering is responsible for the phenomena, but merely on the more geological observations made by in a paper which has just been published. Statue 28 again studies the effect of various hydrogen ion concentrations on barley root tips. The material is thus the same, but in the newer experiments, the hydrogen ions are due to phosphoric acid. There is thus not the same difficulty in regard to surface tension, but unfortunately, in Esabukek's new experiments, he introduces another complicating factor. Plant roots are very sensitive to osmotic conditions. The ordinary nutrient solutions in which they can live are very dilute. The solutions which Spugga 28 uses are apparently too concentrated, and moreover, they do not have the same osmotic strength. Between a pH of 6.8 and 7.5, there is a variation from AEA, approximately M15 to M25 solutions, at HGA Drogen ion concentrations, just under neutral. This is quite contrary to the older work of Kallenbag and TBGA 96 and HIID 96. These authors found that roots of lupinus and C. curbita could live and continue to grow for at least 2 days in N6000. 400 solutions of hydrochloric, nitric, sulfuric, and other mineral acids, whereas ZA roots could even withstand N3200 solutions. And T16400 solution has a pH of about 3.8. Kallenbeg and True exposed the roots for 2 days, or shorter exposures. Beneb 18 found that various plant tissues showed a tolerance for higher concentrations of acid. Mevius 27 grew several types of plant roots at a pH of 3.3. Adams 2 found no evidence of injury in roots of wheat seedlings grown at a pH of 3.68 for a week, see below. In addition to C. Bugab, various other authors have described visible changes in protoplasm following treatment with acids and alkalis. All observers agree that acids, when georesent in sufficient concentration, cause the appearance of new granules within the cell. Clem 95 describes Cornish Ausscheidungen, that is to say, the precipitation out of granules, following treatment with nitric, sulfuric, hydrochloric, phosphoric, chromic acids, as well as by citric, oxalic, acetic, and other organic acids. Clem used a concentration of 0.05% nitric acid, and the other acids were used in about the same percentage strength. No statement is made regarding normality. The plant material used was of various sorts. Root hairs of Trianea and Mormodica, leaf cells of epidermis cells of Tridescantia, etc., according to Clem. Not only may acid cause the appearance of new granules, it may also cause the formation of many small vacuoles within the protoplasm. Pratt 26 states that granules increase in number in onion cells following acid treatment. On the other hand, in the presence of alkahis, there is a tendency for the granules to disappear. A clear description of the formation of granules as a result of acid treatment is given by Lewis 23. She studied cells in tissue culture. In these cells, the cytoplasm is normally free from granules. Lewis studied both cytoplasm and nucleus, and she states on the basis of morphological evidence that coagulation of the nucleus occurs at a pH of 4.6 in all of the following acids: hydrochloric, sulfuric, nitric, acetic, lactic, citric, oxalic, and picric. That all of these inorganic and organic acids should produce an effect at the same pH is rather surprising, for the normal concentrations of the acids and their rates of diffusion into the cell must be markedly different compared to discussion on pH. 184. Lewis claims that the coagulative effect of acids is reversible, that acid coagulation of protoplasm may be reversible, is also indicated by the observations of Heilbrunn. 25. She studied human leukocytes in saliva. Normally, these leukocytes show both amoeboid movement and Brownian movement of granules in their interior. Both amoeboid movement and Brownian movement ceased when the cells were placed in OI%, acetic acid in 0.9% sodium chloride, on washing with 0.9% amoeboid movement. Lewis studied not only the action of acid, but the action of alkalis as well. She used solutions of ammonium hydroxide, ammonium carbonate, lithium carbonate, and sodium carbonate, and found that at a pH of 8, 6 to 9, there was apparently an increase in the fluidity of the protoplasm. At any rate, the cells tended to round up in these solutions. This is, of course, not very good evidence of greater fluidity, as many factors may be involved in CCB, a change in shape. The effect of the alkalis was reversible. Chapek 23 states that Borish in his laboratory treated exuded protoplasm of Vauderia with dilute alkali. Following this treatment, there was an appearance of droplets in Brownian movement. Zapek's statement is too brief to be very helpful. The appearance of droplets would indicate a viscosity increase, provided that they were new formations. The fact that they were in Brownian movement might indicate a viscosity decrease. If there were no Brownian movement before treatment, unfortunately, Zapek's words are ambiguous. In plant cells, alkalis cause the appearance of many new granules. This was first clearly stated by Darwin in his studies of Drosera rotundifolia published in 1875, after showing that ammonium carbonate is very effective in producing what he calls an aggregation of the protoplasm of the leaves. He goes on to show that in the cells of the roots, ammonium carbonate caused a cloudiness of the clear fluid which these cells contained, so that this fluid came to look like a mezzo tinto engraving. In later studies, Darwin 82 showed that ammonium carbonate in solutions of 0.1 to 0.7% caused the formation of many granules in the root cells of various PLR, including, for example, Euphorbia peplus. Even solutions of 0.01% were effective. Apparently, Darwin saw the granules in the cell sap rather than in the protoplasm. PFEP 86 extended Darwin's experiments. He describes the formation of granules in the cell sap of the root cells of Azolla, Euphorbia peplus, Icetus following exposure to solutions of ammonium carbonate, sodium carbonate, or J-potassium carbonate. He also saw similar granules appear in the vacuoles of Escherichia cells. Bocorni 88 states that the granules appear not only in the vacuole, but in the protoplasm of Spirogyra. And he shows many figures of granules produced following treatment, WEC ammonium carbonate. When 1% ammonium carbonate was used, the granules formed very rapidly. They appeared more slowly when solutions as dilute as 0.05% were used. Clem 95 showed that alkalis cause the formation of numerous vacuoles within atherian cells. There is also at least one older observation of the same sort, Cone 54, and there are probably others. Degbin's studies of the phenomenon are especially interesting. Degen 5. He found, for example, when the mycelium of Aspergillus niger was treated for 10 to 15 minutes with 0.1% NAL, that there was a production of so many vacuoles within the protoplasm that the whole mass of the protoplasm became foam-like in appearance. Similar results were obtained for the mycelia of Mucorstolonifer, Dimatium pullulans, and Saprolegnia. These forms, concentrations of 0.2 to 0.5% now were used. Deegan also describes the formation of vacuoles following treatment with alkalis and root tips of Vicia faba in the pollen mother cells of Lydia, Candidum in the root hairs of Trionea and Helianthus, in the gland hairs of Syringa, and C or Eulus axide in the hairs of Urtica. Yamaha 27 also describes the appearance of vacuoles following treatment with alkali. He experimented on the root tips of Vita faba and other plants, but he studied ogilvy fixed material. Abuum's 23 studied the action of solutions of varying pH on the root hairs of wheat seedlings. In her solutions, the acidity was due to phosphoric acid. The root hairs were examined in the dark field with the aid of a cardioid condenser, and the time of exposure was a week. Adams determined coagulation from the general appearance of the protoplasm in the dark field. She states that there is no evidence of coagulation in solutions of pH 3.85 and 3.68, but at pH 3.6 and 3.47, she believes coagulation to occur. Adams' results are consistent with the older observations Kallenbeg and Tabu and of Held, see above. Attention should perhaps be called to the fact that there are a number of GREs in the botanical literature which deal with the effects of acids and alkalis on growth. Thus, Loy 18 showed that BOLN grew faster when either acid or alkali was added to the culture medium. Other authors have shown that when the rate of growth of seedlings or roots is plotted U against pH, a curve with two maxima is obtained. For literature, consult Robbins 23. Robbins believes that the minimum between the two maxima represents the isoelectric point of the protoplasm. The growth of seedlings or roots is a complicated XG process and can hardly be expected to depend on any one physical property of the protoplasm. The entrance of water into the seed must depend to some extent on the colloidal properties of the seed coat. Doubtless, the lifeless cell wall of the plant tissue plays a role, and there is also the plasma membrane to be considered. Moreover, it is hard to see how one can exclude teleocleonein, all PDT cesses of metabolism, from consideration. These are QTON to be variously affected by differences in concentration of iwood and alkali. In attempts to determine the isoelectric point of protoplasm, it should also be clearly understood that JIID of the interior of the cell is not the same as the pH of its environment. In resume, it may be stated that there is general that EID in sufficient concentration causes coagulation of the protoplasm. Various observers note an increase in the GRIA as of the CCDL as the hydrogen ion concentration is increased Clyats-2Gib. The coagulation produced by acid appears to be reversible, at least in some instances, Wicks-Herwarden. There is also some evidence that allyl or diiodoyoid below those which cause coagulation, a decrease in viscosity of the protoplasm as conopamol, Wilgi neutralit. Jacobs found a decreased viscosity following TRTAL melee of protozoa with carbonic acid. Pratt's observations in detail an increased fluidity of the protoplasm of onion cells following equal treatment. There is also a possibility that tluifuen solder rate of streaming in Elodia cells in acid saltiogeous poems may be due to viscosity differences. The liquefying action of acids when it occurs may be on Luke 5 to a fat solvent action of carbonic acid Sitherihu or Rilnujli or Exbuduite by a reaction of acid with carbonate. Or it may possibly be due to a direct effect of the hydrogen ion in Rodat-Singh Ibuiv charge at the surface of the granule. It would teal nui ul like the calcium ion or the aluminum iona. Hoc chapter 10. If Lenortina, there are two may possibilia. Regarding region, the action of alkalis, there is a diffoivancua of the action of acids and alkylation of protoplasm following exposure to dilute solutions of alkalis. It is obvious that our knowledge regarding the action of acid and alkalis on the colloidal properties of protoplasm is far from satisfactory. There is a real need for careful and comprehensive experiments on various sorts of material with different acids and alkalis. Chapter 13. The action of fat solvents. The action of fat solvents protoplasm is extremely sensitive to fat solvents. Very dilute solutions of ether and chloroform have a profound effect on the life of the cell. All fat solvents are anesthetics. They tend to repress or inhibit the activities of the living substance. The theory of anesthesia will be considered in a later chapter. For the present, we are concerned with the physical effect of fat solvents on protoplasm, quite apart from any relation this might have to the life of the cell. Beyond any question, the action of fat solvents is manicided. In the fattiest place, all fat solvents are liquids of low surface tension. Even in great dilution, their aqueous solutions have a surface tension decidedly lower than that of water. When living cells are placed in such solutions, the boundary surface between the cells and the surrounding fluid has its surface tension lowered. If the cell is surrounded by a relatively thick membrane, there is also a lowering of surface tension at the boundary between this membrane and the inner protoplasm. The result of Clese surface tension lowerings is that the cell is no longer subjected to so great a pressure from without. The equilibrium between the various osmotic, elastic, and surface forces no longer holds, and the cell tends to expand. Obviously, the effect is greater in those CUS in which surface tension forces plays a larger role, that is to say, in smaller cells, but even in cells as large as sea urchin eggs, the diameter of the cell can be seen to increase very markedly when the eggs are placed in solutions of low surface tension. The same effect can also be noted in large protozoa, for example, in Paramecium. The action of fat solvents, it seems logical to assume that the increase in volume of cells which occurs in solutions of fat solvents is actually a direct result of the surface tension lowering. One fact speaks against such an interpretation. As the concentration of the fat solvent is gradually raised, there is a more or less definite point at which the increase in volume suddenly occurs. Before this concentration is reached, there is often little or no effect. Thus, Arachi eggs expand scarcely at all in 2% ether, whereas in 3% ether, they very quickly undergo a large increase in volume. The surface tension of a 3% ether solution against air is not very different from that of a 2% solution. One would expect, therefore, that as the concentration of the ether is raised, that there would be a marked increase in volume at first, and then a more gradual increase. Perhaps the reason why this does not occur is as follows. The Arachi egg is surrounded by a rigid or semi-rigid membrane. The rigidity of this membrane offers resistance to an increase in volume of the cell. As the surface tension is lowered, there is first not enough of an effect to overcome the rigidity of the membrane. When higher concentrations of fat solvent are used, there is suddenly a point at which the rigidity of the membrane is no longer sufficient to keep the volume small, and the cell increases in size quite suddenly. This explanation is not entirely convincing, but whether or not it is correct, it is at least certain that in higher concentrations of fat solvent, cells Veron often undergo a marked increase in volume. This is a factor that sometimes becomes of importance in the interpretation of the behavior of protoplasm toward fat solvents. Many authors have studied the action of fat solvents on gelatin and other proteins, and their results have often been taken as valid for protoplasm. With the development of the first methods for the examination of the colloidal behavior of protoplasm, it soon became apparent that protogelasm in no case acted like any simple J-diotene, and that particularly in its relation to fat solvents was the living substance unique. There is also another possible explanation of the increase in volume of cells exposed to fat solvents. Perhaps the fat solvent releases bound electrolytes, compare Rofe and Aldixon 7, and in this way increases the internal osmotic pressure. Against this explanation is the fact that substances like toluol, which age apparently do not enter the cells, also cause increase in volume. In recent years, determinations of protoplasmic viscosity in various types of animal and plant cells have shown conclusive forgery that dilute solutions of fat solvents cause a decrease in viscosity, whereas somewhat more concentrated solutions produce coagulation. These facts were already suggested in the older literature of rotoplasmic streaming. Thus, Heilbrunn, 95 states toluid alloy alcohol is gradually added to a slide preparation of the leaf of an aquatic plant. There is a marked acceleration in the rate of phagoplasmic flow. Only when a relatively high concentration of alcohol is reached does the flow become slower until it gradually ceases. Following Heilbrunn, various other observers showed lower concentrations of fat solvents had an accelerating of protoplasmic streaming, whereas higher concentrations had an opposite effect. Thus, UAC 3 states that when alcohol is radially added, so as to avoid shock, to Chametella, Myodvanventifilcrea, or Trinea protoplasm, there is an NCRAE in LIATC of streaming and concentrations of 1 to 2%. At stronger concentrations, the rate of flow is retarded or stopped. Generally, according to E. Yale, dilute solutions of T. tulier and L. lorokinia accelerate streaming, W for E or stronger concentrations, R, E, L, or stop it. Joseph Oy also reported observations of TIN's sort. Yik-10 studied the rate of streaming in myxomycete eoplasm. D2 found that dilute ether solutions caused it to increase in rate of streaming and that more concentrated solution load or stopped it. Botanists have usually thought that aeration of protoplasmic streaming by dehydrogenate solutions of ether is the result of a stimulation, such as is sometimes timed to occur in animal tissues exposed to dilute solutions of anislage. It has therefore been more or less generally supposed that he of the fat solvents was on the motive force of the Heilbrunn of the results that we shall kuichlorhal findel and proto nila eserminacho. Following treatment with dilute solutions of fat LWD-NT, I've the key decrease in viscosity than to any stimulation and protoplasmic streaming, the gravity method. He found that in LCI H5, the action of fat solvents is hard to tell exactly which concentrations of ether Heilbrunn used, for he gives his figures in percents of a concentrated aqueous solution rather than in actual percents of ether. In Heilbrunn's experiments, both the state of decreased viscosity and the state of increased viscosity were reversible. Weber 22b repeated the earlier work of Heilbrunn on bean cells, paying particular attention to the higher concentrations of ether. He studied the starch sheath cells of Phaseolus vulgaris and made viscosity determinations with the centrifuge rather than the gravity method. His results show that 2 and 5% ether by volume cause a marked increase in viscosity W when the cells are exposed for an hour. The viscosity increase is at least 12-fold, as shown beast relative times necessary to cause movement of starch grams in etherized and control cells. The viscosity increase produced by 2%, 5% ether is reversible. Exposure to 10% ether produced a more pronounced increase in viscosity, which was irreversible. Heilbrunn 17, 20A, 20B studied the effect of ether and other fat solvents on the protoplasm of sea urchin eggs. Using the centrifuge method, he was able to show that various fat solvents caused a decrease in viscosity when present in certain dilute concentrations. In somewhat higher concentrations, they produced a coagulation with a great increase in viscosity. The decrease in viscosity produced by dilute solutions of fat solvents was a phase reversible. On the other hand, the coagulation caused by the more concentrated solutions was always irreversible and involved the death of the cell. In a later chapter, it will be shown that the decrease in viscosity produced by the fat solvents is related to the anesthetic action. these reagents have in preventing cell division in the sea urchin egg. Or the present, we are concerned only with the physical action of the fat solvents and not with any physiological correlations. Heilbrunn 20b published a list of fat solvents with the percents of solution which cause decrease in viscosity and coagulation in sea urchin eggs. In this HSD, no statement was made with regard to the length of exposure. This is really an important factor. Sometimes a solution which causes first a decrease in viscosity on longer exposure causes coagulation. The list published shows the concentration of reagent found to cause a decrease in protoplasmic viscosity. The length of exposure to this concentration of reagent is given in the third column. In the fourth and fifth columns are given the concentrations found to produce coagulation with the length of exposure actually employed. All the data refer to the fertilized eggs of Arbesia. The percents given are volume percents of solution found to decrease Visa solution found chloral hydrate, citronethane, paraldehyde, ethyl nitrate, ethyl butyrate, acetonitrate, propyl alcohol, and amyl alcohol versus SAT, ethyl luthane, mints. And it is presented here only because more accurate data are lacking. Such data are easy to obtain. It would be a simple matter to investigate much more thoroughly the effect of various fat solvents on the physical state of the protoplasm. Careful the action of fat solvent study in this direction might yield many results of considerable value. In a short statement published in 1924, Chambers states that 2% ether does not produce a decrease in viscosity, but rather an increase in the protoplasmic viscosity of sea urchin eggs. He bases this opinion on observations of Brownian movement in etherized eggs, as well as on microdissection tests. However, as pointed out by Heilbrunn 25, these observations of Chambers are irrelevant. In estimating the rapidity of Brownian movement, Chambers employed an arc lamp for dark field illumination and took no precaution to protect the eggs from heat. Arbesia eggs die at only a few degrees above room temperature, and as shown previously at CP 120. Their heat death is hastened by ether, doubtless in his estimates of viscosity with the microdissection needle. Chambers was influenced by his studies of Brownian movement in the heat, coagulated eggs, and was thus led to conclude a higher viscosity when there was actually a lower viscosity, because of the above-mentioned statement of Chambers. Heilbrunn 25 re-examined the question of the effect of ether solutions on the protoplasmic viscosity of sea urchin eggs. Both the unfertilized and the fertilized eggs of Arbesia were studied. Solutions in seawater on the protoplasmic viscosity of unfertilized eggs, the time of exposure, as given in the second column, is important, especially for the experiments with 3% ether. When eggs are exposed to this concentration of reagent, the viscosity of the protoplasm decreases gradually, passes through a minimum, and then after about half an hour, the stage of low viscosity is followed by an irreversible coagulation. It is due to the fact that there was some difference in the criteria employed in determining viscosity in different experiments. Ether reduces the viscosity of unfertilized Arbesia eggs to 53% of its original value, whereas 3% ether reduces the viscosity to 48% of what it is in the controls. No great claim for accuracy is made for these figures. It is nevertheless certain that dilute ether solutions do cause a very pronounced decrease in viscosity. The effect is even greater when the experiments are performed unfertilized. Allowing fertilization in the Sean urchin egg, there is a sharp increase in viscosity, see Chap 15. When fertilized eggs of Arbesia are placed in 2% ether, at a time when the viscosity of the protoplasm is at its height, the viscosity soon becomes lowered to 1/6 or even 1/8 of its original value. Eoth in the starch sheath cells of the bean plant and in the eggs of the sea urchin, dilute concentrations of fat solvent produce a decrease in protoplasmic viscosity, whereas somewhat greater concentrations cause viscosity increase or coagulation. These results are not peculiar to the two types of cells mentioned. Various authors have obtained similar findings for other sorts of protoplasm. Heilbrunn 22 performed some experiments on the effect of ether on plasmodia of the slime mold Reticularia. As a result of these experiments, he concludes that Schwarzsnarkose erhot, why früher vier wieder Faba festgestellt, auch bei Reticularia die Liquidität, stark die Zähigkeit. In other words, low concentrations of ether decrease the viscosity, higher concentrations increase it. Because of the fact that direct contact with ether solutions apparently injured the slime mold, Heilbrunn exposed the plasmodia on a moist glass slide to ether vapor. With such a procedure, it is hard to regulate the exact amount of ether to which the protoplasm is exposed. Because of the uncertainty of the conditions of the experiment, we shall not cite Heilbrun's the action of fat solvents observations in detail. His general conclusion, as given above, is surely justified. In 1921, Webb 21C published a long series of experiments on the effect of ether solutions on the cells of the alga Spiragira. The viscosity was tested by the centrifuge method. WB's results are very clear and convincing. With weak concentrations of ether, that is to say, concentrations of 1 to 2 Ju%, there is a diminution of viscosity when the filaments are exposed 1 to 2 hours. With higher concentration, about 3% or over, there is an increase in viscosity. The PBR cents are volume percents. At the critical concentration of 2%, 3%, the effect varies with the time of exposure. A 1-hour exposure causes a decrease in viscosity, whereas an exposure of 3 or more hours has the opposite effect. These results of Weber show that Spiragira cells behave toward ether concentrations of 1 to 3% in almost the identical fashion that sea urchin eggs behave. The similarity of concentrations and times of exposure are required to produce decrease and increase of viscosity in the two widely different types of protoplasm is truly remarkable. There is, however, one difference that the plant cells show. In sea urchin eggs, only the decrease in viscosity is reversible. The increase in viscosity produced by higher concentrations of the fat solvent is associated with death. On the other hand, the Spiragira cells can also undergo a reversible increase in viscosity, although with concentrations of ether higher than 3%. The viscosity increase is just as irreversible as it is in sea urchin egg. The difference between the plant and animal cells may in part at least be occasioned by the presence of the cell wall around the plant cells. When a sea urchin egg is exposed to a relatively concentrated solution of a fat solvent, it undergoes a change sometimes called cytolysis, and this cytolysis is often associated with a marked increase in the volume of the cell. Such a volume increase would be prevented by the cell wall of plant cells. In a later paper, Weber 24D showed both by the plasmolysis form method and by centrifuge tests that a half-hour treatment with 10% alcohol has a pronounced liquefying action on the protoplasm of Giardia cells. Using the plasmolysis form method again, he was able to demonstrate a decrease in the protoplasmic viscosity of the stalk cells of Colloid repens, one of the Gomellinaceae, following an hour's exposure to 2% ether. Finally, Balabatic 25 has described a Viacody decrease in the protoplasm of Alochlea cells following tretinoate with chloral hydrate. However, the method used by Belarodmic is open to serious question, CP51. Rusley 28b, an increase in Brownian movement when Joamuba is oxy to ether, chloroform, or alcohol. The action of fat solvents has been carefully studied in TIA plasmodia, in the leaf cells of Elodia, and in the stalk coals of Colloid repens. The results on all these different typos of protoplasm are fully concordant, and it can now be regarded as solid fact that in general dilute solutions of fat solvents kechiize a decrease in protoplasmic viscosity, whereas more cononitrat UL solutions cause increase in viscosity or coagulation. It should perhaps be stated that Edisot.U and Doniam 27 have examined under dark field illumination Isolational frog nerve fibers treated with various alcohols. Chioi Koyuchi from their studies that ethyl, methyl, and propyl alcohol. It has already been pointed out that Tui-Darkvi dial method is not applicable to the study of the interior of the NOR-VT fiber, CP159. It might also, he mentioned that, and Joachim's results with ethyl alcohol do not accord with WDL unavu physiological facts. Etish and Joachim's report EUA Liahig action with ethyl alcohol solutions of 0.3 to 0.9% by volume. DNL. Many observations in the literature show that much higher concentrations of alcohol have no deleterious effects on in-wit nerves. Thus, to quote only one or two instances, Lehi Chiek and Tu found that when they injected alcohol up to I5H of the weight of a frog, i.e. about 2 by volume, there was. In Thoho experiments, the nerves of the frog were almost certainly bathed in a solution of alcohol of about 2%. Yologby 27 raised tadpoles in one HEVL observations of the same sort could be cited. It's Sketio, meaning that Edish Joachim's results are due either to AEO the action of fat solvents before leaving the subject of the action of fat solvents. And before considering the possible explanation of the peculiar effects produced by these reagents, we shall make brief mention of the effect of ordinary distilled water on protoplasmic viscosity. This discussion is not out of place at this point, for it seems improbable that water alone does actually exert a solvent action on the fats of living cells. Thus, Hanstein CB 22 describes a solution of the fats or lipoids of the outer protoplasmic layers of plant cells when these are treated with distilled water. Moreover, when sea urchin eggs are exposed to solutions containing a high percentage of distilled water, there is apparently a disappearance of the fat particles which ordinarily show so plainly when the egg is centrifuged. Approximately 15 minutes if the exposure to the hypotonic solution is 30 minutes instead of 15 minutes. Then coagulation occurs in 60% distilled water, probably infinity, but in these determinations of the viscosity of the protoplasm of cells in hypotonic solutions, there is a large source of error which must now be considered. In the hypotonic solution, the cell increases in volume, and the distance between the adjacent granules accordingly becomes greater. In a previous discussion, CP 58, it has been shown that the ease with which the granules move through a cell depends in part on the distance which separates them from one another. Cunningham's factor, which we have called Q, is less as the granules are farther apart. In order to determine the effect of this change in Cunningham's factor, Q was calculated for the case in which the eggs are in 25% distilled water, i.e. 75% seawater solution. For method of calculating Q, see chap 5. As a result of the calculation, Q was found to be approximately 8.1, that is to say 77% of what it is when the eggs are of the normal size. Correction for the viscosity value obtained for 25% distilled water shows that the proper value should be 9 instead of 7. The viscosity of the protoplasm of the Aracia egg in 25% distilled water is thus about CO% of what it is in seawater. An interesting check of this method of calculation may be obtained by studying the effect of hypotonic solutions in the cold. At a temperature of about 4 dC, the fat solvent action of the distilled water is much lessened, so that the main effect on the speed of granular movement under the influence of centrifugal force is apparently the change in Q. In one experiment in which Aracia eggs were exposed to 25% distilled water for half an hour at 4E, the speed of granular movement at the end of this period was 71% of what it was in the control eggs in seawater. The action of fat solvents from this experiment, it is certain that when eggs are placed in 25% seawater, Q cannot be less than 71% of its original value. It is certain, therefore, that when the protoplasm of the sea urchin egg becomes diluted with distilled water, there is actually a pronounced decrease in viscosity. This is quite comparable to the effect of ether. In 25% distilled water, the viscosity of the Aracia protoplasm is about 60% of its normal value. In 2% ether, it will be remembered the viscosity drops to 53% of what it is in the controls. Moreover, Just as in higher concentrations of ether coagulation occurs, so too as the concentration of the distilled water increases beyond a certain limit, there is a sudden coagulation of the protoplasm. It has repeatedly been shown that fat solvents cause a decrease in the viscosity of the protoplasm when they are present in relatively low concentration, whereas in higher concentration, they cause coagulation. Obviously, a most important question is the reason for this peculiar behavior of the protoplasmic colloid. This is a question which, for the present, we must leave unanswered. That dilute solutions of ether and other fat solvents should have such a pronounced effect on the protoplasmic viscosity certainly indicates that lipoids are a very essential part of the living substance. It seems not at all unlikely that TI micellae of the protoplasmic colloid are surrounded by a lipoid film. Some evidence in support of this view has already been presented, CP 30. But exactly how a dilute solution of a fat solvent would affect such micellae so as to decrease the viscosity of the whole colloid is by no means clear. Concerning the coagulative action of more concentrated solutions of fat solvents, we now have some real information. This coagulative action can be seen morphologically to be accompanied by certain distinctive visible changes. In Arbacia eggs, for example, there is a very noticeable loss of pigment from the pigment granules of the cell, and this is soon followed by the appearance of numerous vacuoles throughout the protoplasm. It may be somewhat more, for in the cold the viscosity of the protoplasm tends to be lower than at room temperature, and this would make the relative value of Q2 low. But the effect of cold is more or less neutralized by fat solvents, Heilbrunn 20A, and hence probably also by distilled water. All these changes are associated with a very definite and characteristic series of reactions, which are found not only in sorchin eggs, but in other cells as well. The fat solvent, when present in sufficient concentration, can initiate this series of reactions, and the ultimate result is a coagulation of the protoplasm. What is known concerning the nature of these reactions and how their initiation depends on the gerosince of fat solvent will be discussed in a later chapter, see Chapter 14. Chapter 14. The surface precipitation reaction. When a living cell is cut or torn so that its interior flows out, a film is immediately formed at the surface of the emerging protoplasm. At any rate, some cells behave in this fashion. The phenomenon is well known through the oft-quoted observations of Nageli 55. And Pifu, many books refer to these observations as though they were practically the only ones on the subject. As a matter of fact, there are numerous similar observations scattered through the literature of the last 150 years. Merely from the standpoint of historical interest, it may be worthwhile to mention some of this neglected literature. The early students of protozoa knew that when a protozoan was injured or crushed, the protoplasm might disperse through the surrounding medium. To the French workers, this was decomposition par diffluence. The Germans refer to it as a Zerfleihen. But not always did a protozoan show diffluence. Sometimes, as the protoplasm began to emerge from the bounding membrane of the cell, it became coated with a film so that a droplet or droplets were formed, which were sharply delimited from the surrounding medium. These droplets were called by Hujabdira globules de Sarcode. Following DXRB, the Germans called them Sarcodtrophen. They were seen by observers long before Dijambin. Mulleb 1786 describes sarcode droplets in a species of Uloda, and in an injured specimen of Corona, he calls them moleculae mucidae. The early observers did not know how to interpret the globular extrusions of protoplasm, and they usually thought of them as representing organs of one sort or another. Uriarisbret believed them to be stomachs. Dujayadin gave the first sensible interpretation. See, for example, D. Exichardan. There can be no question but that the sarcode droplets of Dujabdik and the similar appearances described by authors before and after him really represent the form of reaction that we shall consider in the present chapter. One of Dujardin's figures is shown in Figure 9. It is taken from his monograph published in 1841 and shows film formation around droplets of protoplasm emerging from Leucophorus striata, a protozoan parasite of the earthworm. Following Dujardin, sarcode droplets were described and figured by various subsequent observers. These include many students of protozoa interested either in the function of the nucleus or in the capacity of parts of the cell to regenerate, have performed cutting experiments on many different types of single-celled organisms. In the course of these experiments, it has often been incidentally noted that the naked protoplasm Leucopleura striata, after Dutardin 41, exposed by the cut, was capable of covering itself with a surface film. Indeed, it is probable that only in those cases in which such a film forms rapidly are cutting experiments possible. Modern books on protozoa contain many references to experiments in which different types of protozoa have been cut. Some of the early work is referred to by Gruber 8693. From the first, amoeba and stentor were favorite forms for exodermentation, and it is obvious that in these cells the naked protoplasm must quickly form a membrane or film. Cutting experiments have not been confined to protozoa. Students of experimental embryology have frequently cut or broken up the egg cells of marine invertebrates. Most of these experiments were performed on sea urchin eggs. In the embryological literature, one finds very little said about the formation of a membrane or film at the surface of naked protoplasm. This is in part due to the fact that, for a long time, there was a difference in opinion as to the true structure of the sea urchin egg. The outer membrane of the unfertilized egg has a refractive index close to that of seawater, and it is not very clearly visible. In the early part of the 20th century, it was generally believed that the sea urchin egg was naked, in spite of the fact that previous workers had clearly described the egg membrane or vitelline membrane. Concerning the Arbasia egg, Bill Braggs, EG1D5A states, if the egg contents be made to flow out from the membrane, or if the egg be cut or shaken into fragments, a new membrane immediately forms about the momentarily naked protoplasm. Such a membrane has the same chemical properties as the vitelline membrane. It is much easier to break a large cell than a small one. This explains why practically all the observations on cut or torn cells have been made on protozoan or egg cells. There is, however, one old observation on small cells which is very interesting. Henson 61 crushed blood cells of the frog, and in one of his figures he shows the formation of a membrane or film about the droplet of exuded protoplasm. In view of our later discussion, it is worth mentioning that the emerging protoplasm from the blood cell loses its pigment. This is perhaps comparable to the loss of pigment from protoplasm pressed out of I. Arbasia egg, see below. The literature on plant cells really begins with Naoelt 55, although one finds occasional instances in which still earlier workers apparently noted the formation of a membrane or film about the surface of protoplasm squeezed out of cells. See, for example, Cone 54. Naegeli clearly describes the formation of a membrane about droplets of protoplasm which have emerged from Cleara cells. He also studied the same phenomenon in the colored cells of grapes and other fruits. Following Naegeli, there is a long succession of workers who have described the formation of membranes about naked protoplasm. DeBerry 59 studied the behavior of naked slime mold protoplasm. Hanstein 72, cited by Strasbourg 76, described the formation of films about exuding droplets of Vaclaria protoplasm. See also Hanstein 80. Line 72 cut the mycelium of the fungus Pilohalus and noted the emergence of droplets Phantium 75 notes film formation following wounding in the mucorin. Stiesbuger 76 describes the emergence of irotoplasm from swarm spores of Vaucheria sessilis, and he says that a precipitation membrane is formed at the fresh surface. Strass-Puchiger also describes the e.g. formation of precipitation films on the freshly formed surfaces of Plasmodia of Aethylium cephticum. Peak 77 crushed root hairs of Hydrocharis and saw films form at the surfaces of the emerging droplets of protoplasm. This work has been often quoted. Sachs 82 figures film formation around the naked protoplasm of a crushed filament of Vaucheria, and he cites older observa A from Sachs 82, B, C, D from Hanstein 8. B, 0 and D show successive stages tines of Schmitz on Valonia. Pfeiffer 90 made some observations on slime mold protoplasm. There have also been some newer studies, Prowazek 7 on Vaticuria, Stubble 8 on Nitella. Cephas 21 on Usaphus, on eggs of Fucus, and on pollen tubes of Iris. Nichols 22 on Voschigeria. Cladophora, Chiamera, and Natella, and Lepeshki 26b on Bryopsis. It is apparent that one of the favorite objects of study has been Voschigeria, and many figures have been published for this type of protoplasm. AG10 is taken from Sachs 82 and from Hochstein 80. Sachs's drawing is especially interesting in that it shows the formation of vacuoles in the exuded protoplasm. We shall have frequent occasion later to refer to such vacuole formation. Figure 11 is taken from 590 and shows the formation of a film or membrane at the cut surface of slime mold protoplasm. Ill view of the large number of papers that have been written on the formation of membranes on the surface of protoplasm. Exposed in one way or another, it is rather surprising to find that some recent authors apparently regard the subject as new. Thus, Cephas 21 and Nichols 22 write as if the FHSD experiments were made with the microdissection needle. In books on cytology and general physiology, it has often been figure 11. Reaction at the cut assumed that the protoplasm of all surface of slime mold protocells forms surface films or memplasm after Pfeffer 90 brains on exposure to the surrounding medium. Perhaps in a certain sense this may be true, but as was Leon to Muller and Dujardif many years ago, if one crushes various types of cells, one frequently finds that the protoplasm flows out through the surrounding medium without the appearance of any film or membrane. Thus, though very beautiful films may be obtained with stentor, some races of paramecium show no film formation when the cells are pressed under a cover slip. In a few tests with colpidium, it was found that usually when the cells were crushed under a cover slip, there was no appearance of a film about the emerging protoplasm. On the other hand, if the pressure to which the olpima were subjected was a very gentle one, films could sometimes be seen to form. As a matter of fact, even in those cells in which film formation appears most clearly, a very rapid extrusion of protoplasm may occur without any visible fine then form. It is possible that in all cells films form if the protoplasm flows out at a slow enough rate, and that only in those cells in which the reaction is relatively rapid can it ordinarily be observed. Many invertebrate eggs do not show film formation on being crushed. Thus, tests made on the eggs of the mollusks Cumengia and Ilianassa, and on the eggs of the Annelids nereus, Gigiopteris, Aspidiopatra, failed to show film formation, however. 12. The surface precipitation reaction in the Yoechi egg of Arlacia from Heilbrunn 27, Bibon Pid 25 describes film formation on extravates from Ascaris eggs. One of the prettiest objects for study is the egg of the Sorchin Arbacea, when this egg is crushed under a cover slip so that the outer membrane is ruptured. The protoplasm which flows out soon becomes surrounded by a clearly visible film. In Figure 12, this film or membrane can be seen surrounding the film at the outer surface. There are a number of internal films which appear as vacuoles. These internal films or vacuoles vary greatly in number. When an egg is very gradually compressed, so that the protoplasm emerges slowly, Ply may be few or even no vacuoles. With greater speed of emergence, Ply number of vacuoles increases. Apparently, their formation depends in part, at least on whether or not the protoplasm in the interior of the emerging drop comes into contact with Ply surrounding medium. What is the mechanism of the film formation on drops of naked protoplasm? For years, the commonly accepted view has been that the film is the result of an accumulation of surface active substances at the outer surface of the emerging protoplasm. It is certain that protoplasm contains substances which lower surface tension. In accordance with the Jeeves-Thomson rule, these would accumulate at a surface, just as peptone accumulates at a water-air surface and forms a solid film or membrane there. This explanation at first glance seems self-evident, and it has been accepted by practically every authority on cytology or cellular physiology. Until recently, it was never examined experimentally. On the face of it, the comparison of protoplasmic films with the adsorption films which form at the surface of ieptone solutions is not an accurate one. Peptone and other surface active substances become concentrated at the surface of water and air or water and oil, But when protoplasm is cut, there is no surface tension between it and the surrounding watery medium. This follows from the fact that protoplasm itself has water as a dispersion medium. When an aqueous solution is brought into contact with pure water or with another aqueous solution, there is no surface film at the boundary. Hence, if we are to assume that the film which form around emerging droplets of protoplasm is an accumulation of the surface active constituents of the cell, then we must first imagine some sort of surface for these constituents to accumulate. If protoplasm were a gel, we might imagine a surface to exist between it and pure water. But surely it has been abundantly proven that the protoplasm of the sea urchin egg is not a gel, see chapters 5 and 6. And yet in this very instance, film formation is especially easy to observe. The idea that the films which form about naked protoplasm are adsorption films can also be shown by experiment to be untenable. As is well known, adsorption occurs more readily in the cold. With a rise in temperature, surface tension decreases, and there is then less tendency for surface active materials to become concentrated at a surface. On the basis of the old theory, cold, since it favors adsorption, should favor the formation of protoplasmic films. Experiment shows it to have the opposite effect. When sea urchin eggs are crushed at temperatures a degree or two above freezing, usually no film forms about the protoplasm as it emerges. Sometimes an incomplete film makes its appearance if the pressure on the cover slip is very gentle. But it is readily apparent that low temp extemperatures retard rather than favor the process. The films which form about naked protoplasm are not adsorption films. As a droplet of fluid protoplasm streams out of a cell, some sort of a coagulation process must occur at the lie boundary of the droplet to prevent its diffusion throughout telea surrounding medium. The entire process, or processes, which are involved in the formation of a film on the surface of exeozod protoplasm, have recently been called the surface precipitation reaction, nil beyond 27b. Experimental study of the surface precipitation reaction has shown that in various cells, Stentor, Arbacea eggs, Ichthyodarocheus eggs, the presence of calcium is a necessary prerequisite. If Arbacea eggs are washed in isotonic sodium chloride solution for a minute and are then transferred to a second dish of isotonic or crushed, the protoplasm streams out through the sodium result can be obtained if the eggs are crushed in calcium-free seawater. This experiment can be simply performed by adding a small drop of M4 ammonium oxalate solution to a drop of seawater containing eggs. After an interval of a minute or less, the eggs on being crushed showed no surface precipitation reaction. Apparently, a certain amount of calcium is necessary for the reaction to proceed. It should be noted that washing for a minute in isotonic sodium chloride solution or treatment for a minute with oxalated seawater can scarcely remove the calcium from the egg material. Perhaps the calcium within the egg is not in an avalol form. This is a point which will be brought up again. It is probable that in amoeba also, the surface precipitation reaction depends on the presence of calcium. At any rate, one may perhaps so interpret the experiments of Chambers and Rezanov 26. These workers tore amoebae with microdissection needles. When the amoebae were torn in dilute calcium chloride solutions, there was an immediate repair of the torn surface. Such a repair did not occur in dilute sodium chloride solutions. But Chambers and Arbes-Nicoe found also that the repair that they described took place in distilled water, supposedly in the absence of both sodium and calcium. From the effects of that distilled water may free calcium from the interior of the amoeba. In the normal movement of an amoeba, the internal gerotoplasm medium, and form a new film about itself. Perhaps in this normal movement, the surface precipitation reaction plays a part. It is interesting to note that in the case of a marine amoeba patent, 26 found no movement possible in the absence of calcium. Grade 22, CP47, states that the exuding PIA otoplasm of Nutella rapidly forms a film about itself, but that this reaction does not occur when the protoplasm flows out into a dilute Schwach solution. For Haji, the effect of the psoas is to remove calcium. Whatever our interpretation of these experiments of others, it is certain that in sea urchin eggs and in stentor, the surface precipitation reaction cannot take ecstasy in the absence of free. The magnesium ion cannot take the place of the calcium ion. Eggs washed with magnesium chloride solution do not give a surface precipitation reaction. On the other hand, the presence of small quantities of strontium permits the reaction to exorosei. In other words, strontium can replace calcium. Our X3-incremental analysis of the surface precipitation reaction has established the fact that calcium plays an important role in at least two widely different types of protoplasm. Further study may show that the presence of calcium is an essential factor for Tly reaction in all types of living material in which it occurs. In the sea urchin egg, the pigment granules of the egg interior also play a part in the surface precipitation reaction. Figure 12 shows that as the protoplasm of the egg emerges from its containing membrane, the pigment granules disappear. Their color diffuses throughout the entire extravasate. When eggs are crushed in the absence of calcium, there is no surface reaction and no breakdown or disappearance of pigment granules. This observation led to the idea that perhaps the pigment granules were directly involved in the surface precipitation reaction, unlikely as such a participation might seem. The idea was soon tested. Eggs were centrifuged and were then crushed. In those eggs in which the cell membrane ruptured at the heavy end of the egg where pigment granules were present, an excellent surface precipitation reaction occurred. When the rupture was at the opposite hemisphere, there was no reaction in the absence of pigment granules. But the failure of the reaction to take JLS might be due either to the lack of pigment granules or to the lack of the small colorless granules, for these are also absent from the light end of the centrifuged egg. Another test was then made in which eggs were first centrifuged and were then allowed to stand for 45 minutes before being crushed. Within 45 minutes, many of the colorless granules returned to the light pole of the egg, but the pigment granules, for the most part, remained near the heavy end. Under these conditions, when the protoplasm was forced out of the eggs by pressure on the cover slip, no surface precipitation reaction occurred in the absence of pigment granules, even though colorless granules were present. Of the granular constituents of the egg, it is the pigment granules, and these alone, that take part in the reaction. Our analysis so far has shown, 1. That the surface precipitation reaction is retarded or inhibited by low temperatures. 2. That calcium or strontium is essential. 3. That in the sea urchin egg, the pigment granules play a role. In studying any colloidal reaction of protoplasm, it is always of interest to compare it with the reactions which may be observed in test tubes. Most colloids show no specific relation to the calcium ion, but both in the coagulation of blood and the clotting of calcium plays an important part. The fact that blood and milk are biological colloids makes this resemblance more interesting. The surface precipitation reaction also resembles blood coagulation in another respect. Both are retarded or the surface precipitation reaction prevented by cold. Perhaps, too, the breakdown of the pigment granules in the surface precipitation reaction is to be compared to the breakdown of the blood platelets in blood coagulation. In view of these similarities, it was thought worthwhile to study coagulation. Unfortunately, the literature on blood coagulation is in a very confused state. There are dozens of theories, many with distinct terminologies for various known and assumed substances, but this much at least seems clear. There are 2 and perhaps more stages in blood coagulation. The first stage involves calcium and results in the formation of a substance called thrombin. In this stage or previous to it, the blood platelets break down. In the second stage, the thrombin produces a clot, i.e. fibrin, by combining with or acting upon fibrinogen, a protein of the blood plasma. Calcium plays no part in the second stage of the blood coagulation process, and thrombin can act in its absence. Clotted blood contains thrombin, both in the clot and in the serum which emerges. A DMJ of scrum will thaw for clot a fresh sample of blood in Tly absence of calcium. It has been shown that calcium and pigment granules are involved in the surface precipitation reaction in the Arbacia egg. Might it not be possible that there is a still closer correspondence to blood coagulation, until yet the need for coagulation is limited to a primary stage, following which calcium is no longer necessary? Just as the student of QT blood coagulation prepares thrombin from clotted blood, so it might be possible, to prepare a comparable substance from the cells which have undergone surface preoipification. This possibility was easily tested. A thick suspension of Sorchin eggs was shaken up vigorously with splintered glass, so that the eggs were broken up into immemorable fragments. To the broken up eggs, oxalate was added to remove calcium. When a drop of this preparation was added to a drop containing eggs in calcium-free seawater, and the eggs were then broken by pressure, the emerging protoplasm showed a surface precipitation reaction. Thus, the preparation of broken EIP eggs contains a substance which can produce a surface precipitation reaction in the absence of calcium. Although when this substance is not present, calcium is necessary for the reaction. The similarity to blood coagulation is close. In cells, as in blood, there are two stages to the coagulation reaction. In the Arbacia egg, the first stage involves an interaction of pigment granules and calcium. The second stage does not require calcium. Does it require pigment granules? Apparently not. For several tests in which centrifuged eggs were placed in oxalated seawater, plus extract of broken up eggs showed a definite surface precipitation reaction when the protoplasm was made to emerge from the light pool. If these experiments are correct, in the presence of broken up eggs, a surface precipitation reaction can occur in the absence of calcium and in the absence of pigment granules. Thus, the first stage of the surface precipitation reaction seems to consist of a reaction between calcium and pigment granules. The result of this reaction is the formation of a substance which can produce a surface reaction in the absence of calcium and probably also in the absence of pigment granules. It has just been shown that there is a substance in a preparation of broken up eggs which can produce surface films in the absence of calcium. But because of the apparent similarity to the phenomena of blood coagulation, it was proposed to call this substance ovothrombin. The name ovothrombin should be regarded as provisional for two reasons. In the first place, similar substances may very possibly be found in cells other than eggs. If this proves to be the case, cytothrombin might be a better term to use. Secondly, when a true understanding of blood coagulation emerges from the welter of present-day theories, it may be found that the ovothrombin of the sea urchin egg more truly resembles some one of the numerous other substances now suffering from a multiplicity of names than it resembles thrombin itself. But for the present, the name ovothrombin will suffice as a label for the substance which can cause a surface precipitation reaction in the Arbacia egg in the absence of free calcium. In the literature on blood coagulation, there has been much discussion as to whether or not thrombin is an enzyme. In two important respects, ovothrombin does not resemble typical enzyme preparations. It is not destroyed by boiling. Preparations of ovothrombin can be boiled for 10 or 15 minutes without losing their efficiency. If anything, they seem to gain rather than to lose in potency as a result of boiling. Secondly, the Schurich-CV precipitation reaction thrombin diffuses reduv through parchment membranes. In preparing relatively pure preparations of ovothrombin, it was found convenient to boil the broken up eggs and then to place them in a parchment dialyzer. The ovothrombin then passed through the dialyzer. Ovothrombin is surface active, that is to say, it lowers surface tension. This is shown by the fact that in solutions of ovothrombin, the surface film of the solutions shows greater evidence of activity, that is to say, is more efficient in causing a precipitation reaction than is the fluid in the interior. Some preparations of ovothrombin were found to deteriorate after 2 or 3 weeks. It is destroyed by weak solutions of was tried. The addition of 0.2 cc of N100 hakiol to 1 cc of an ovothrombin preparation caused a loss of activity of the latter after an hour and a half. Although ovothrombin can cause a synphase precipitation reaction in the absence of pigment granules, Sometimes the pigment granules do break down when eggs are crushed in oxalated seawater in the presence of ovothrombin. Thus, although you see some experiments, one sees a perfect surface precipitation reaction without loss of pigment on the part of the pigment granules. In other cases, the initiation of a reaction by ovothrombin is accompanied by a fading of the pigment granules, or even by their cometid disappearance. Apparently, the greater the concentration of the ovothrombin, the greater the tendency for the pigment granules to break down, but no accurate experiments were made to settle this point definitely. It seems certain that when ovothrombin initiates the surface precipitation reaction, the breakdown of the pigment granules is not a necessary preliminary, but is more in the nature of an after effect. Not infrequently, the surface precipitation reaction occurs first, and the pigment granules then slowly break down or lose their pigment later. This suggests an interesting possibility. It may be that in the second phase of the surface precipitation reaction, calcium is again set free, and that it is this calcium which causes the breakdown of the pigment granule. If this were true, we might have the following scheme for the stages of the surface precipitation reaction in the Arbacia egg. If this scheme represents the true state of affairs, it is obvious that a small amount of ovothrombin can have a great effect. The reaction is self-propagating. The fact that a living cell contains a substance which resembles thrombin need not be at all surprising. Many authors have held that extracts of various types of cells can have an accelerating effect on the coagulation of mammalian blood. Consult, for example, Schmidt 92. Not only cells of vertebrates, but even cells of plants have been found to yield extracts which hasten the coagulation of the blood of higher animals. It would be interesting to discover whether or not ovothrombin has any effect on the coagulation of the blood. The fluid in the body cavity of a sea urchin clots when it is poured out of the animal. It might be thought that this reaction would show similarities to the surface precipitation reaction of the egg cell, and that perhaps a thrombin could be isolated from sea urchin blood, which would have an effect on the surface reaction of the egg cell. But the clotting of sea urchin blood is a very different phenomenon from the clotting of mammalian blood. It depends almost entirely on the clumping together of the blood cells. Apparently there is no thrombin produced, and the clotting occurs quite independently of the presence of calcium. 1 or 2 crude experiments were tried with human blood serum. It was not found to have any effect on the surface precipitation reaction in the Arbacia egg. It has been shown that the surface precipitation reaction in the Arbacia egg can be separated into 2 stages, in the first of which calcium reacts in some fashion with pigment granules to form ovothrombin. This first stage of the surface precipitation reaction can be subjected to a closer analysis. We can seek to discover what sort of a process takes oxalis when calcium and pigment granules are brought together. It is possible to remove the pigment granules from the cell interior and to study their behavior when they are in a known medium, rather than in the protoplasm. Ordinarily, the pigment granules disappear when they are pressed out of a cell, but in the absence of calcium, they remain intact. One has only to remove calcium from the surrounding medium in order to obtain a suspension of pigment granules. Reagents can be studied, and some observations have been made in this manner. However, this method of study has its disadvantages. Pigment granules are only about a micron in diameter. They are, of course, easy to recognize either within the egg or just after they have been pressed out of an egg. But in a suspension of broken up eggs, there are many small granules about a micron in diameter, and not all of them are pigment granules, nor is the color an absolute criterion that is easy to apply. Not only do the pigment granules fade somewhat, but also other granules apparently take on some of their pigment. It is much safe. You identify the pigment granules just after they have emerged from the egg, when there can be no doubt as to their identification. If eggs are placed in various solutions and are then crushed raw, one can constantly the behavior of the pigment granules under various conditions. Many such experiments have been performed or manned, and some interesting results have been obtained. It is indeed possible to study the reaction between calcium and pigment granule outside of the cell. So far, although numerous observations have been recorded, the work is in a preliminary stage, and no satisfactory interpretation has been arrived at for some of the P6 layer results. We shall therefore present only a BIIFEF BKET of this work in order to indicate the type of results that e'en be obtained. It has been stated that whenever a trace of calcium is present, the pigment granules break down. This is true under the usual conditions. But if eggs are placed in concentrated solutions of NTLVO very considerable amounts of calcium, the pigment granules do not break down. With concentrations of ARC sufficient to produce a disappearance of the pigment granules, but when the concentration of sodium chloride is raised to 1.5 molar, much larger amounts of calcium can be added without any visible effect on the pigment granules. A curve may be allotted which will show the exact concentration of sodium necessary to prevent the action of calcium when it is present in varying amounts. Such curves are very interesting, especially when one notices the variations produced in them by the addition of reagents of one sort or another to the reacting system. Work of this sort is now in progress, of calcium on pigment granules. Ammonium chloride, lithium. When one compares the action of these salts, it can be shown that the various cations are not equally effective. The following is the order of their efficiency in inhibiting the reaction. Between this order is that of the lyotropic series. In studying the hydrolysis of an ester, Arrhenius found that ammonium salts exerted a greater inhibiting influence on the reaction than sodium or anions had a greater inhibiting effect than the less readily absorbed anions. So too, in the Arbacia pigment granule reaction, the sulfocyane ion tends to prevent breakdown of the granule more than does the chloride ion. There is thus a hint that the first stage of the surface precipitation reaction in the Arbacia egg may involve the hydrolysis of an ester, in view of the fact that cytoplasmic granules may very well be surrounded by a lipoid film, EP. 30. The theoretical possibilities are interesting. The products of the reaction might favor the hydrolysis of a fatty substance. It is hardly worthwhile to push the theoretical discussion any further. We must wait for further experimentation. Fortunately, the experiments are very singular to perform. We are really in a position to study a single reaction system of protoxolasm outside of the living cell, but yet in definite relation to one of its morphological constituents. This is indeed a great opportunity. It must not by any chance be thought that the reaction of pigment granule plus calcium is simple. In order to give an idea of the complexities, it may be stated that if eggs are crushed in concentrated calcium chloride solutions, there is no breakdown of pigment granules. This is indeed surprising. However, if the eggs are crushed immediately within 5 seconds after entrance into the calcium solutions, there is a breakdown of pigment granules. Apparently, in the concentrated calcium chloride solutions, or perhaps in any concentrated solutions, there is the formation within the egg of a substance which protects the pigment granules from breakdown. The fact that dilute calcium solutions favor the first stage of the surface precipitation reaction in concentrated solutions prevent it reminds one of the fact that dilute traded solutions retard it. In the preceding pages, we have emphasized the fact that in many respects the surface precipitation reaction of the sorption egg resembles the blood coagulation of higher ant. One such difference lies in the fact that whereas the coagulation of mammalian blood can only occur between fairly narrow limits of hydrogen ion concentration, the cell reaction is rather independent of the pH. Kugelmassen showed that blood coagulated only between pH 5 and pH 8. On the other hand, the surface precipitation reaction can alkaline media. It Tilde-Boe remembered, however, that ovothrombus destroyed by acid. The student of blood faced with the problem of why the blood does not coagulate witly in the vessels, and he has proposed various types of explanation to account for the fact that the blood remains fluid as long as it stays inside the arteries. There is a similar problem involved in the surface precipitation reaction of living coals. Why does it not occur in the cell interior? Within the celi, both calcium and pigment granules are present. One would expect that these would react, that the pigment granules would disappear, and that films or membranes would be formed within the cell. It seems probable that the surface precipitation reaction does not occur in the cell interior because of the absence of free is washed for a single minute in isotonic sodium chloride solution. That this treatment is sufficient to prevent a reaction when the cell is crushed Walk treatment for a minute could hardly remove the calcium from the egg interior. We have reason to believe, therefore, that the calcium of the cell is not in a form available for the surface precipitation reaction. Perhaps it is adsorbed at the surfaces at some of the cell granules. If this were the case, then if we place the eggs in an isotonic solution of calcium length, there would be enough calcium to cause a breakdown of the pigment granules. This is actually what happens. When Arbacia egg cells are immersed in isotonic calcium chloride, at first there is a liquefaction of the cytoplasm, but then following an interval which varies greatly under different conditions, but which usually lasts several hours, there is a breakdown of pigment granules and the egg interior becomes filled with numerous small vacuoles. Beyond much question, these vacuoles are comparable with the vacuoles which can be seen when an egg is crushed under a cover slip, and which are shown in figure 12. The wall of the vacuoles is doubtless the same sort of a film as the film which is produced in the surface precipitation reaction. By introducing additional calcium into the egg, we have thus produced a surface precipitation reaction throughout the cell. Such a cell full of tiny vacuoles and with its pigment granules broken down presents a picture which since the studies of Loeb has usually been referred to as pale cytolysis. If it be true that cytolysis in the Sorchin egg is to be identified with a surface precipitation reaction throughout the cell, this is a very important conclusion, for it enables us to investigate the problem of such cytolysis from a new angle. This subject will be taken up in the next chapter, Chapter 15, a specific colloid chemical reaction peculiar to living organisms. Peculiar to living organisms. In this chapter, an attempt will be made to bring together information concerning a reaction of the limb-gene protoplasm, which has been studied by botanists, zoologists, physiologists, and pathologists. In its different forms, it has been called a host of names at one time or another. And as a result, workers in various fields have usually failed to recognize the extremely general nature of a phenomenon of wikiai. They have studied only a single aspect. If, in the pages that follow, it can be demonstrated that there is one essential reaction which is fundamentally the same for many, and perhaps all T3 eggs of protoplasm, a great deal will have been accomplished. Often, what is difficult or impossible to understand for a given type of cell is relatively simple for another. By bringing together all the information from various branches of the biological field, much can be gained. The originator of the protoplasmic concept, Heilbrunn, was well aware of the tendency of protoplasm to undergo vacuolization. And indeed, he regarded this as one of its essential, if not its most essential characteristic. In his first paper on protoplasm, or sarcode, published in 1835, Lai says, "Que mi alla proprio six la plus d'étrange du barcode chest à production spontanée sphérique. Later, l'on petit cavite sphérique." In 1841, he writes that when he first defined the concept of sarcode, he felt that the peculiar property which living substance possessed of filling itself with watery vacuoles was the very property which distinguished it most clearly from inanimate substances such as gelatin, mucus, or albumin. Since Heilbrunn, no one has so strongly emphasized the vacuole-forming capacity of protoplasm, but there have been no lack of observations. Some of these we shall now proceed to consider. If a living cell is placed in a hypotonic solution or in distilled knowledge, but it does not seem to be so generally known that when a cell is placed in a sufficiently dilute solution or in water, vacuoles appear in the protoplasm. As a matter of fact, this phenomenon was novel long before our present understanding of the osmotic behavior of a cell was arrived at. As it became increasingly evident that the laws of osmotic pressure constituted a satisfactory explanation of the expansion of a cell in water and hypotonic solutions, the older literature regarding vacuole formation was forgotten. The botanists of the middle of the 19th century seem to have regarded the entrance of water into a cell as primarily due to the formation of watery vacuoles double within the protoplasm. This view is hardly feasible now, but it is nevertheless of interest to know that there are numerous observations which show that water does cause vacuole formation in plant protoplasm. Who was the first to describe this phenomenon is not certain. In 1867, Hoymeister, in his book on plant cytology, discusses the subject as though it were rather old and the facts well known. He says, Die Entstehung und Ausbildung von Vakuolen ist direkt wie Beobachten an Alien. Von der graphieren Dichtigkeit der peripherischen Schicht abgesehen. Homogenen Protoplasmamassen, welche in Wasser gelangen. Then he goes on to cite numerous examples, and he attempts a theoretical explanation of why entering water should cause vacuole formation in protoplasm. Wieser-Gekiu, in studying yeast cells, noted that both when rapidly, by placing the cells in concentrated solutions, vacuoles apex reared in the protoplasm. The fact that either a gain or a loss of water can cause vacuole formation is important. Compare also Custer-Tate. Some years later, Schwartz, 87, published some descript FC yawns from his monographs. It shows a young cell from the root tip of a pea seedling. Following treatment with water, the surface protoplasmic layer has become filled with small vacuoles. Schwartz also describes vacuole formation in the nuclei of some cells following exposure to distilled water. And there is, moreover, a vacuolization of chloroplasts. Doubtless a thorough search of the botanical literature would bring to light many other descriptions of vacuole formation following treatment with distilled water. However, it is hardly necessary to multiply references. The fact is clear that many other agencies also cause vacuoles to appear in the protoplasm of plant cells. Unfortunately, no one seems to have been especially interested in the process of vacuole formation, so that in going through the literature, one has to depend on sporadic observations made by various authors in the course of investigations on one subject or another. Apparently, fat solvents act like distilled water on plant protoplasm. Nadson and Meiseltoy described the formation of vacuoles in onion epidermis cells following treatment with chloroform. Both acids and alkalis cause a vacuolization of plant erythrocytosis. This was very carefully described by Q. Kilme Sem 5. W studied a number of different types of material. See also Duin 5 and compare HAP. When an electric current is sent to Lero plant tissue, vacuoles often appear in the protoplasm of the cells. This phenomenon has been described by Velton 7b and by Clem 95. Radium radiations also cause a vacuolization of plant protoplasm when plant cells are subjected to pressure or to mechanical injury. Vacuoles appear in them. Velton 76b states, bei starker Einwirkung des elektrischen Stroms, ebenso bei Druckwirkungen entstehen in den verschiedensten Zellsien, wie dies bereits bekannt vacuolen. Unfortunately, he gives no references to the older literature. Nemec 1b describes the formation of vacuoles in the cells of onion roots. When a root is cut or injured, vacuoles appear in the cells in the neighborhood of the cut and Fig 13. Vacuoles in the proto LFSDN of a root cell which has been exposed to distilled water. There is a wave of vacuole formation which proceeds in all directions from the injured surface. Heilbrunn 21 describes vacuolization of slime mold protoplasm following mechanical injury. Guidero 26b states that numerous vacuoles appear in onion epidermis cells as a result of mechanical coagulation. Many authors have described vacuolization of plant protoplasm following exposure to high temperatures. These include Georgievich 10, Hartmann 18, 19, Wasserman 21a and b, Yamaha 27b. Practically all of the work has been done on fixed material. Nake 4 Eli 55 described the formation of vacuoles in degenerating plant protoplasm. From our brief survey, it is apparent that distilled water, fat solvents, at any rate chloroform, acids and alkalis, mechanical injury, heat, and the electric current all cause the appearance of vacuoles in various types of plant cells. The literature on animal cells is more complicated, and we shall have to approach it from a number of different angles. In the first place, the observations on protozoan protoplasm are much like those which have been made for plant cells. Schulze 63 describes the formation of vacuoles in the protoplasm of Foraminifera treated with distilled water. A similar observation has recently been made by Lepisokin 25A. Passage of an electric current causes vacuoles to appear in amoeba for Warren96b, and in paramecium, Wallengren 3, Statkowicz 6, radium treatment produces vacuolization in amoeba, Zwelzer and Philip 25. Lack of food may induce vacuole formation in protozoa, Wallengren 2. As a matter of fact, vacuoles frequently appear in protozoan protoplasm under natural conditions. This is very apparent if one glances through Butchley's monograph on the protozoa 8289. Interesting figures of the extent to which this vacuolization may go are shown in a Ceoper by Speak 24. There have been occasional descriptions of vacuole formation in blood cells. FBOM on 84 frequently refers to the appearance of vacuoles in the blood cells of crabs. Boettcher 66 describes the production of vacuoles in vertebrate blood cells treated with distilled water and with chloroform vapor. One or two other instances of vacuoles in blood cells will be mentioned later. Vacuolization phenomena in the tissue cells of vertebrates have not infrequently been studied by pathologists. GA literature, a specific colloid, GAP alkyl reaction, etc. 237 is not always easy to interpret. This is in IDART due to the fact that most of the work has been done on section material, in part to the variety of terms used by different workers. V.M. Chow, see Vibcha 70 Warren, coined the expression cloudy swelling, Treibschwellen. And since Virchow's time, this term has been a favorite one in the pathological literature. A cell which shows cloudy swelling is larger and consequently paler, and it has in its protoplasm granules which have evidently been precipitated there. Apparently in animal cells, as in plant cells, see Clem 95, there are two TJPs of degenerative change which may occur separately or together. One is the precipitation of new granules, W do thin the cytoplasm. The other is the formation of vacuoles there. Perhaps if the term cloudy swelling is to be specific, it should be restricted to those cases in which new granules appear in the cytoplasm. This appears to be in accordance with the original idea of Virchow. Some recent authors, however, identify cloudy swelling with vacuole formation, see, for example, Pumjezritsu 26a. The appearance of new granules in the protoplasm, in many cases at least, is due to the precipitation of protein material, and we have already had occasion to note instances in which this undoubtedly occurs. It may be that such precipitation may later involve vacuole formation. This we can at present not decide. It is at least certain that in degenerating cells, vacuoles very frequently 3-pair the protoplasm. Such vacuolization was described by Galeotti 95 for the kidney cells of poison salamanders. The most enthusiastic observer of vacuole formation was undoubtedly Albrecht. He published numerous papers between 1898 and 1907, papers which are richer in ideas and interpretations than in actual observations. An understanding of Albrecht's ideas is perhaps best obtained from his general review written in 1907. Albrecht found that when kidney cells were treated with water, numerous vacuoles appeared in the protoplasm. He described the same phenomenon for various other cells, including sea urchin eggs. Albrecht seems to have regarded protoplasm as essentially a mixture of two fluids. Under certain conditions, one fluid might separate from the other, just as droplets of phenol separate out from a mixture of phenol and water. Because of this supposed resemblance, Albrecht always refers to vacuole formation in protoplasm as Tropfige-Entimischung. Albrecht apparently never attempted to identify the fluid VH formed in Nodiac-Cocois phase in his imaginary system. And he never showed that the vacuoles contained anything but water. In some cells, the vacuoles must surely consist of water, for the volume of the vacuoles is greater than the entire volume of the cell before vacuolization. This indeed is true of the sea urchin egg studied by Albrecht. How such a cell could produce a larger volume than its own of non-aqueous fluid, when 3/4 of its original volume was water, is indeed a mystery. It is surprising, therefore, that the term Tropfige-Entmischung has received as serious consideration as it has. When cells are studied in tissue culture, degeneration is generally accompanied by the appearance of vacuoles in the cytoplasm. This has been noted by a number of investigators. Lewis 19 states that this vacuolization of the cytoplasm is one of the most common modes of CE degeneration and death in tissue cultures. Other references to vacuolization in tissue cells are the following: Lewis and Lewis, 15 Burrows, and Neyman 7, Lewis Pa B. Lewis, and Lewis 24 BT Gem, Jansu 27, and Luna 7. However, vacuoles described by Luna are believed by Lewis 19 to be mitochondria vesicles rather than degeneration vesicles. An observation of Lewis and Lewis 15 may have importance. They find that in many cells there is often a coincident change in the shape of the mitochondria, until in cells which contain more vacuoles within the cytoplasm, the mitochondria are no longer in the shape of rods and threads, but then appear as small granules. Perhaps the appearance of the vacuoles is associated with a decrease in the amount of the formed elements, and especially the mitochondria of the protoplasm. When vacuole appear in kidney calls, certain rodlets characteristic of these cells disappear, Albrecht 0. And in sea urchin eggs, as will be pointed out later, vacuole formation is always accompanied either by a disappearance of definite granular elements in the protoplasm, or by a loss of substance from these elements. And the granules which disappear, the pigment granules, are perhaps homologous with mitochondria. CP20. It is in sea urchin eggs that the process of vacuole formation has received the most attention. Kilbright 99 was perhaps the first to notice it in these eggs, and in his papers he often refers to the process in these cells and compares it with similar vacuole formations he observed in other types of living material. But a specific colloid chemical reactions or etc. Our knowledge of vacuole formation in sea urchin egg double is due mostly to Loeb. His attention seems to have been first called to it in 1904. In that year, Loeb 4, Lai describes what he calls a cytolysis of the CNRChin egg. The cytolysed egg loses its pigment and becomes converted into a pale shadow, at the same time increasing greatly in volume. A similar change had probably been seen by O and Arth. Here at WEO 87, for in their paper, Loiry referred to the characteristic glassy appearance of eggs treated with a toxic dose of chloroform. Loeb Eva is the first to use the term cytolysis for marine eggs. Tai seems to have borrowed it from the literature of immunology, wherein it has had a long history for a survey of the older immunological literature, Skolnotsy too. But Loeb does not use the term in exactly the same sense in which it is used by the immunologists. From its derivation, cytolysis moans, a solution of cells, and in a thiacal or perhaps WH2, say a complete cytolysis, the cells dissolve and dissipate. In modern immunological literature, cytolysis donnelly such IgA solution or disappearance of cells, see for example, lac BMK 27. In the sea urchin egg, according to Loeb, cytolysis involves increase in volume and loss of pigment. Later, he adheres to this gyrocytolysis is pale cytolysis, in contrast to a dark cytolysis in which the egg does not increase in volume. In this dark cytolysis, the egg breaks up into a mass of small dry pieces. As a matter of fact, in both types of cytolysis, as distinguished or vacuole formation throughout Tilugigi. In Loeb's book published in 1913, he shows no drop cues or vacuolyses in an egg, cytolyzed in distilled water, figure 58. But vacacia is clearly shown in the case of an egg cytolysis like Altaichi. Figure 51. It is, however, true that vacuoles do appear in SGG cytolyzed in distilled water, compare Album 99. It is an unfortunate circumstance that no OUI has visa studied from a morphological standpoint, Loeb chose to call cytolysis. From many unpublished observations on cytolyzed eggs, it seems certain that in every case hydroxates or vacuoles appear in the protoplasm. It is an oxyem question whether or not it is advisable to retain the term cytolysis as applied to sea urchin eggs, but it is at Jekwai certain that in these eggs there is a reaction in which numerous vacuoles appear in the cytoplasm. This vacuole formation is always accompanied by a diffusion of each euaginate from the egg into the surrounding medium in the eaves of those eggs which have a visible pigment. In the Aracia egg, the pigment granules may break down completely, or they merely lose some of their pigment and still remain distinguishable within the egg. In what Loeb called paleocytolysis, the pigment granules completely disappear. In his dark cytolysis, apparently they do not, and in this latter form of reaction, the egg does not undergo a great increase in volume. In Loeb's studies on what he called cytolysis of sea urchin eggs, he was influenced by the literature on hemolysis and especially by the papers of Koppe, see Koppe 3. Koppe had found, and his knowledge is really much older than Koppe, that hemolysis of red blood cells is caused by water, by heat, by acids, by alkalis, and by fat solvents. Loeb and his student von Nafelens studied the effect of these various agents on sea urchin eggs, and it is Loeb's opinion that all of them cause a cytolysis of the cortical layer of the egg. Loeb's ideas on the subject of this cortical cytolysis were always a little vague, but he was certain that any agent which produced activation or the beginnings of development in sea urchin eggs also occasioned a cortical cytolysis. On longer exposure to these parthenogenetic agents, there was a complete cytolysis. From von Nafelens's work and from Loeb's book 13, it is clear that water, heat, fat solvents, and also the well-known hemolytic agent saponin all cause cytolysis of the sea urchin egg. According to von Nafelens, 8 acids have no such action. On the other hand, Regueixi-Villais and Rose 23 describe a cytolysis in the presence of mineral acid. The difference in results may be due to a difference in material. From von Nafelens' account, it is not clear whether alkalis cause cytolysis or not. Certain it is that most of the agents that typically cause hemolysis of red blood cells do actually cause vacuole formation and loss of PGC from sea urchin eggs. It should be pointed out that when sea urchin eggs are left in dishes containing seawater, they undergo a degeneration process, or a cytolysis, which also involves a vacuolization, and a loss of pigment. It is apparent, therefore, that in sea urchin eggs there is a reaction which resembles, on the one hand, the vacuolization phenomena as observed in plant cells and in tissue culture cells, and on the other hand is related to the well-known hemolysis process of red blood cells. Just what does this hemolysis consist of? When a red blood cell becomes hemolyzed, pigment escapes from it. This appears to be the essential criterion of hemolysis, although in many instances escape of pigment is accompanied by marked increase in the volume of the red blood cell. It is then said to become a ghost. Noef 9 defines hemolysis as a destruction or deterioration of the red blood cells, and he says that when it is complete, the cell completely disappears. Our knowledge of hemolysis is very old. Milne Elwab's 58 cites various references to older literature. The hemolysis of red blood cells in distilled water was described by Moulin 3082, and in his book published in 1835, Tillich also described other types of hemolysis. In his studies of the physiology of blood, Muller was apparently not very conversant with the older work of Hewson. He rarely refers to this author, and indeed he was severely criticized by one of his contemporaries for this failure to make the Roper reference. In a posthumous work of Hewson 1785, CP 14, there is a clear description of hemolysis extraduti by water, ubi multum aquae addendum est, vesicula sensum tenuir magis pellucida rediter, et tandem dibsovel vetur. When much water is added, the vesicle, that is to say, the blood cell gradually becomes thinner and more pellucid, and at last dissolves. Hewson also describes hemolysis in dilute solutions of mineral acids. It is not improbable that there are even older descriptions than those of Hewson, partly because of the importance of the subject and partly also because it is a relatively simple matter to make observations on the escape of pigment from red blood cells. Many authors have studied hemolysis. The literature is now vast. Bohl in 1909 cites well over 200 titles, and he limited himself to the literature of the 20 years preceding. As a matter of fact, practically all of his references are to J. Sears P. N. Blish between 1908. Since 1908, there has been no decline in the output of papers on hemolysis. The bacteriologist, the clinician, the physiologist, even the chemist has attempted to contribute to the solution of the problem of the destruction of the red blood cell. All of his literature does not concern us. The bacteriologist is frequently interested in hemolysis merely because it is a convenient index of the presence or the properties of materials which he is unable to define chemically. Our primary aim is to show first that certain types of hemolysis involve a species of cell reaction which is found in many cells other than blood cells. And it is our hope that starting from this line of approach, we may be able to throw light on some of the mysteries of hemolysis, especially insofar as they aren't related to the colloid chemistry of the protoplasm of the blood cell. In general, the student of physiology studies only end manifestations. Either of our example, he measures carbon dioxide produced by cells or organisms, or the electric current generated in the corso of one vital phenomenon or another. But it is the handicap of the science that in the main, the physiologist has little conception of the actual reactions or changes which are responsible for the end result. He is able to measure. In no field of cellular physiology is this more true than in the study of hemolysis. Whereas many cells are favorable for microscopic study, the red blood cell is extremely small, and it shows little under the microscope. The study of hemolysis has proceeded almost purely along physiological lines. This is a severe handicap in larger cells in which react and is similar to liquidnolysis go on. Physiological study can better 1-0 combined with morphological study. Usual type of hemolysis of the red blood cell finds its exact counterpart in the process of cytolysis or vacuole formation as it occurs in the egg of the sea urchin Aracia. In both sorchine eggs and blood cells, there is typically an increase in volume accompanied by a loss of pigment. And as Loic B first pointed out, the regions which cause hemolysis of blood cells regularly cause what he termed cytolysis of sea urchin eggs. Distilled water, saponin, bile salts, fat solvents, soaps on the one hand produce hemolysis, and on the other cause cytolysis of Archaca eggs. It might be thought that there is a difference in the fact that vacuoles appear within the cytoplasm of eggs, whereas one clocks not ordinarily think of them as being formed during hemolysis. Against this argument, it can both stated first that vacuoles are not always very easy to sec if UI in cells as largo as sea urchin eggs. Loeb often failed to notice them when they double ERC present, compare P239. In small blood calls, they would be still harder to recognize. Actually, some of the older anatomists in their drawings of lemolized blood cells do show structures within the stroma which might very well be interpreted. A specific colloid chemical reaction, etc., 243, as vacuoles. In the larger blood COS of amphibia, vacuoles can more really high be observed. In Vo 5 Ebnip's revision of Kolker's Handbuch, von Ebner 2p. 728, he notes the appearance of vacuoles and such cells following treatment with ammonium chloride. This reagent is a well-known hemolytic agent. Vox E. Bixer also figures human blood cells with what he calls vacuole-like spots. Pointer 66 describes the appearance of vesicles and vacuole MLV structures in various THEDs of blood cells when they were treated with chloroform vapor and with distilled water, both typical hemolytic agents. In some cases, he apparently mistook vacuoles for nuclei of mammalian red blood cells. Doubtless more thorough search of the literature would reveal other instances in which authors have described vacuole formation following hemolysis. Perhaps it would be simpler to make a few studies of the hemolyzed blood cells of salamanders. It is rather surprising that in recent years these cells have been so little used in studies of hemolysis. But as a matter of fact, additional evidence is scarcely necessary. From the previous discussion, it must be evident that hemolysis of blood cells and what Loeb called cytolysis of egg cells have much in common. It must not be supposed, however, that every reagent which causes a loss of pigment of blood cells would cause a typical cytolysis of the sea urchin egg. Some reagents cause loss of pigment in arhata eggs without apparently causing any pronounced vacuolization. This is probably due to the presence of complicating factors. It is perhaps easier to assume that hemolysis and cytolysis of egg cells are related phenomena than to assume that cytolysis of egg cells corresponds to the vacuole formations, which occur in plant cells. One reason for this is the fact that the evidence that lies at our disposal is very fragmentary. If one considers the action of a reagent like distilled water, it is logical to assume that the vacuoles it produces in plant cells are similar in origin to the vacuoles it produces in protozoa and in sea urchin eggs. It might, however, be urged that some of the agents which cause vacuole formation in plant protoplasm have no such effect on animal protoplasm. Thus, in the botanical literature, mechanical injury has frequently been described as producing small vacuoles in plant protoplasm. At first sight, it might be thought that this did not occur in animal cells. For in recent years, at least, there has been almost no literature on the subject. But in the last chapter, it was shown that when sea and urchin eggs are torn, many vacuoles appear in the cytoplasm. See Figure 12. And it may also, he remembered that Dugidin 1835 described vacuole formation in protozoa and protoplasm following injury. It is interesting to compare the action of the electric current on various types of living substance. In both plant and protozoa and protoplasm, the electric current regularly causes vacuole formation. In Arbacea eggs, it causes cytolysis, Lillian Cattell 25. In red blood cells, as is well known, it causes hemolysis. Compare, for example, Rolette 71. It should be noted that in none of these instances was the experimental procedure such that the effects of the electric current could possibly have been due to the production of electrolytic decomposition products. It might be argued that one of the characteristics of hemolysis and cytolysis is an escape of pigment, and that this has not been observed in plant material. It is true that this phase of the subject has received very little attention on the part of botanists, but Boas 2021 has shown that saponin causes an escape of pigment from some plant cells. And Zepekel brought together many instances in which fat solvents cause the escape of various substances from plant cells. But perhaps even in those cases in which pigment is not seen to be located in visible granules, it is bound to SID microns, that is to say, to ultramicroscopic particles. Our general conclusion is that in all types of protoplasm, there is a definite type of response to such agents, as distilled this response is typically characterized by the appearance of numerous small vacuoles within the protoplasm. Frequently, though not always, it is accompanied by an increase in the volume of the cell. When pigment is present in the cell, this always escapes. The reaction that we have described is definite enough to deserve a name. The term tropphige Entmischung used, L.Y. Albrecht, for certain aspects of the reaction cannot very well be used, for it carries with it a highly improbable interpretation of the constitution of the pigotopal as amyl. The term cytolysis has certain advantages in spite of the fact that in its original use in the science of immunology, it carried various other meanings. But cytolysis, as it is now used, seems to imply destruction, and it is very doubtful if in all cases of vacuole formation, especially in plant protoplasm, lethal changes are involved. Velton 76b clearly states that the vacuoles once formed may disappear again. There can be no question but that a new name is needed, but it will probably be best to wait until the reaction is better understood and more completely and thoroughly described before a distinctive name is given to it. We are now in a position to inquire into the mechanism of the reaction we have been considering. In no case can the reaction be studied more clearly than in the egg of the sea urchin. At any rate, in no other type of protoplasm aside from blood cells has there been any study of the mechanism of the reaction. In the remaining pages of this chapter, therefore, an atomothrete will be made to outline some of the factors on which the reaction in the sea urchin egg depends. In the course of the discussion, occasional reference will be made to the process of hemolysis in blood cells. In spite of the fact that Loeb many times emphasized the importance of the so-called cytolysis of sea urchin eggs, he made little effort to explain the mechanism of the process. In pages 188 to 190 of his book, T3, he expresses some vague general ideas regarding the possible nature of cytolysis. Saponin and benzene, he states, dissolve the corian or jelly of musk and amlet eggs. He then cites Vogt-Canel-Lenz's views to the effect that cytolysis is due primarily to the liquefaction of lipids. And he makes the following quotation from von Affoley-Benz: "The mechanism of cytolysis consists in the liquefaction of the lipids, and thereupon the lipoid-free protein swells, or is dissolved by taking up Loeb. Thus apparently believes that cytolysis in sea urchin eggs S is primarily a liquefaction. F. The protoplasm, this it most certainly is not, for viscosity tests indicate beyond. The question of a doubt that during cytolysis, the viscosity of the egg protoplasm increases enormously. Mubtsevina and B. published 2 papers on the mechanism of cytolysis in sea urchin eggs. From a study of the temperature coefficient of hot water cytolysis, he voices the opinion that cytolysis is due to a monomolecular chemical reaction. G, in view of the fact that most chemical reactions in colloidal media behave as monomolecular reactions, this is equivalent to Sajing cytolysis is due to a chemical reaction. Very possibly it is, but unless we know which substances are involved in the reaction, we have scarcely made much progress toward a solution of our problem. In the next few pages, we shall seek to interpret at least certain aspects of what Loeb called cytolysis in sea urchin eggs. This discussion cannot be very satisfactory, for it is based for the most part on experiments now in progress and still unpublished. As has already been stated, in the Arbacea egg, treatment with various reagents is followed by the appearance of numerous vacuoles within the protoplasm, so that the protoplasm eventually becomes a mass of such vacuoles. At the same time, pigment is given off by the eggs. Frequently, the egg volume increases, but this volume increase does not always occur. And when it does, it is probably rather a cause of the reactions that follow than a result. This will be more clear later. The first point that should be clearly recognized is that the escape of pigment from Arbacea eggs does not depend on the increased permeability of the egg membrane, or rather it is very little dependent on such permeability change. This follows immediately from the fact that the egg membrane or plasma membrane of the Arbacea egg is at all times freely permeable to the egg pigment. Anyone who has worked with Arbacea eggs knows that the water standing over them is continually colored by pigment escaping from them. Moreover, the pigment within the eggs is not for the most part free in the cytoplasm, but is definitely concentrated in easily visible pigment granules, which are shown both in Figure 12. Frequently, these pigment granules can be seen to break down, and then the pigment is released both to the rest of the cytoplasm and to the medium surrounding the eggs. As a matter of fact, the reaction we are considering involves a visible change in these pigment granules, and in most instances, they disappear completely, with the result that their pigment is dispersed. It is probable that in blood cells, too, the escape of pigment is not so much due to a change in variability of the membrane of the blood corpuscle, as it was the fashion to believe around 1900, but rather more to some change within the corpuscle that sets the hemoglobin free. This follows in part from the fact that the hemoglobin can scarcely be contained in solution within the corpuscle, for the corpuscle contains more hemoglobin than it would be possible for it to have in a state of solution. Apparently, a specific colloid chemical key action, etc. 247 among students of hemolysis. There is a growing tendency to attribute the retention of hemoglobin within the blood cell more to the binding of the pigment by substances of the cell protoplasm and less to the impermeability of the cell membrane. See, for example, MON 25. Our second point, and this is really the crux of the entire argument, is that the formation of vacuoles throughout the exotoplasm of the sea urchin egg depends on the same reaction that we have called the surface hydroprecipitation reaction, and which we have considered in the last chapter. When an arbata egg is crushed or torn, so that the eutrotoxelasm exudes, it may rarely form only a single fine or membrane about its free surface. much more usually as jovially if the protoplasm exudes rapidly and there is plenty of calcium in the surrounding medium. The film or membrane formation is not confined to the outer boundary of the emerging droplet. As was pointed out in the last chapter, other films are formed within the main mass of the exuding protoplasm, and these films are in the form of vacuoles. Such vacuoles are clearly shown in Figure 12. That they are the exact equivalent of the vacuoles which appear in so-called cytolized eggs is certain. From eggs which show one or two or a few vacuoles, there is every gradation to those which are filled through their entire volume with vacuoles, and are thus, in the common sense of the term, cytolid. In order to produce such cytolysis, it is only necessary to crush the eggs very violently. That the reaction which produces vacuoles is exactly the same as that which causes a membrane to form on the surface of the naked protoplasm follows also from the fact that the same substances play a part in the reaction. Cytolysis, as generally described, involves a release of pigment from the egg, and this ends, of course, on a breakdown, or at least a partial breakdown of the pigment granules. Thus, for vacuole formation in the cell interior, just as for the surface precipitatory reaction at the border of an emerging Droxstat, the pigment granules are essential. Secondly, calcium is also a prerequisite for the appearance of vacuoles. In eggs crushed in the absence of calcium, there is an absence of the surface precipitatio.N reaction, and also a complete absence of vacuole formation in the exuding protoplasm. We can therefore regard our second point as established. Vacuole formation, as it occurs in the arbata egg in what has been called cytolysis, is really an internal surface precipitation reaction. In the last chapter, it was shown that whenever the pigment granules of the Aracia egg were placed in the presence of free, which was the first of a series of reactions comprising the surface precipitation reaction. Apparently, the reaction between pigment granule and calcium can occur in the egg interior as well as in the gerotoplasm pressed out of the egg. If this is the case, then it is only necessary to increase the concentration of free concentration within the cell beyond a certain point in order to produce a vacuole formation throughout the entire egg, or in other words, a cytolysis. If we make the assumption that the calcium within the egg is for the most part bound with some fatty or lipoid substance, then we can offer an explanation which will fit practically all of the known facts. This assumption is certainly a logical one to make. Analyses of protoplasm show a high content of salts, and it is certain that the ions of these salts are in large measure combined with some constituent or constituents of the protoplasm, CP. 25. They can be combined either with proteins or lipoids, or both. For the sake of our argument, we will assume that the calcium is for the most part combined with lipoid. And furthermore, the calcium is set free when the lipoid is dissolved. There is evidence that the surfaces of the protoplasmic granules have a lipoid film. CP30. If the calcium were absorbed at such a film, solution of lipoid would certainly set calcium ions free. If our reasoning or our assumption is correct, the free calcium ion concentration of a cell may be increased in the following one. By a rupture of the cell membrane, 2. By a decrease in the volume of the cell, as for oxalate, when the cell is placed in a hypertonic solution. 3. By long exposure to isotonic solutions of calcium salts. 4. By any treatment which will tend to cause a solution of protoplasmic lipoids. Such treatments include B. solvents, C. decrease in the salt content of the cell, i.e. increase in the cell volume, a specific colloid chemical Fe action, etc. 249. 4C is perhaps not so readily understandable, but it must be remembered that Hanstein-Sinke 22 showed that distilled water caused a solution of protoplasmic lipoids of plant cells. Ruzernstoyan 23 has moreover presented evidence to show that lipoids in the sea urchin egg are dissolved. YDN, the eggs are placed in hypotonic solutions. Compare also P200. We shall now proceed to show that every one of the methods of treatment outlined in the above scheme does actually cause pigment granule breakdown and vacuole formation in Aracia eggs. We have already considered the case in which there is a rupture of the cell membrane. When cells are placed in hypertonic solutions, the decrease in volume is, of course, accompanied by a corresponding increase in the calcium concentration of the interiors. In sea urchin eggs, strong hypertonic solutions cause what Loeb called a dark cytolysis. In this dark cytolysis, much pigment is set free from the pigment granules and numerous vacuoles or vesicles form within the cytoplasm. It may be remembered that WCA69 also described the formation of vacuoles in yeast cells following treatment with hypertonic solutions. In the literature, there is no reference to cytolysis following long exposure to calcium solutions. However, in unpublished experiments, it has frequently been found, as stated in the last chapter, that after long exposure to an isotonic calcium chloride solution, pigment granule breakdown occurs generally throughout the egg. Apparently, when eggs are placed in such solutions of bound or adsorbed, so that there is no reaction between it and the pigment granules. But eventually the limit is reached. No more free calcium increases beyond a certain point, and an internal surface precipitation reaction occurs. Apparently, most cytolytic agents act first by causing a solution of fats. Von Knöfel-Lentz 8 believed that all cytolytic agents acted by dissolving fats, but very obviously, neither isotonic calcium chloride solutions nor hypertonic solutions can be thought of as having a fat solvent action. Heat cytolysis was described by V. Knöfel-Lentz. Unpublished observations indicate that in this form of cytolysis, at least in the sea urchin egg, there is not a complete breakdown of pigment granules, but only a loss of pigment from them. Von Knöfel-Lentz also described the action of fat solvents. As a matter of fact, in the action of fat solvents, there are really two factors to be considered. Probably the fat solvent acts directly on the lipoids of the cell, at least in most cases, but it must be remembered that the cytolysis in fat solvents is always accompanied or preceded by an increase in the volume of the cell. This is apparently due to a decrease in the surface tension of the membrane of the cell, see below. The increase in volume produced by fat solvents could in itself cause a solution of lipoids. There are in general three types of agents which cause increase in volume of sea urchin eggs. In the first place, high GK tonic solutions or distilled water have an effect on the cell readily understandable from an osmotic standpoint. Secondly, substances which have a marked effect on surface tension by lowering the surface tension of the egg membrane can cause a great increase in the volume of the cell. This phenomenon is not confined to sea urchin eggs, but occurs in many types of protoplasm. The cell membrane is the seat of various forces, osmotic forces, as well as others, and some of these press inward, while others press outward. Normally, there is, of course, equilibrium. The surface tension of the surfaces at the periphery of the cell exerts a force which presses inward. When this force is diminished, the equilibrium is upset and the volume of the cell increases, either until coagulation sets in or until the increase in inward pressure due to the distension of the elastic cell membrane compensates for the lowering of surface tension. Substances which lower surface tension certainly cause an increase in volume of free cells generally. But if our conception of the cell membrane is correct, if it is an elastic membrane stretched by the osmotic forces from within, it is obvious that a change in the elastic physiological properties of the cell membrane can also cause a change in the volume of the cell. Many substances cause visible changes in the cell membrane of the Aracia egg, and these visible changes are doubtless associated with changes in the elastic properties of the membrane. If the membrane becomes changed so that it takes less force to distend it, then obviously the cell will increase in volume. G. This is apparently the explanation of the increase in volume which occurs in isotonic solution of sodium chloride, and a specific colloid chem-cal reaction, etc. 251 various other salts in an isotonic solutions of sodium chloride. The egg remains at its normal size as long as there is a small quantity of seawater or calcium in the surrounding medium. If this is washed away, then the egg membrane can be seen to swell and it also becomes sticky. These changes are accompanied by an increase in the volume of the egg, and it goes through a typical process of cytolysis. Such cytolysis in isotonic solutions of sodium chloride and other salts is well known through the observations of H.S. Lilly. For references, see Lilly T22 effect and sodium chloride. They cause a more pronounced change in the properties of the cell membrane. Increase in volume, no matter how it is caused, always results in release of pigment and vacuole formation in the Searchen egg. The release of pigment is in no sense the result of a change in the permeability of the cell or plasma membrane. Very possibly such a change in permeability may occur, but the escape of the related to visible changes in the pigment granules, for these can actually be seen to give you their pigment. The release of pigment from Aracia eggs eptos to isotonic solutions of sodium chloride, or sodium sulfoxide, or ammonium chloride does not depend directly on the presence of these ions at all, but merely on the fact that in solutions of these salts, the eggs increase in volume. Sodium iodide and sodium sulfoxide have no effect on the release of pigment from the pigment granules. This can be shown by crushing eggs in solutions of under these conditions. The pigment granules remain perfectly intact and do not lose pigment when they emerge from the egg. Likewise, the effect of saponin is due not to an action on the pigment granules, but to some secondary effect. Pigment granules exposed to saponin show no breakdown or release of pigment. On the other hand, when entire eggs are treated with saponin, the pigment granules in the interior do break down and disappear. The effect is probably to be attributed to the great increase in volume of the eggs in saponin solutions. A small amount of saponin M, the surrounding medium, certainly lowers the surface tension of the egg membrane. Whether this is its only effect is not certain. For the purposes of our discussion, it is enough to know that the egg undergoes a great increase in volume, and regarding this fact there can be no doubt. It is believed that the scheme that we have outlined offers an explanation of every type of cytolysis known to occur in the sea urchin egg. It may not prove to be the correct explanation, but at any rate at present it is the only hypothesis that even attempts to explain the occurrence of E3 Igmanot T granule breakdown and vacuole formation. In all cases in nuclei, it does occur. Perhaps this statement should be qualified. Lilly and Cattell 25 mention the fact that F-alloiding treatment with electric currents. Some cytolysis was observed in Arbacia eggs. If this is the case, it is a phenomenon that merits further study, especially in view of the fact that the electric current is known to cause the formation of vacuoles in plant protoplasm, and in Protozoa CP235 and P236. Lilly and Cattell do not state whether the cytolysis they observed was accompanied or preceded by an increase in volume. It is possible that the passage of an electric current may cause the decomposition of those compounds of protoplasm, which contain bound electrolyte, EWEs. This certainly seems plausible, and it is not at all unlikely that calcium ions are set free when an electric current is sent through a cell. It seems clear that pigment granule breakdown and vacuole formation in the sea urchin egg, or acetylysis, depend on the occurrence of a surface precipitation reaction in the interior of the cell. We can then conceive of the reaction as occurring in 1. Calcium is set free in the cell interior. 2. Calcium reacts W at the pigment granule or some constituent of it to produce a substance which we have called ovothrombin. 3.12. Ovothrombin reacts with a substance in the exotoplasm, presumably a protein, to cause vacuole formation. These three stages of the reaction can be studied separately. The various ways high which calcium may be set free in the cell interior have already been indicated. Throughout the discussion, it has been assumed that those agents which dissolve the lipoids of protoplasm set free calcium as a result of this action. As a matter of fact, there is another cytosol effect of a fat solvent. Perhaps instead of releasing calcium, and thus indirectly causing a breakdown of the pigment granules, it acts directly by dissolving either the pigment itself or some other fat-soluble constituent of the pigment gland, which then reacts to form ovothrombin. It is possible that the disappearance of pigment granules in eggs exposed to fat solvents depends not so much on a breakdown of the granules as on a loss of pigment and a change in refractive index, so that the granules cannot easily be recognized, even though they may be present. Stage 2, in which calcium reacts with the pigment granule, can very readily be studied separately, and such study is now in progress. By taking the pigment granules outside of the egg and watching their behavior toward calcium in the presence of various other substances, much information may be obtained regarding the actual nature of the reaction which occurs between pigment granule and calcium. Some of the results that have already been obtained from this study have been given brief mention in the preceding chapter. In order to arrive at definite and certain conclusions regarding the nature of the reaction which occurs between calcium and ground, further study is necessary. Fortunately, the experiments are easy to perform, and it is probably only a question of time before the reaction will at least in some measure be understood. Stage 3, in which ovothrombin reacts to form vacuoles or precipitation films, has been studied scarcely at all. Further study of this stage is also isolated. In this clyapter, it has been shown that there is a reaction common to many and perhaps to all types of protoplasm. In this reaction, films or vacuoles typically appear in the protoplasm. Frequently, an escape of pigment or other dissolved substances can be noted. In all the types of protoplasm which have been studied, it seems evident to lie that the reaction in question occurs under essentially the same conditions and following treatment with the same reagents. It is therefore plausible to assume that the mechanics of reaction are fundamentally the same in all types of living substance. Our study has then brought us to the realization that there is a specific type of colloid chemical reaction which occurs in all sorts of living things and which is probably closely related to the life process itself. Study of the reaction in the egg of the sea urchin Arbacia has indicated that in many respects the reaction is similar to that involved in the coagulation of the blood in higher animals. This is to be deduced from the fact that the reaction is intimately related to the surface precipitation reaction discussed in the previous chapter, and it is thus essentially an internal surface precipitation reaction. No one who has read the chapter on the surface precipitation reaction can very well doubt that this reaction is similar to that which occurs when blood clots. We have thus but another illustration of the general sameness of living things. In spite of a manifold diversity, the underlying principles which govern the behavior of living organisms are everywhere much the same. In our interpretation of the mechanism of the reaction we have described, we have depended entirely on the facts presented by a study of the egg of the sea urchin Arbacia. Only future study can tell in how far this interpretation will fit the phenomena as they come to be known in other cells. This much at least seems certain. The hemolysis of red blood cells is so essentially similar to the so-called cytolysis of sea urchin eggs that it can hardly be supposed that the mechanism of the two types of reactions can be very different. In practically every instance, reagents which cause hemolysis also cause cytolysis and vice versa. In view of this similarity, it is especially satisfactory to note that already in the case of hemolysis, one or two authors have pointed out resemblances between the process of hemolysis and blood clotting. Several substances which prevent blood clotting have been found also to afford protection against hemolysis. See Pickering and D'Souza 23, Pickering and Taylor. Chapter 16. Cell division. In previous chapters, we have considered protoplasm more from a static than from a dynamic standpoint. But living substances continually undergoing changes which in themselves constitute life. In the final 2 chapters of this book, an attempt will be made to subject certain aspects of protoplasmic activity to colloid chemical analysis. Unfortunately, there is relatively little that can be said about this extremely important phase of the colloid chemistry of protoplasm. Our knowledge is as yet fragmentary, nor can we be certain that all the fragments rest on the sure basis of fact. Of all the visible, that is to say, mechanical processes that go on in living cells, the process of cell division is certainly the most universal. Every living cell is produced by the division of a parent cell. It is thus a characteristic of protoplasm to be able to reproduce itself by division. Because of the importance of CU division, and because too this process has been more carefully studied from a colloid chemical standpoint than some others, we shall begin on our discussion of protoplasmic activity by taking up the known facts with regard to the division of cells. To be usual, the process of cell division, or mitosis, is extremely complicated. As all books on biology and cytology explain, it involves such structures as centrosomes, astral rays, mammal fibers, etc. Mitosis takes place in a series of stages which are described in detail in cytological texts. The description as given in these books is usually based on the appearances in fixed material. In living cells, unless conditions are unusually favorable, not nearly so much can be seen. With certain types of relatively large and transparent cells, most of the structures seen in fixed cells can also be seen. In the live gene cytological studies made around 1880 C, early showed the presence of chromosomes in living cells. Astral rays and centrospheres are also sometimes visible. On the other hand, there is apparently no evidence of any fibers connecting the two centrospheres in living cells, in spite of the fact that such fibers absorb very clearly in fixed material. From a theoretical standpoint, there are a number of interesting problems involved in the division of a cell. In the first place, there is the question of what initiates cell division. Many cells in higher animals never divide. Protozoan cells divide usually only after they have attained a certain volume, but the mere size of the cell is not the only factor involved. The problem of initiation of cell division is especially interesting because of its close relation to the problem of cancer. This disease primarily involves an uncontrolled division of cells with a resultant increase in the mass of certain tissues. In the second place, the student of mitosis is interested in the chemical and physical changes which occur when a cell passes through the various stages of the process. An understanding of these changes would open the way to a physico-chemical interpretation of the mechanism of mitosis. At the present time, there is no lack of theories of cell division. Usually, it is easier to make a now theory of cell division than to test an old one. Most of the F. Rosen theories and hypotheses are based on pure speculation. An author postulates a mechanism which might conceivably affect the division of a cell, or he seeks to find in inanimate nature appearances which simulate the structures described by cytologists in dividing cells. There is no field of biological thought that has suffered more from the use of analogies than has the theoretical study of cell division. Even the crudest superficial similarities between mitotic structures and inanimate phenomena have been regarded as having real significance. Concerning the various theories of cell division, we shall have but little to say, and we shall for the most part confine our attention to the actual facts regarding the physical changes which take place during mitosis. The problem of the initiation of cell division has been attacked from various standpoints, and with various types of material. Some authors have studied the effect of certain chemicals on the division rate of protozoa. Thus, for example, Cephate 20 showed that the lithium ion had a specific effect, high initiating, or at least hastening cell division in paramecium. Some authors have also claimed that extract of the thyroid gland had an accelerating effect on cell division in paramecium, but this has subsequently been denied. In working with an organism like paramecium, it is hard to distinguish between an effect on the animal itself and an effect on its food. An interesting type of work is that of Heilbrunn. See Heilbrunn 21, also p. 254. This author found that various types of plant cells were stimulated to divide when they were placed in contact with injured or dying tissue. The effective substances given off by the injured or dying, as Heilbrunn calls wound hormones. In recent years, much interest has been aroused by the experiments of G.T. Gurewitz and his pupils. See Gurewitz 26. Gurewitz claimed that dividing cells give off radiations which induce divisions in other cells. The radiations were thought to pass through quartz but not glass, and Gurewitz believed them to be composed of ultraviolet rays. Unfortunately, Gurewitz never demonstrated that dividing cells gave off ultraviolet rays. He says that such rays are strongly absorbed by gelatin and cannot be photographed. But the books on spectroscopy give directions for preparing photographic emulsions relatively free from gelatin and sensitive enough to detect ultraviolet rays of even shorter wavelength than those which pass through quartz. Since the above was written, von Gutenberg 28 has published the results of observations made with such emulsions sensitive to ultraviolet rays. There is no evidence of any emission from root tips. Moreover, von Gutenberg has rather convincingly shown that Gurwich's striking results are most probably the result of uncritical experimentation. See also Schwartz 28. Because of the great interest in cell division, there have been various other types of work besides those mentioned. Many references to the literature may be found in the extensive review of Pratt and Malkowski, 27. For a review of the more morphological literature, see also Wasserman, 26. Much of the work on mitosis has been concerned with the investigation of factors which might possibly operate during the life of the cell and cause it to divide. But from the standpoint of the mechanism of mitosis, WC are concerned not so much with these factors, but rather with all the factors, normal or otherwise, which can incite a cell to divide. That there are many and varied types of treatment which stimulate to cell division is clearly shown by the work on artificial parthenogenesis. This work has the advantage that it is not concerned with an increase in the division rate of the cell, but with an initiation of cell division. An egg protoplasma, myography of Heilbrunn cell does not divide at all unless it is fertilized. And yet when certain types of eggs are treated with the proper reagents, THEIY can readily be made to divide. The initiation of cell division in marine ova was first discovered by Zero and R. Neutradwotke. Following their experiments and the experiments of Moai, a subject was studied in great detail by Loeb, Delaub, R.S. Luxi, and many others. These workers were for the most part interested in the theory of fertilization. They hoped insofar as possible to obtain a normal development of the treated eggs. But in spite of the fact that the students of artificial parthenogenesis did not consider the theoretical study of cell division as their main aim, the results in this field can nevertheless 1-0 used in the attempt to explain the mechanism of mitosis. When an unfertilized egg is treated with reagents which incite it to develop, the first important step in the development is the division or segmentation of the egg. Any reagent Wickli can initiate a fairly normal division, is almost certain to start the eggs on a dopmiant, which will proceed for a considerable time. For once the process of development is begun, the successive steps follow automatically. In the early work on artificial parthenogenesis, it was EY felt that certain specific substances were necessary for TULO, initiation of cell division, and subsequent development. Latu experiments, however, disclosed an ever-increasing ranges of treatments which were effective in causing segmentation in one type of egg or another. When an egg is fertilized, there is usually some sort of cluage in the egg cortex, and this cortical change is then followed by changes in the egg interior. So too in artificial fertilization, or rather artificial parthenogenesis, both cortical and incubal changes occur. This was first emphasized by Loa ECCO Joab Mill. His favorite method of inducing artificial parthenogenesis involved two successive treatments, often the reagents which, and appropriate cortical change, have little or no FET in initiating segmentation. According to Loeb, however, they constitute the real initiating factor. The second treatment is in the main. This general idea has been severely criticized L.Y. Jute 22, who points out that treatment of sea urchin eggs with hip eutonic solutions alone is as effective as any combination of successive treatments. It should nevertheless be clearly understood that in every type of egg studied, normal segmentation can only be successfully induced if it is preceded by some type of cortical change. Probably this cortical change has, among other things, the effect of removing the constraint due to the presence of a rigid membrane, Heilbrunn 15a 20c. Most eggs before fertilization are surrounded by a stiff membrane, and we can hardly conceive of their being able to constrict as long as this membrane is present. The removal of this obstacle occurs most frequently either as a result of a swelling or softening of the membrane, or following a lifting off of the membrane. In the egg of the clam, Cumangia, the membrane becomes swollen. In sea urchin eggs, the membrane is normally elevated, but with certain reagents and also certain conditions of insemination, it too becomes swollen. The cortical change can therefore be thought of as removing a block to segmentation. Concerning the nature of the factor which really produces an initiation of segmentation, there have been many theories proposed. In Loeb's later work, he held that initiation of development was due to a superficial cytolysis and an increase of oxidations. He thought of the superficial cytolysis as involving the swelling of a colloid at or near the surface of the egg. This colloidal swelling produced membrane elevation if the egg was left in the proper solution for just the right length of no longer treatment involved swelling of colloids throughout the entire egg, and the whole cell became cytolyzed. Loeb believed further cytolysis in itself produced an increase in the rate of oxidation, and TLUAs he also regarded as an important factor. From Tiho preceding chapter, it is evident that cytolysis cannot be thought of as simply involving a swelling of protoplasmic colloids. Instead of producing a liquefaction, as Loeb supposed, it really causes a gelation or coagulation of the egg contents. Nor does cytolysis of itself necessarily involve an increase in oxidations, in spite of the fact that this was claimed by Loeb and Weissnies 13b in their measurements of the oxidation rate of cytolyzing eggs. Loeb and Weissnies determined oxygen by an iodine titration method, and their determinations were rather a measure of the amount of iodine adsorbed by the organic matter so abundantly given off by cytolyzing eggs than a measure of the amount of oxygen consumed. See Heilbrunn 15b. In SEI of these criticisms that can be launched against Loeb's ideas, it is not at all unlikely that there is a certain amount of truth in the idea that cytolysis plays a real role in the initiation of segmentation. Loeb showed that very frequently too long an exposure to parthenogenetic agents produced cytolysis in the sea urchin egg. We shall see later that there is some reason for believing that the parthenogenetic agents produce an incipient cytolysis in the egg, and this is not so very different from a superficial cytolysis. Concerning the permeability theory of artificial parthenogenesis, we shall have little to say, as the discussion would lead too far afield. The chief advocate of the theory has been R.S. Lilly. His papers are cited in various books on experimental embryology. See, for example, E.R. Lilly. Perhaps initiation of cell division does involve an increase in the permeability of the cell membrane, but certainly the evidence that has been gathered is far from satisfactory. In recent years, fortunately, there has been a growing tendency to regard the permeability data critically. Most of the older observations, which seem to show increased permeability in eggs stimulated to divide, would hardly stand the test of this modern criticism. Various authors have suggested that artificial parthenogenesis, or the initiation of segmentation, might be due to a coagulation of the protoplasm. Loeb seemed to favor the idea in 1900, but he soon abandoned this view and became a violent antagonist of it. Delage for many years maintained that artificial parthenogenesis was the result of a coagulation, followed by a liquefaction, and he rather unwisely considered membrane elevation as one evidence of such a coagulation. However, in his book, Delage and Goldsmith T3, he became an adherent of the Lilly theory of increased permeability. Largely on theoretical grounds, the coagulation theory was vigorously proposed by Fischer and Oswald Fifield. They argued that all parthenogenetic agents are of such a nature as to produce coagulation of the protoplasm. At the time of Fischer and Ostwald's paper, there was no knowledge of which agents would or would not cause coagulation of the protoplasm, so that their arguments were not very convincing. Later, Ostwald 7 retreated from his original position and admitted that coagulation in artificial parthenogenesis might be only secondarily produced as a result of increased oxidation. The first experimental evidence either for or against the coagulation theory was that of Heilbrunn T5. He studied the initiation of cell division in the egg of the sea urchin Arbacea. Using the centrifuge method, he showed conclusively that all of the agents which incited the egg to divide actually did cause a coagulation or dilation of the protoplasm. Following treatment with saponin, with chloroform, with toluol, with isotonic sodium, was a very pronounced increase in the viscosity of the protoplasm of the eggs. In general, in the Arbacia egg, as Hetla Biciani points out, successful artificial P. bruthinogenesis is only possible if the egg is treated with hypertonic solutions. When eggs are treated with distilled water or with fat solvents, it is but a small % of the eggs that shows any sign of division, and this division is always abnormal. Apparently, only in the case of the sea urchin egg is treatment with hypertonic solutions an efficient method of inducing segmentation. Other types of eggs do not respond to this treatment. In the case of the annelid Nereas, just D5 found that treatment with temperatures of 33 to 35.0 caused a high percentage of the eggs to divide, and the division was generally normal. When Nereas eggs are exposed to temperatures of 33 to 35 DO, the first change is an extrusion of jelly from the cortex. At the same time, the large nucleus or germinal vesicle breaks down. This breakdown of the nucleus is accompanied or followed by a liquefaction of the protoplasm. If the eggs are exposed to the warmth just long enough to produce this breakdown of the nucleus and the subsequent liquefaction of the protoplasm, no segmentation occurs. But if the eggs are exposed a little longer to the warmth, then the protoplasm begins to show a coagulative ojange, which is very clearly demonstrable by the centrifuge method, Heilbrunn 25B. The coagulative change was found to occur after about 19 minutes exposure to a temperature of 33 K, and after about 13 minutes exposure to a temperature of 34.9. If the eggs are to be stimulated to divide, they must be allowed to remain in the warmth until this coagulative change has occurred. Similar results were obtained with the egg of the mollusk Kumengia. This egg may also be stimulated to divide by heat treatment, but again, only exposures long enough to produce coagulative changes in the protoplasm are effective. Finally, in the egg of the sea urchin Aracia, in which exposure to heat produces only a small percentage of divisions, the heat must cause a coagulation of the protoplasm if it is to have any effect. It is evident that the experimental evidence, as far as it goes, is all in favor of the view that stimulation to cell division, in the case of the eggs of marine invertebrates, is always accompanied or rather preceded by coagulative changes within the protoplasm. Apparently, in plant cells, too, stimulation to division is dependent on a coagulative change in the protoplasm. In various types of plant tissues, injury or wounding causes many cells to divide, which would otherwise have remained in the resting state. It is a well-known fact that when plant tissue is cut or otherwise injured, there is a stimulus to growth or proliferation, which may manifest itself in the formation of a callus, or may result merely in the regeneration of missing elements. This knowledge is very old and must go back to the ancients. Unfortunately, the standard books on plant physiology, usually so full of pertinent information, tell very little about this process. The books on regeneration are more concerned with the problem of form than with the stimulus to growth. A useful paper which may be consulted is that of Massot 98. This has references to some of the older literature. In recent years, the subject has been studied experimentally by Heilbrunn 1321. This author has brought forward evidence to show that the stimulus to division is produced by the formation of special chemical substances, which he calls hormones. Toward the present, this point need not concern us. What Heilbrunn has clearly shown is that when various types of plant tissue are cut, a stimulus to division passes to the cells only several layers distant from the actual cut. Now it seems certain that in these cells the protoplasm becomes coagulated, or rather undergoes a great increase in viscosity. This has been shown by various authors, CP 128, but especially by Buenin 26 AB. It will be remembered that this author in 3 types of plant material found viscosity increases of about 400 to 600% in the protoplasm of cells near a cut surface. The viscosity increase was reversible. It is interesting to note that the magnitude of the viscosity increase is very similar to the magnitude of the viscosity increase which precedes mitosis in some types of marine egg material, see below. Leonard's work clearly demonstrates that the initiation of cell division, which occurs in plant material in the neighborhood of a cut or wound, is accompanied or preceded by a gelation of the protoplasm. Perhaps in animal tissue, too, the stimulus to division, which follows a cut or a wound, and which results in healing or regeneration or sometimes in abnormal growth, may depend on the production of a coagulative change or a gelation in the protoplasm of the cells which divide. But in animal tissue, there is no direct evidence in support of this point, and in most animals, the problem is complicated by the migration of cells to the neighborhood of the cut. Gutting or wounding is not the only stimulus which incites plant cells to division. Heilbrunn, 19 B and 20 describes cell division following treatment with hypertonic solutions in the hair cells of Colloid linearis, in the cells of the scales of the onion, and in cells of Elodia. These cells therefore behave like sea urchin eggs. There is not much question but that in the plant cells too, the hypertonic solutions cause a marked increase in protoplasmic viscosity, both in various types of exolant tissue and in the eggs of marine invertebrates. Those agents which stimulate cells to divide regularly cause a coagulative change or gelation in the protoplasm. Concerning the possible nature of this change, we shall have more to say later. That it is directly related to the division of the cell is indicated by the exothrecrements of Heilbrunn 20A. In these experiments, it was shown that if the protoplasm of sea urchin eggs was kept fluid, no mitosis ever occurred. There is an abundance of evidence, see chap 11, that fat solvents generally, if they are present in low concentration, have a liquefying action on protoplasm. Wholly owing fertilization, herbaceae normally complete their first division after about 50 minutes, and the division is preceded by a coagulation or gelation of the protoplasm. If, however, Herbaceae eggs after fertilization are in solutions containing fat solvents in the X3 per concentration, the gelation which precedes cell division is prevented, and under these conditions the egg always fails to divide, although it is not permanently injured. We are thus led to conclude first that stimulation to cell division, no matter how P-induced DJ, always involves a gelation. That is to say, a large increase in protoplasmic viscosity. And second, that prevention of this gelation prevents the division of the cell. This is strong evidence that the gelation is intimately related to the division process. To know that a gelation or a sharp increase in protoplasmic viscosity precedes cell division is important, but it is still more important to inquire into the nature of the gelation and to discover, if possible, the mechanism which underlies it. Before going into this more theoretical phase of the subject, we shall consider the evidence concerning the viscosity changes during the whole course of the mitotic division. Here we are on more certain ground. Reinke 95, upon subjecting unfertilized and fertilized urchin eggs to pressure, noted that the protoplasm flowed less readily from the fertilized eggs. Similar observations were made by Abe Keck 98. The differences noted by these authors were in part due to changes in viscosity, but they were also quite as much due to differences in the boundary conditions of the emerging protoplasm, especially in view of the fact that the fertilized urchin egg is surrounded by very different membranes from the unfertilized egg. Kite 13 and Kite and Chambiz 12 dissected various types of dividing cells with microdissection needles. Apparently, they noticed no changes in the viscosity of the protoplasm, although they must have been on the lookout for such changes, for Kite makes some vague general statements as to viscosity changes which might theoretically be supposed to occur. Heilbrunn TF5 centrifuged fertilized and unfertilized Herbaceae eggs simultaneously, and found that the granules moved much more readily through the egg before fertilization than after. He concluded that the sperm caused a gelation of the protoplasm of the egg, and that this gelation was directly related to the formation of the mitotic SX triangle. Heilbrunn's experiments were not very numerous, and moreover he did not guard against a disturbing factor, which tends to obscure the results in the fertilized sea urchin egg. In Herbaceae, as soon as the egg is fertilized, the pigment granules migrate closer and closer to the egg periphery. They assume a position near the outer border of the cell. This is a fact long known, but not very often mentioned in the literature. The much more accurate data regarding the viscosity increase in the protoplasm of the sea urchin egg following fertilization and during the various stages of mitosis were obtained by Heilbrunn 1728 al. He centrifuged the eggs at short intervals during the whole period between fertilization and cleavage, and his results show clearly the viscosity changes which occur in the main mass of the protoplasm. As has just been mentioned, following fertilization, most of the pigment granules migrate to the cortex, and there they are not affected by centrifugal force, but remain firmly lodged when the egg is centrifuged. This failure of the pigment granules to move may be due either to their position close to the outer membrane of the cell, or to the fact that there is some sort of a gelation or a coagulation which occurs in the cortex of the egg following fertilization. Heilbrunn 23, on the basis of cutting experiments with fine steel needles, is of the opinion that there is such a change. But, as has been pointed out previously, CP 41, no definite conclusions can be drawn from this sort of evidence. In the main mass of the protoplasm, there is a very pronounced viscosity increase soon after fertilization. This reaches a height just before the appearance of the mitotic spindle. As soon as the spindle has been formed, the viscosity decreases again, finally rising just before division, Heilbrunn 21. In 1917, C.R. Ambers again took up the microdissection of dividing cells, and in his preliminary note, 17a, he describes changes in viscosity during mitosis. According to this account, the astral rays are fluid, and the regions between them solid. Chambers states, a periodic reversal of the soul to the gel state and vice versa has been demonstrated in the cell protoplasm during division. The steps taken may be divided into the following series. A. When the monoster is fully formed, the greater part of the cell is a gel. As the centrifugal fluid collects on the two poles of the nucleus, the cytoplasm reverses to a sol state, and the monastral radiations fade out. C. The formation of radiations about the centros gigahertz, one on each side of the nucleus, produces the diester and is accompanied by a return to the gel state. D. A return to the sol state later takes place in the equator of pli cell. E. The nuclear spindle now divides, followed by a constriction around the middle of the cell, which continues until the cell is cut into 2F. The reversal of the gel to the sol state usually starts in the equator of the cell and spreads to the poles. Apparently in this account, Chambers believes that the protoplasm is first a sol, then a gel, and a sol again, then a gel, and then a sol again. As to the times when these changes occur, he supplies no precise data. Chambers 17b gives very little additional information concerning viscosity changes in the mass of the protoplasm following the entrance of the sperm, an aster is formed. That the cytoplasm around this has been rendered fairly solid is shown by the fact that the aster at this stage may be ushed and rolled in the surrounding liquid cytoplasm. Certain other viscosity changes are inferred, but no definite times are given. After us CAs of time, about 10 minutes, a change takes place in the cytoplasm about the equator of the nuclear body, which appears to be a reversal to the sol state. I was not able to convince Mysek on this point by the use of the needle. Apparently this is the change referred to under D in the quotation from the 1917 paper, but now Chambers is not certain of a change in viscosity. Chajvabi's 19 reports further observations on the changes in viscosity during mitosis. A practically complete statement of these observations is given in the following quotation. Immediately after fertilization, the cytoplasmic granules readily flow by the moving needle. This is taken to indicate low viscosity. When the sperm aster is at its full development, the highly viscous state of the protoplasm is detected by the needle. This condition is at its height 10 to is minutes after fertilization. 15 to 20 minutes after fertilization, the radiations of the aster begin to fade from view, with a reversal in the cytoplasm of the semifluid toward the end of this stage. Toward the end of this stage, which lasts for 20 to 30 minutes. Shortly before cleavage, about 40 to 50 minutes after fertilization, an increase in firmness sets in the time of appearance of the amphiaster until completion of cleavage lasts from 10 to 15 minutes. The increased viscosity of the egg during this amphiaster stage. After the completion of the cleavage process, There are indications that the firmness of the cytoplasm persists. In 1919, Chambers believed in a succession of sol, gel states, but in 1921, Chambers 21, he has again returned to his 1917 opinion that just before division there is a liquefaction of the protoplasm. In order to understand Chambers' views, an attempt was made to record his 1919 observations in rough graphic form. No exact reproduction is of course possible, for the time intervals given by Weber's W will be different for every temper ratio, and Chambers makes no mention either of temperature or of the total time between fertilization and cleavage in any given experiment. In Figure 14, the dotted line represents Chambers' 1919 results. The C's indicating the pumps at W at each observations were recorded. On the same graph are plotted the results of the unbroken line is plotted from the centrifuge data. Heilbrunn, the dotted line is a rough estimate based on the microdissection of experiments, Heilbrunn centrifuge tests, mostly from the 1920 data. These centrifuge data refer to Aracia egg. In Chambers' experiments, it is not always clear which T3RP of egg he is referring to. The abscissae of the graph are in minutes, and in every case, for the centrifuge data, the time has been corrected to fit in with a standard time between fertilization and cleavage of 50 minutes. For example, if the actual time between fertilization and cleavage was found to be 60 minutes instead of 50, each time interval was multiplied by 5/6. The ordinates represent viscosity values. The height of the curve is more or less speculative, but is based to some extent on unpublished experiments. In the ordinate at that point, the inverted V's indicate viscosity values greater than the value for the ordinate at that point. The length of the V's is also significant, a large V indicating a much lower. It is to be noted that both Chabs and Heilbrunn find the same cycle of viscosity changes, but the two curves are far from identical. The very stage in which Heilbrunn records the lowest viscosity is for Chambers a stage of highest viscosity. Chambers thinks that at the time of the amphiaster, the viscosity of the protoplasm is at its peak. Centrifuge tests indicate that when the spindle is fully formed, the viscosity is low. The dew point is an important one, for on the basis of his belief, Chambers has expressed a general theoretical interpretation of the mechanics of mitosis. The microdissection estimates of Series 20 agree with the centrifuge tests in regarding the amphiaster stage as one of low viscosity. It might be worth mentioning that if centrifuge tests are made at relatively high temperatures, for example at 27 or 28 degrees, results more like those of Chambers can be obtained. Under these abnormal conditions, the liquefaction which normally follows the appearance of the spindle becomes less and less pronounced at higher and higher temperatures, and eventually at temperatures just below the death temperature. No such liquefaction occurs in the injured cells. Perhaps the effect of higher temperatures may be a factor in explaining Chambers' results, for in his earlier experiments, he was apparently rather careless about guarding against heating effects, CP27. But it is doubtful whether it is worth our while to attempt to explain the divergent findings of a method of viscosity determination, which up to the present has never given dependable results. As the reader of this book must realize, in every instance in which the microdissection method has been used as a means of testing viscosity, it has failed to yield results consistent with those obtained by various other methods, all of them superior to it in accuracy. See Chapter 4 for a discussion of the principles of viscosity measurement. So far, we have considered only the egg of the CN archene. The Semingia egg, the viscosity changes during cell division have been studied in much more quantitative fashion. This egg is in the metaphase of the first polar body division when it is shed into the seawater. At this time, the egg protoplasm is highly fluid, and only a few turns of the handle of a hand centrifuge are sufficient to cause a complete stratification of the granules of the CP45. The protoplasmic viscosity of the Semingia egg during the interval between fertilization and the first cleavage increases just before the completion of the first maturation division, drops again as the second polar spindle develops, rises as the second ilar body is given off, to decrease again with the appearance of the amphiaster of the cleavage spindle. The complete curve of the viscosity changes in the Gumingia egg is given in figure 15. W Fitch is taken from Heilbrunn and Toile in this graph. The x-axis represents time in minutes after fertilization, the total time between fertilization, and the moment when 50% of the eggs segmented, being taken as 50 minutes. This was regarded as a standard time interval, and observations made on eggs, which required a longer or shorter time, for the period between fertilization and cleavage, were plotted by making a correction for the time. The ordinates in figure 15 are arbitrary viscosity units and represent seconds of centrifugal turning necessary to cause easily visible migration of the granules of the egg. As a matter of fact, these arbitrary viscosity units happen to be in rather close agreement with absolute viscosity presents a centrifuge test. The V's and inverted V's have the same significance as in figure 14. Inspection of the curve shows that the viscosity undergoes an increase of at least 500 or 600% and then decreases again to approximately its original value. The viscosity changes in the C. meningia egg are more pronounced than those of the Aracia egg. In the egg of the annelid nereis, there is even less change in the viscosity of the protoplasm during mitosis. When the nereis egg is shed into the seawater, it has a large germinal vesicle. The viscosity of the protoplasm at this stage is very high. At any rate, the granules do not move through the protoplasm when the egg is centrifuged. Soon after fertilization, the nucleus or germinal vesicle breaks down and there is a marked decrease in the viscosity of the protoplasm. This is followed by 3 successive cycles of viscosity change at a temperature of about 2040. The viscosity rose to maxima at 20:35 and 47 minutes after insemination, and these TEM.E's probably correspond to the times when the two polar body spindles and the cleavage spindle were forming. In this connection, it should be noted that the telophase of one division is really the prophase of the next, so that when the viscosity rises during the telophase of one division, it remains high during the prophase of the next division. At 65 minutes after fertilization, there is another period of high protoplasmic viscosity, and this is followed in a few minutes during the segmentation of the egg. 50% of the eggs show cleavage furrows at 69 minutes after insemination. The changes in the viscosity of the near egg are not very great and are not easy to follow. It is interesting, therefore, to note that Fobbs and Thacker 25, in the course of other observations, reported an increase in the protoplasmic viscosity of the nereis egg 20 minutes after insemination. Thus, even in the nereis egg, in which the viscosity change is relatively slight, it has been possible to confirm Heilbrun's observations. It is much easier to confirm them for the sea urchin or Gumingia egg. Many observers at Woods Hole have noted the cycle of viscosity change in the egg of Arbata. Hunstrom 2T has also reported confirmation for a European species of sea urchin. It is an advantage of the centrifuge method that its findings are sufficiently objective so that they can be repeated. From Rippert's brief statements, P. Hupper 24, it seems safe to conclude that in the Ascaris egg there is also a decrease in viscosity following the appearance of the mitotic joindle. Rippert used the centrifuge method, but his main interest was in the effect of ultraviolet rays rather than in the viscosity changes which occur during cell division. In Arbata, Gumingia, and Nereis eggs, there is a high viscosity of the protoplasm in the prophase before the completion of the spindle. Then, as soon as the seahedal is fully formed, the main mass of the protoplasm becomes more fluid again. Division of the cell is preceded by a sharp increase in viscosity. This viscosity increase occurs in the moment or two before the appearance of the furrow, just at the stage when the cell elongates preparatory to division. In figure 15, this elongation is referred to as mitotic elongation. It is what German authors call karyokinetische Streckung. If the main mass of the protoplasm is fluid during metaphase and anaphase, it must not be thought that the spindle itself or the astral rays are fluid structures. The fact that the spindle itself is moved as a whole by centrifugal force is evidence enough that it has form. That is, therefore, a solid or semi-solid body. Practically all the authors who have used the centrifuge in embryological experiments have described such movements of the spindle. The experiments of Koikern 17 are perhaps particularly impressive in this connection. Moreover, Heilbrunn 20a has brought forward evidence to show that the astral rays are elastic structures. When our beta eggs are centrifuged soon after fertilization, the contour of the egg remains smooth or even, and the eggs are generally spherical, at least after they have been removed from the centrifuge tubes. But as soon as the mitoculation begins, the eggs assume irregular shapes when centrifuged. They do this not only when the main mass of the protoplasm is of high viscosity, but also in the anaphase when the protoplasm as a whole is flipped. Heilbrunn concluded that there are gelatinous strands which retain their connection to the egg surface and pull upon it when the egg is centrifuged. Heilbrunn also cites additional evidence to show that the astral radiations exert a pull on the surface of the cell and are thus gelatinous strands. Soon after fertilization, the surface contour of the untreated cell is perfectly smooth. But with the first appearance of the amphiaster, it becomes crenate, as though tension while being exerted on it from a number of points on the inside. These crenatina occur more plainly if the egg is viewed from one pole of the mitotic spindle. They can be seen under the low powers of the microscope, and in view of the fact that the Arbacia egg has been so often studied, it is rather surprising that other authors have not commented on their occurrence. If the astral rays pull on the asymphyse membrane of the cell, this is of course an important factor to be considered in any theoretical interpretation of the mechanics of mitosis. The pull of the astral ray would create local differences in the tension of the cell membrane, and these would of course vary with the development of the asters and their connection or lack of connection with each other. So far, we have considered only the viscosity changes in the dividing eggs of marine invertebrates. Many students of sea experimental embryology have centrifuged the eggs of the frog, but their interest was almost wholly in problems concerning the organization of the egg. From this older literature, Oddquist has attempted to obtain information concerning the viscosity of the egg protoplasm at various stages of mitosis. He cites 3 papers, G8 and Kano Ioka 8. From Giravitz 8, he rescues the following statement. Der Zelldurchskamierung Wick auf der FM'schen Bildung geht eine betriebliche Vordichtung des entsprechenden Plasmastreifens voran. See footnote on P517. Is though only statement in UHS papers that can be taken as indicating viscosity change, and as far as can be judged from reading the context P40V1 materials in the centrifuge egg. Kono full. Apparently the egg protoplasm, it losa use 15 minutes after fertilization than before fertilization, and apparently it is still less viscous 1.5 to 2 hours after fertilization. Just after the first segmentation, the viscosity is like that of the eggs 15 minutes after fertilization. Interpretation of viscosity change in the frog egg is complicated by the fact that the egg takes up water after it leaves the oviduct. Bogyukiekis criticizes Konopaka's results on the ground that she did not use eggs from the same batch in comparison experiments. Ogyukiekis self-centrifuged eggs 1, 2, and 3 hours after fertilization, just before the first cleavage. Just before the second cleavage, etc. His results indicate viscosity differences during the mitotic cycle, and he believes that in general they fit in with Heilbrunn's results for marine eggs. In view of the fact that the viscosity of protoplasm may rise and fail, all within the space of a few minutes, it seems certain that viscosity tests made at intervals of an hour are not sufficiently frequent. Oddquist centrifuged the eggs at more frequent intervals during the second cleavage. Here his results are more interesting, especially in view of the fact that in this period there are no maturation divisions. He found relatively high protoplasmic viscosity 20 minutes after the first cleavage, and decidedly lower viscosity 20 minutes later. The time between the first and second cleavages was 50 minutes. These results fit in very well with the results shown in figures 14 and 15. However, immediately after each cleavage, the viscosity in the frog's egg appears to be relatively low. This evidently constitutes a difference between the two types of material. It seems probable that in many cells, the successive mitoses are not as closely linked as they are in the eggs of marine invertebrates. In sea urchin eggs, the end of one division is the beginning of the next, and for this reason, perhaps, there is a viscosity increase at the conclusion of each division. Such a final viscosity increase may be lacking in other cells, and there is good evidence that it is lacking in the case of the frog's egg. Zimmermann 23 studied the viscosity of the protoplasm in dividing cells of the alga Aesophacillaria fusca. Normally, the protoplasm in the cells of this alga is in the form of lamellae between vacuoles. In order to study the viscosity of the protoplasm during mitosis, Zimmermann seems to have first plasmolized the cell, or at any rate to have changed the protoplasm in such a way that it was no longer a foam. Concerning his results, Protoplasma Monographien 1, Heilbrunn 18, he says, Wir können also in der Prophase eine Zunahme der Plasmaviskosität beobachten, in der Metaphase eine rasche Abnahme nachher. Wieder eine allmähliche Steigerung. Die Versuche stimmen lieber ein mit Zentrifugierungsversuchen, die amerikanische Forschungsstätten IAW Bill Betten 1920 an tierischen Zellen unternommen haben und ferner mit meinen Beobachtungen über die Brownsche Bewegung. Some of Meitnen's data do not appear to be very completely not included in the published paper, but as far as his results go, they are in direct confirmation of the results of Heilbrunn on marine eggs. Combex, in his chapter in Cowdery's General Cytology, Chambers 24, is in error when he states emphatically that Zimmermann's results are in contradiction with those of Heilbrunn. This error was pointed out to Chambers, and it does not appear in a later imprint of the book. It is evident that in various types of cells, Mayle's changes in the viscosity of the protoplasm occur during the process of cell division, and these viscosity changes are doubtless associated with changes in the colloidal state of the colloids of the protoplasm. There is also other evidence in support of this view. As long ago as 1882, Fleming in his book on the cell, Fleming 82, see for example p. 206, pointed out that during the process of mitosis, there are changes in the refractive index of the cytoplasm. Ho states that during the prophase, the refractive index increases. Fleming regarded these differences in refractive index as due to changes in the consistency of the protoplasm, and to these changes in consistency, he attributes the differences in the standing properties of dividing cells observed both by himself and by Van Beneden in 1875. When fixed with osmic acid, dividing cells are much darker than resting cells, see especially Fleming 91b. In living extrailium cells of the salamander, Fleming describes a clear zone in the center of dividing cells, a zone which probably corresponds to the clear area between the astrospheres in dividing sea urchin eggs. This clear zone Fleming regards as more fluid, for he can see Brownian movement in it. But this is hardly a valid criterion, for Brownian movement would always be more vigorous in the presence of fewer granules. In a study of dividing connective tissue cells, Fleming 91a, CP 261, notes that cells which had become dark in the early stages of mitosis became light again in the late anaphase. Finally, in a general paper, 91b, Fleming again takes up the question of the optical changes of cells during division, urging that these changes are a constant characteristic of dividing cells, and that they must have physiological significance. He notes with regret that practically no mention of these optical changes is made in the many papers on cell division. That A3 appeared between 1882 and Levi 17 also describes changes in the refractive index of dividing cells. But his observations are not as complete as those of Fleming 4, and he is apparently unaware of the older literature. Fleming and Levi estimated the refractive index of the cells they studied. The first attempt at a quantitative measurement is that of Lull 21AB. Inslee had the ingenious idea of using the sea urchin egg as a miniature lens, and then from a measure of the focal distance of this lens to calculate the refractive index of the protoplasm. In making his calculations, Valais is obliged to neglect the presence of the fertilization membrane of the egg. He says that the presence of this membrane can have only a negligible effect, for it must have a refractive index extremely close to that of seawater. Actually, there are two membranes around the fertilized egg of the sea urchin, the fertilization membrane and the hyaline layer. Neither of these membranes has a refractive index very close to that of seawater, for both are clearly and easily visible, and this would not be the case if their refractive indices were very close to that of seawater. Hence, Vles's method of calculating the refractive index is not exact. In Vlies's observations, he finds a decided change in refractive index only at the time when the egg changes in shape, just before division. There may be a real change in refractive index at this time, but it is equally possible that the difference which Vlies noted may have been due all or in part to the inaccuracy of his method of calculation, based as it is on the assumption that the egg is not surrounded by membranes which differ appreciably in ogidical qualities from the seawater. If VNH's method of calculation is inexact, this inexactitude would only become apparent while the egg change shape. It is possible to remove the fertilization membrane from fertilized sea urchin eggs by shaking them vigorously 1/2 minute after they are inseminated. In this way, it would be possible to get rid of one of the disturbing membranes, and one could make somewhat more accurate determinations of refractive index by Vlies's method. But even if the fertilization membrane were shaken off, the hyaline membrane would still remain. Vlies 24 also studied the surface forces of the egg during cell division. By surface forces, he understands all those forces which resist deformation. See Chap 6, P99. If the astral rays are our strands which pull on the periphery of the egg, CP 272, it is obvious that when the amphiester is fully formed, the surface forces of the egg, that is to say the forces which tend to prevent the egg from flattening, would be relatively great. From his observations, Vlies actually concludes that this is the case. Unfortunately, the data host submits are not very satisfactory. The presence of a fertilization membrane would interfere with Weiss's method of measurement. For a discussoire of this method, see P99. In order to prevent the appearance of a fertilization membrane, Weisses removes the jelly from the eggs. He is apparently under the impression that membrane elevation in the sea urchin egg depends on the presence of the jelly. This is certainly not true, as has frequently been shown in recent years. To remove the jelly, Vlies treats the eggs with 3% KCN. It would have been simpler to shake them or to centrifuge them, and far less injurious. Vlies's conclusions are based on the study of 5 eggs. 1 hour and 20 minutes after insemination is the time at which the surface forces are greatest, and the tendency to resist flattening the greatest. Vlies's actual data for this time show that 2 of the 5 eggs did resist distortion more than the average. A third was about average, and a fourth resisted distortion less than the average. The 5th egg Vlies did not measure exactly at 1 hour and 20 minutes after insemination, but in view of the fact that this egg reached the diester stage some time before the others, it is more appropriate to use the measurement made at 1 hour 10 minutes after insemination. This 5th egg resembled the 4th egg in showing less tendency to resist distortion than the average. It is thus difficult to obtain any certain information from VNH's de Fora. Moreover, it is not easy to understand how he could have derived any information regarding surface forces of the egg from a study of its eccentricity or flattening during the time the egg was elongating just prior to division. It is not surprising that at this time Vlies found the egg more eccentric. It might, of course, have been possible to draw some conclusions if in the egg's measured. The axis of the spindle had in every case been exactly in the axis of the horizontal microscope, for if one looks at an ellipsoid from one end, it appears spherical. But of the 5 eggs studied by Vlies, only one had the axis of its spindle exactly on the axis of the microscope, and even in this case it is hard to see how one could be certain that the 2 axes were identical. If Lee's experiments are ever repeated, and it is to be hoped that they should be performed on 100 eggs instead of 5, there should be no treatment with 3% KCN, and the fertilization membranes should be removed by vigorous shaking 30 seconds after insemination. In some cells, the protoplasm shows amoeboid movements just before the cell divides or during division. This was noted by Pebmasoko in 1879. More recently, it has aroused the interest of the students of tissue culture B. Teach Browse, T3, and Lisi Browse, 23, Busciante, 27. Strangeways finds that in the prophase, the protoplasm is quiet, but that in the anaphase, it shows very vigorous movement. This is probably an indication of greater fluidity, although the mechanical disturbances caused by actual division may be responsible for the movement. Probably in cells which do not undergo rapid successive divisions, the viscosity of the protoplasm is low at the conclusion of cell division. Many authors have described the rhythmic variations in the susceptibility of dividing cells to various toxic substances. Such rhythmic changes in susceptibility were first noted by Leon Dufour, and they have been studied by many subsequent workers. Most of these workers interpret them as due to a periodic change in the permeability of the cell during mitosis. But this explanation is scarcely tenable, for there are rhythms of susceptibility not only to the action of chemicals, but also to the action of heat, cold, and light, for rhythmic susceptibility to HHIT during mitosis. See Heilbrunn 24. Apparently, the rhythmic susceptibility of dividing cells is to be correlated with the rhythms of colloidal change, which are known to occur during mitosis. In the discussion so far, it has been shown first that all the agents which stimulate cells to divide cause a pronounced viscosity increase, that is to say, gelation or coagulation of the protoplasm. And in the second place, it has been shown that certain characteristic changes in viscosity occur during the division of many different tick-seas of cells. It has moreover been shown that the presence of these viscosity changes may be correlated with other known facts regarding the behavior of the cell during mitosis. From the mass of evidence that has been presented, it seems safe to conclude that our knowledge of the viscosity changes in the protoplasm during the division of a cell rests on the sure basis of fact. We know that there is a viscosity increase during the prophase of the mitotic division, and TLI evidence is good that this increase is closely related to the mechanics of the division process. But as to the causes which underlie the viscosity increase, we are still very much in the dark. If we are really to come to an understanding of the colloid chemistry of protoplasm, we must seek to explain the colloidal changes of the living substance in terms of the colloid chemistry of inanimate materials. Probably this is as yet scarcely possible, but it may be worthwhile to consider what possible explanations may be offered for the mitotic gelation. Apparently, there is some sort of correlation between the size of the nucleus and the viscosity, BF the protoplasm. In general, the larger the size of the nucleus, the more viscous the protoplasm. Eggs in the germinal vesicle stage always have a very viscous protoplasm. It has already been mentioned that the immature Nereis egg has J neurotopiasm of high viscosity. This is also true of Arbacia eggs, which are immature and still have a germinal vesicle. And as a matter of fact, it seems to be generally true of all types of immature eggs. Moreover, in stages of mitosis, in which the nuclear membrane is broken down, and there is no nucleus as such, the viscosity is always low. But the correlation is not absolute. Thus, in the Cumingia egg, when the cleavage spindle forms and the exorotoplasm changes from a state of high viscosity to a state of low viscosity, this change does not occur just at the stage when the pronuclei break down, but slightly before. Again, in the Cumingia egg, when the second polar body is fully formed, the viscosity of the protoplasm is low, and yet at this outtime the male pronucleus is intact. It is thus not at all certain that there is a direct relation between the size of the nucleus and the viscosity of the protoplasm. Although in some instances, at least the breakdown of the nucleus does appear to have an effect on the 3 protoplasmic viscosity. In a series of interesting papers, Reinenstrom 24AB, C has attempted to interpret the mitotic gelation which occurs after fertilization in the sea urchin egg. He believes he can demonstrate by dark field study that the lipoids of the egg undergo an increase in degree of dispersion. Following this increase in degree of dispersion, Tingsbaum believes that the lipoids unite with the proteins, The following quotation is from the 24cc. Unsere Arbeitshypothese über das Wesen der Plasmaveränderungen bei der Befruchtung kann nun etwas weiter ausgearbeitet werden. Die Lipoid befinden sich bei den unbefruchteten Eiern in einem verdichteten Zustande, was die Wasserbewegung bei diesen hemmt. Nach der Befruchtung wird die Dispersität der Lipoide erhöht. Diese verbinden sich dabei zum Teil mit den Eiweißstoffen zu einem kolloidalen Komplexe, das andere Eigenschaften wie die einzelnen Bestandteile erhielt. As a working hypothesis, this is perhaps as good as any. The truth of the matter is that we are still very much in the dark. There are, however, a number of definite facts that are known regarding the mitotic gelation. These may be summarized as 1. Comparing the eggs of Cumingia, Arbacea, and Nereis, the gelation is more pronounced the smaller the size of Tli egg. 2. The gelation is prevented and also reversed by ether and other fat solvents. 3. Cold has a similar effect. In addition, it may be worthwhile to refer to some preliminary experiments, which it is hoped to extend and amplify sometime later. Before fertilization, the protoplasm of the sortian egg behaves as a true fluid. That is to say, the viscosity, as determined by the centrifuge method, is independent of the amount of centrifugal force. It might be thought that, following fertilization, it would no longer show this behavior, for we might very well expect that with an increase in viscosity, it would show evidence of elasticity. However, in a few tests, no difference in viscosity of protoplasm of fertilized eggs could be noted when tests were made at different centrifugal speeds. These tests are not very easy to make, for one must compare the protoplasm of 2 different sets of eggs at the same number of minutes after fertilization. In the Cumingia egg, the tests that have been made also indicate no difference in viscosity values of protoplasm, centrifuged at different speeds at comparable times during the mitotic cycle. Thus, even during periods of high viscosity, the protoplasm of the Arbacea and Cumingia eggs seems to show the properties of a true fluid. Further evidence of the same sort is provided by the fact that Walls was unable to find any appreciable anisotropy in the dividing circle egg. According to Valal, Zabrera also failed to find anisotropy in dividing plant cells. The observed cells were under the pressure of a cover slip, and under these conditions one would expect a gel or an elastic fluid to show anisotropy. See chap. 6. These results seem to indicate that the viscosity increase of the protoplasm during mitosis is not due to the formation of a network or anything of the sort in the protoplasm between the granules. They also do not favor the view that the viscosity increase can be due to any attraction of the granules for each other, as might have been supposed. Almost the only interpretation that is left is to assume that the mitotic gelation or increase of viscosity is due to the precipitation of granular elements within the dispersion medium which lies between the granules. In order to test this view, fertilized Arbacea eggs during the period of high viscosity were centrifuged for a very long time at a high rate of speed. If the high viscosity is due to the formation of new granules, and if these are moved by centrifugal force, then there ought to be an increase in the volume of the accumulation of granules thrown to one side of the egg by the action of the centrifugal force. No such increase could be noted, and if it occurs, it is certainly not great. It might thus appear that there is no possible interpretation that could fit the facts, but there is one way out of the difficulty. We can assume that the mitotic gelation is caused by the precipitation out of the fluid part of the cytoplasm of granules so small as to be for the most part invisible, and so small also that they would not readily be affected by centrifugal force. At present, this is merely a formal explanation, and it would need more suppository evidence before it could be accepted. It is a point of view that can perhaps be built upon. One might assume that the hypothetical granules were lipoid, and that they were dissolved by fat solvents. One might also assume that the granulae had something to do with the formation of the astral rays and spin. The advantage of hypothetical granules is that one can assume anything about them, but until more actual evidence is forthcoming, we must admit that this hypothesis is too uncertain. There is another line of approach that is also interesting. Following fertilization in the sea urchin egg, there is a sudden increase in the amount of pigment given off by the eggs. This is rather a well-known fact, and it has been used by deist Lily Levina as evidence for an increase in permeability following fertilization. In the preceding chapter, it has been pointed out that escape of pigment does not depend on permeability, but rather on the loss of pigment from the pigment granule. Moreover, it was noted in the last chapter that escape of pigment was always associated with a specific type of reaction, which, when it was complete, involved a vacuole formation in the cytoplasm. Is the fact that pigment is given off immediately after fertilization evidence that the reaction which we described in the last chapter is really at the basis of the metaticulation? Apparently, it cannot be so regarded. For in the sea urchin, egg vitalization is accompanied by the lifting off of a membrane. The momentarily naked protoplasm covers itself with a new film, the so-called hyaline layer or hyaline membrane. And this film formation is certainly a form of the surface precipitation reaction, and as such probably involves a loss of pigment from the pigment granules. The escape of pigment following fertilization in the Arbacia egg thus seems to depend primarily on this reaction, and it may have nothing to do with the metaticulation. On the other hand, it must be remembered that the occurrence of a surface precipitation reaction at the periphery or cortex of the egg may very well, and probably does involve the production of an excess of the substance we have called ovothrombin, Cg 226. Granted, the formation of this substance and our problem is perhaps on the road toward a solution. In this connection, it is interesting to note that Heilbrunn 27 in some preliminary experiments reported evidence of an antithrombin H-key substance as being present at some stages of the mitotic cycle in the Arbacia egg. One could also interpret an experiment of Bunsbaum's in this fashion. Bunsbaum 24 states that when one subjects unfertilized and fertilized eggs to pressure, there is a decided difference in the appearance of the extrovate. The protoplasm that emerges from the unfertilized egg is full of vesicles. E.g. Enstrom thinks of these as emerging from the egg. But this is doubtless an error on his part, whereas vesicles only appear sometime later in the protoplasm. Of the fertilized egg, compare figures 14a and B of E.G. Enstrom's paper. The vesicles figured by Runstum are doubtless the equivalent of the vacuoles so often described in the previous chapter, and it is interesting to note that the vacuoles are less rax-3-edly formed in the protoplasm emerging from the fertilized egg. This might be regarded as evidence for the presence of an antithrombin in the fertilized egg, but it must be remembered that the conditions of outflow are different before and after fertilization, and the speedier production of vacuoles or vesicles in the unfertilized egg might very well be due to a greater SGG in the emergence of the protoplasm. More certain evidence that mitosis may be related to the type of reaction considered in the previous chapter is provided by the experiments of Loeb, see Loeb 13. It will be remembered that in Loeb's later studies on artificial parthenogenesis, he took the stand that all agents which cause cytolysis were parthenogenetic agents. It is perhaps doubtful if all cytolytic agents do really cause segmentation in the sea urchin egg. Many of them are too toxic. But there is a general tendency for a cytolytic agent to cause mitosis, providing that the exposure to which the egg is submitted is just of the right length. Moreover, in eggs, other than the sea urchin egg, P5, perhaps the most common method of inducing segmentation is by exposure to heat, and such exposure may very well induce the type of reaction that Loeb called cytolysis. Note, too, that in vertebrate eggs, or rather in the egg of the frog, the only successful parthenogenetic method is the traumatic method. The eggs are pricked with a needle, and as a result of this injury, they begin to divide, CG Site 7, Batoil. Injury by the prick of a needle must produce a surface precipitation reaction, and in the last chapter, it was shown that this type of reaction is one and the same as the reaction which Loeb called cytolysis. From this line of reasoning, we are thus able for the first time to bring together the facts regarding parthenogenesis of invertebrate eggs and our knowledge regarding parthenogenesis of the frog egg. Both types of parthenogenesis depend on the same type of reaction. We are also able to understand why the introduction of blood serum into a frog's egg is a more effective method of parthenogenesis than the pie yolk of a needle, which is not coated with such serum. It has been shown in the two preceding chapters that the surface precipitation reaction is closely related to the reaction involved in the coagulation of blood, and that a thrombin-like substance is involved in it. What more natural to suppose than that the thrombin present in the serum of frog blood would favor a surface precipitation reaction in the egg of the same species? Finally, it should be remembered that in plant tissue, there is an abundance of experimental evidence that wounding causes an initiation of cell division. It is probable that in this phenomenon, a surface precipitation reaction takes place in the cells directly affected by the wound, and that as a result, a thrombin-like dot substance is produced by these cells. This thrombin-like substance would then be responsible for the gelation in the cells near the wound, a gelation which is almost certainly related to the initiation of mitosis in these cells. Hence, in this type of experiment also, there is evidence that the reaction which we described in the last chapter is responsible for the mitotic gelation. Against the point of view that we have been developing, it might be argued that in normal mitosis, there is no evidence of vacuole formation within the cytoplasm. This is apparently an important objection, but fortunately, it can be met. In some experiments performed during the summer of 1927, it was found that ovothrombin could produce not only surface films and vacuoles, but also long fibrils in the protoplasm of the sea urchin egg. In these experiments, the eggs were crushed in oxalated cytoplasm, streams out, and there is no surface precipitation reaction. If now some ovothrombin is added to the side of the cover slip which rests over the crushed eggs, the protoplasm which has streamed out of the eggs is suddenly seen to form beautiful long strands or filaments. Examined under high power, these filaments appear quite hyaline, but along their length here and there, protoplasmic grannies are attached to them. The filaments or strands behave as thoroughly rigid structures. When they are pushed to one side or another by water currents of the fluid on the slide, they tend to resume their original positions. In a position of rest, the filaments are always perfectly straight. From these experiments, it is evident that the type of reaction which we have described in detail in chapters 13 and 14 is capable of forming not only vacuoles, but also filaments or strands, which bear a close resemblance to our usual concept of astral rays or spindle fibers. There is thus a considerable body of evidence that reactions of the same type as the surface precipitation reaction really play a part in mitosis. So far, the evidence is far from conclusive. Much more work needs to be done before we can be in a position to satisfactorily understand the mitotic gelation. Nevertheless, it is believed that the point of view here developed has the decided advantage that it offers a means of understanding all known facts regarding the type of agents which can cause initiation of cell division. In spite of the fact that different types of cells are stimulated to divide by very different reagents and treatments, it now seems possible that all these reagents and treatments result in a single type of reaction, a reaction which results in mitotic gelation and subsequent division of the cell.